Now, to start with the event, I would like to invite our Chief Patron and our Research Director, Dr. J.S. Yadav, for his welcome address.
Good morning to all of you, to our campus at Indra University. And we started university, actually we got approval in 2017, but academic courses started in 2018. So I'd like to welcome to our campus. And there are many people who are from far also, and from locals. So everybody is welcome. Today's, there are few name I can tell you, but I cannot tell everybody. So our chief guest today is Dr. Vinod Singh, and he's one of the leading organic chemists everybody knows, but he's a leading institutional builder. You know, I said Bhopal, when we visited together, it was there at mud. But in a very short time, one of the best ICER in India. Then we have among our guests, Dr. D.S. Reddy, and he's there. He's known from us from ever since I started doing PhD, and he's the director of our IICT Hyderabad. Then we have Raja Mannar somewhere, our very long association from almost 30 years association now, and he's the vice president of Sun Pharma. And there are many. Then Mukesh is there, who is again a very leading scientist from IIC Bangalore. Then we have Tapas Kundu, who was earlier director of CDRI Lucknow, and at present he is in JNCR. There are many. I think, if I give all this name, it will be going to only say I say that we welcome all of you, and then for gracing this place. This seminar was organized mostly, I will say that, with the efforts of Vishnu Maya, who is here. He is also a student of our Dr. Vinod Singh. And she has chosen very good speakers, starting from Europe, in India. And I request all of you to listen. You will have a future direction in this area of sustainable synthesis. Secondly, we'll have also, she will tell you some, I don't know, 5:30 around, that interaction with the student, again, with the people who are having lot of experience, number of years of experience. So you take benefit of that. Only thing I can say that I am very glad that all of you are here. Today I'm sure in this 3 days period, you will learn a lot when you go back home, and you will definitely will have a very good message. And from this outcome of this conference. Only thing that our people have tried to make your stay and also food and everything as comfortable as possible. If any difficulty, please, we have Praveen Kumar. I don't know whether he is here. You can directly interact with him. He will help you out. Then we have also administrative staff. Rupesh Kumar is there. I don't know whether he still is somewhere. So these two people will assist you if anything you need. And of course, transport and everything has been sending back to you and all this thing. So only, again, I will say that we have tried to make your stay very comfortable. I hope you'll enjoy the seminar, enjoy the locality. I'll also enjoy this forest-like atmosphere, which is pollution-free. No noise pollution, no any air pollution here. It's very this one. Only thing you will hear noise from chattering of birds many times. They are the one they become alarm to us in the everyday morning. So with these words, again, I like to thank you for coming and also wish you best of luck for this conference. Thank you very much.
Thank you so much, sir. Further, I invite Dr. Vishnumaya Bisai, Associate Professor of Department of Chemistry and convener of SIDD 2025.
Very good morning to one and all. Honorable Chief Guest, Padma Shri Professor V.K. Singh, renowned academicians and esteemed speakers, advisory board members, distinguished experts from industry, participants, invitees, and dear students. Myself, Vishnumaya, I feel deeply privileged to be the convener of this conference on sustainable innovations in drug development, SIDD 2025. And on behalf of the organizing committee, we extend our greetings from Indrashil University. Welcome, one and all. This conference on sustainable innovations in drug development is conceptualized to bring together leading researchers, industry professionals, and academicians to discuss recent advancements in drug discovery and development, to educate practicing chemists and researchers and give them a comprehensive overview and a guided path to follow. Starting from today, next 3 days in this conference, will be emphasizing mainly sustainable methodologies, innovative synthetic strategies, and academic as well as industry collaboration to address global healthcare challenges. Starting from insightful keynote lecture by Professor V.K. Singh, plenary lectures by distinguished scientists all over the world, invited lectures by accomplished as well as practicing chemist researchers. This platform will serve as a serve to transform ideas for eco-friendly and cost-effective drug development. We also have oral and poster sessions by practicing chemists and young researchers, which is going to be peer-reviewed by experts for awards by leading publishing houses like the Royal Society of Chemistry and American Chemical Society, in the name of their flagship journals, as well as oral presentation awards by Indian Photobiology Society. We are very thankful to our distinct lineup of speakers for their kind consent to participate in this event, creating value, which led us to bring on board generous sponsorship support from the ANRF, DST, then Guj Cost, Gujarat, CSIR, Council for Scientific and Industrial Research, the Brooker India, Indian Photobiology Society, Aditya Scientific, and JSK Chemicals, and more importantly, our premier sponsor, Cadila Pharmaceuticals. So apart from that, we have expert plenary lectures and representatives from Sun Pharma, virtual sessions by the Royal Society of Chemistry, and on verticals of Royal Society of Chemistry and its benefits. Noteworthy to mention here is that since we in the Shell University is educational initiative by Kerala Pharmaceuticals, our premier sponsor have some delegates, valued delegates. I would like to name them. Dr. Vinay Kumar Srivastava, he is Professor of Practice at Indrashil University, as well as Assistant Vice President, VFS Cadila, and Dr. Debesh Das, President, Branded SBU, Cadila Pharmaceuticals. So outside this seminar hall, they are exhibiting their new launched innovative products like Thrabis, Cadila Flu, Tetra, Sepsivac, Envas, Calcirol, and many more for the benefit of the direct beneficiaries like us. So further, a dynamic student-centric, student special session will be organized and we'll bring together top academicians and pharmaceutical industry leaders to guide students on future research and career prospects in the is mainly the highlight of today's event. Let's set forth to this journey of knowledge and innovation. On behalf of SIDD 2025 Organizing Committee, I welcome this prestigious gathering of scientists, academicians, and industry experts to this wonderful event. Have a memorable stay at our Indrashil University campus. Looking forward to a lively and stimulating session. Thank you so much.
Thank you so much, ma'am, for that comprehensive overview. Now I would like to call upon Professor K.M. Tiwari, Dean of School of Science and Chairperson of SIDD 2025, to share about the School of Science.
Thank you, Alisa. Good morning to all. I don't hear any response of good morning. Good morning to all. Yeah, that's the spirit. So morning should start with some energy actually. So first of all, most honorable Professor V.K. Singh sir, Institute Chair Professor IIT Kanpur, our Research Director, Mentor Professor J.S. Yadav, dignitaries on the dais, off the dais, all the delegates, invited speakers, our Registrar Dr. Karthik Jain sir, Dean Research Dr. Vijay Singh sir, all deans, head of the departments, my faculty colleagues, and most importantly, all dear participants. It gives me immense pleasure to welcome you all in our beautiful campus of Indrasil University for this first international conference, that is Sustainable Innovations in Drug Development, SIDD 2025. First of all, I would like to thank all of you to accepting our invitations and coming to our campus. As Sir rightly mentioned, Indrasil University was established as an educational initiative by Cadila Pharmaceuticals Limited by our Honorable CMD Dr. Rajiv I. Modi sir in the loving memory of his parents. The main objective of Indrasil University is to foster academic and research excellence in the area of science, pharmacy, engineering and technology, and management. Along this line, we have 4 schools in our university: School of Engineering, School of Science, School of Pharmacy, and School of Economics and Management. In the School of Engineering, we are offering programs like BTech in Computer Science and Engineering, BTech in Chemical and Biochemical Engineering, BTech in Mechanical Engineering, including programs like BSc IT in BCA. Similarly, in the School of Economics and Management, we are offering programs like BBA and Postgraduate Diploma in Health Management. In the School of Pharmacy, we are offering programs like B Pharmacy. Importantly, School of Science, we have 2 departments: Department of Chemistry, which offers program like BSc Chemistry, MSc in Chemistry, specialization Organic and Analytical, and PhD in Chemical Sciences. Similarly, Department of Biosciences offer programs like BSc Biotechnology, MSc Biotechnology, MSc Microbiology, MSc Zoology, and PhD in all biological and life sciences related domains. All UG programs of Indrasil University are fully aligned with National Education Policy 2020 with choice-based credit system, having a plenty of credit choices. Our all PG programs are completely aligned with industrial requirement with the 6-month complete internship at industry, which is the purpose of actually learning and outcomes. We have signed MOU with several academic and research institutions like IIT Guwahati, Central University in Gujarat, along with several CSIR laboratory like IICT Hyderabad, CSIR NCL Pune, CDRI Lucknow, and CMCRI Bhavnagar, in order to boost our training, internship, and placement opportunity to our students. We have also signed more than 45 plus MOUs with our industry partners in the area of chemical, pharmaceutical, agriculture, petrochemical, and other related materials where our students are going their internship and training program and getting placed. Our research infrastructure facilities has been designed as per industrial requirement, as it is a baby child of Cadila Pharmaceuticals. So our all faculty members are PhD from premium institutions like IIT, CSIR labs, with most of them have a postdoctoral experience from abroad. So our research, we are being mentored by Professor J.S. Yadav sir at Inaugural University. He is looking after all research and academic activities. And we are very happy to share we have received 8 extramural grants by our faculty members by various funding agencies like DST, Guj Coast, GSBTM, and many more than 3 crore rupees just in 6 year of span of time. We have also received 2 very important grants for infrastructure facility development like DST Feast and Gujarat Commissionerate, totaling amount more than 5 crore Indian rupees. So these are the achievements. Our faculty members are actively engaged in research and development activities. And based on that, Inaugural University, just span of 6 years, has published more than 460 publications in Scopus Index Journal, and with more than 15 patents in this short span of time. 2 of our faculty members, Professor J.S. Yadav and Dr. Vijay Singh, recently featured in top 2% scientist list released by Stanford University, which is a great achievement for our university. So coming to this conference, chemistry, as we know, it has been recognized as one of the integral part in addressing the issues related to Sustainable Development Goals, SDG, which are decided by UN in 2015. So starting from agriculture advances to water purification techniques or creation of eco-friendly materials, maybe chemicals, pharmaceuticals, or biochemicals, chemistry has been providing a useful information and technology that are needed to address the sustainable concerns. Our subdomain of chemistry, that is medicinal chemistry, we are going to talk about, is very much dedicated towards drug discovery and development. And it is a global cause where we are all working for the well-being and health concerns. So as we navigate away our journey towards sustainable future, we strongly believe that the discussions like this forum is going to provide a great opportunity to all of us to learn the challenges that is coming on the way towards drug development and how we can find long-lasting solutions for these kind of problems that is coming. It's not only about making the products, but it about making the products which does not impact our environment, which should be economical, which should be beneficial to the common public or human society. So this is the main theme of this conference, where we are going to discuss many lectures are going to be delivered based on this sustainable innovations in drug design and development. Importantly, I urge all participants, especially young participants, to basically engage and interact with our panel members, all our experts, and listen all the lectures, deliberations, so that you can decide your career pathway in a very nice and useful way, where you can have your skill development in that particular area. So I invite all the participants also to visit our labs, interact with our faculty members, to see our infrastructure facilities. And if you are working for any specific research or project, we are very happy to offer all the helps, whether it is technical or any kind of scientific expertise. All our faculty members are open to discuss and help you all. So I wish next 3 days we are going to have a very wonderful session and a memorable stay to all our speakers and delegates, guests to our campus. And I also wish to congratulate all the poster and oral presentations who are going to happen in next 3 days. I wish all the best to all the presenters who are going to win the awards in different categories. And at last, I wish to thank you all for coming to our campus. Thank you very much.
Thank you very much, sir, for the insights. Now I invite Dr. Paulami Sen Gupta, SIDD 2025 Organizing Committee member, to introduce our distinguished chief guest.
Good morning. I feel hugely honored and privileged to introduce today's chief guest, Dr. V.K. Singh. Professor V.K. Singh is a professor emeritus and holds an institute chair at IIT Kanpur. He also serves as the director chair professor at IISER Bhopal and has played key leadership roles in various scientific and academic institutions. Currently, he is the chairperson of the Research Council of CSIR M3 Bhopal, the chairperson of the Recruitment and Assessment Board of CSIR, and the chairperson of the Governing Council of the DST Institute of Advanced Study in Science and Technology, Guwahati. Additionally, he is a member of the CSIR Society, chaired by the Prime Minister of India and responsible for mentoring CSIR AMPRI and CSIR IIIM Jammu. Professor Singh is also the president of National Academy of Sciences, India, that is NASI. His research primarily focuses on synthetic organic chemistry, particularly in the area of asymmetric synthesis. His noteworthy contributions include the use of gem diphenyl-based pyridine bioxazolene ligands for a wide range of reactions and simple organic molecules-based prolinamide organocatalysts. One of the organocatalysts developed by him is popularly known as Singh's catalyst, which is one of the best and highly efficient catalysts known in the literature for asymmetric aldol condensation reaction. His distinguished contributions towards science, research, and society is recognized in the form of several awards, including the Swarna Jayanti Fellowship, the Shanti Swarup Bhatnagar Prize, and the Padma Shri in 2014. As you all must be knowing, Padma Shri is the fourth highest civilian award by the Government of India. Professor Singh has also been elected fellows of National Science Academy, the Indian Academy of Sciences, National Academy of Sciences, the World Academy of Sciences and all. And he has contributed significantly in academic publishing, serving as the editorial advisory boards of Asian Journal of Organic Chemistry, Tetrahedron Letters, Organic Chemistry Frontiers. He was the editor of Tetrahedron Letters, associate editor of Synthetic Communications, and a member of editorial advisory board of Organic Letters and Journal of Organic Chemistry. He has held several prestigious positions. I would mention the founding director of IISER Bhopal for 10 years. He was instrumental in setting up entire campus. He also served as the mentor director of IISER Behrampur, director of SPA Bhopal in additional charge, and the chairman of BOG of several renowned institutes. Professor Singh has served as member of the Scientific Advisory Council to the Prime Minister's office and was the president of the Chemical Research Society of India and chairperson of the Governing Council of ISS Kolkata. His leadership, both in research and institutional development, continues to shape India's scientific and academic landscapes. And with this, I welcome sir on the behalf of organizing committee, and he has been my sir too. He has taught me Chem 402 in MSc second semester. Thank you, sir.
Thank you, Paulmi. My speech will be shorter than the Paulmi's introduction. Dr. J.S. Yadav, Dr. Vishnu Maya, Dr. Tiwari, and other guests, I'm very happy to be part of this symposium titled Sustainable Innovations in Drug Development Program. Quite apt and suitable for Inaugural University because it is connected with the industry. I'm already giving a talk just after tea. I was thinking maybe my talk could have been this time. If I talk here too much, I'll not be able to give a talk in my keynote address. But I must say that I'll reserve my speech or talk, whatever I want to say, for my keynote address, which is kind of a general talk. Vishnu Maya invited me to give a talk. I politely declined. I said I'll come and spend 2 days and listen to the talk, mainly because I do not work directly in drug discovery. And number two, I somehow didn't have enough time to make slides, to be honest. And then now you can see that I'm now giving a talk as a keynote speaker. So one thing I like that drug discovery, wherever I go, and there's a symposium titled Drug Discovery. Any symposium with drug discovery is incomplete if Dr. Raj Mannar is not there. And I tell you, I have a lot of respect for him as a great person working in drug discovery program. Everyone knows it. Of course, there are others who work it, but he's one of the key person, and that is going to add beauty to this symposium. Drug discovery program actually is a very important thing for any country. But it should not be a lip service. It should be a real drug discovery program. It should not be like writing a proposal that I'm synthesizing this molecule for fungicide, herbicide, and it ends there. It has to go to the next step. Even people who do synthesis, they do synthesize biologically active molecules. But they must take those molecules to the next step. If they are not going to take it to the next step, I assure you, in India also, the funding will be a difficult problem. Whether it is drug discovery or materials or anywhere, time has changed. You have to bring innovation in your research so that the government can fund it. Even industry will not fund it unless you get into real worth of the fundamental research one is doing it. And that is why this symposium is very, very important. Also convey to young people and students especially, drug discovery is nothing a different science. And I don't want to say too much, otherwise I'll not be able to say anything in keynote address because it will be repetition. Unfortunately, I had planned to stay 2 days, but because of some emergency, important work, I have to leave tomorrow morning. I apologize for this. I feel very ashamed and guilty when I have to leave the symposium quickly. Nonetheless, I promise I'll visit Inaugural University and spend a couple of days to interact with the students and faculty. And I convey my best wishes for this 2.5 days deliberations on drug discovery, and I wish all the best for the success of the conference. Thank you.
Thank you, sir, for your insightful and inspiring inaugural address. We now proceed to another significant moment of today's ceremony, the release of the SIDD 2025 Souvenir, which captures the essence of this conference. For this, I would like to request the dignitaries on dais to please proceed with the release of the Souvenir SIDD 2025. Also, I would like to request Dr. D.S. Reddy, Director, IICT, and Dr. Sriram Kanva Gudimada, the HOD Chemistry IIT Gandhinagar, who are also our valued SIDD Advisory Board members. Further, I request Dr. Manjul Joshipura, Vice President.
Okay.
In absentia.
Yeah, unfortunately, he is under weather, so Dr. Manjul Josipura is not, he has not come. We would like to request Dr. Rajamannar, also non-executive director, Sun Pharma, to come on the stage. Dr. Vinay Kumar Srivastav, Cadila Pharmaceuticals. Dr. Deves Das, Cadila Pharmaceuticals. And our registrar, Sir Dr. Karthik Jain. Yes, sir.
With this, the souvenir is officially released. I request the dignitaries on the dais to take their respective seats. To access the soft copy of this, one may scan the QR code on the cover page of the notepad shared in your welcome kit. Alternatively, visit our website to see that. Now, I invite Dr. Shelly Rana, co-convener of SIDD 2025, to deliver the vote of thanks.
Good morning, everyone. It is indeed a great honor and privilege to extend a vote of thanks on behalf of the organizing committee for the inaugural session of the Conference on Sustainable Innovation and Drug Development 2025. Myself, Dr. Shelly Rana, co-convener, SIDD 2025. At the outset, express my deepest gratitude to our chief guest, Padam Shri, Professor V.K. Singh, for gracing this occasion with his esteemed presence. We look forward to your kind guidance throughout this 3-day journey of this conference. A sincere thank you to our distinguished industry expert and renowned academy expert for agreeing to participate, the educators toward innovation in drug development. We are immensely grateful to our sponsor, Kerala Pharmaceutical, DST ANRF, RSC, ACS, Indian Photobiology Society, and Aditya Scientific Industry, JSK Chemicals, and Holy Scientific Sponsor for their generous support. Your contribution has been instrumental in bringing this event to life as evident by the grand presence of this hall. A special thanks to our Provost Dr. DJ Shah, Research Director Dr. J.S. Yadav, Registrar Dr. Karthik Jain, and Dean of SOS, SOE, and SOP, whose leadership and constant support have been crucial in the organizing this conference. I also extend my heartfelt appreciation to Admin Mr. Paveen Kumar and Financial Officer Mr. Harshit Gandhi for their encouragement and timely support whatever we need. A big thank you to the core organizing committee member whose dedication, planning for their and tireless effort have assured the seamless execution of this event. Our sincere appreciation goes to our chairperson, convener, and all faculty members of School of Science for their commitment and hard work in shaping this conference. To the student of the School of Science and all enthusiastic participants, your curiosity and engagement are what make this event truly meaningful. We deeply appreciate your active participation. Lastly, I extend my gratitude to every individual directly or indirectly involved in making this event live. We look forward to an insightful and productive conference ahead. Thank you all. Everybody requested to be stand on their side for the national anthem.
With this, we are all set to start our technical sessions. Before that, we cordially invite you all for a very short tea break outside the seminar hall. We request all of you to join us again sharply. हाँ मतलब मैंने वही बोला ना Everybody please stop. Be seated. Everybody please wait inside.
Yes. We are sorry for the inconvenience. We now move on to our first technical session. To chair this session, I would like to invite Dr. J.S. Yadav sir and Professor G. Mugesh sir. First, our first talk will be delivered by our chief guest, Padma Shri Professor V.K. Singh. For that, I would like to invite Yadav sir to introduce and invite our keynote speaker. Yeah. We request Yadav sir to felicitate our chief guest with a sapling. I would like to request Professor V.K. Singh to come over the rise.
Only thing is, I I don't think Dr. VK Singh needs any introduction. Already Palomi has given. Without taking much time, I will just invite. Only thing I will say that we are all Guru Bhai. Elder one was Govardhan Mehta, then I am in middle one, then he is youngest one. Vinod Singh. तो हमारे यहाँ बोलते हैं कि बड़े वाले का तो बाप होता है, छोटे वाले की माँ होती है, बीच वाला का माँ बाप कोई नहीं होता. So it is like that. So he has mother, my elder brother has father. I am in between this. So with this, I invite Dr. Vinod Singh to deliver his lecture. I think there is a caller. Something. You need caller mic? Yeah, something is there. Hello.
Working? Okay. At the outset, I again thank Dr. Vishnu Maya for the invitation. As I mentioned, I do not work in drug discovery program. I understand a little bit. Then I thought to talk something because most of the speakers I saw, they're synthetic chemists, and several of them synthetic organic chemists. Mugesh, I always consider synthetic organic chemist because he did his postdoc with one of the few best uh, synthetic organic chemist, Professor Nicolau. And somebody doing PhD in a different area and doing postdoc in a totally different area, you need several times bigger skill to achieve the goal. The reason I wrote the title, Creativity in Organic Synthesis and Implications under Discovery, I believed, based on my experience, and more than that, almost 40 years ago, I met a person from industry in US, and I was involved for a brief period of time working in a pharma companies. And then when they were trying to hire people for drug discovery program, so I asked a question, what kind of person you are looking at? The person was a senior vice president, and he said, we don't hire medicinal chemists for our drug discovery program. We want to hire a person who is exposed to synthetic organic chemistry, who has dealt with complex molecule with complexity, who understand stereochemistry. And unless somebody understands synthetic chemistry, especially synthetic organic chemistry, he or she may not be a good medicinal chemist, because it takes only a few months to understand the jargons of medicinal chemistry. And my statement, what he made, what I believed in all along, and you see that some of the people, I mentioned Raj Mannar, he worked with a very good synthetic organic chemist, and so as several other people you see in the audience. I don't want to name it because I may miss some names. So synthetic organic chemist has great impact in drug discovery program, especially if you look at 20 years ago, 30 years ago. Even if biologics are coming, but the importance of organic chemist or synthetic chemist will never be diluted. I used to say, if somebody doesn't know PK, you cannot be a good medicinal chemist. This is my habit, and because the whole biologics you see, your temperature goes 1 degree up and down, creates havoc. Your pH goes little bit up and down, it creates havoc in body or outside body. So in half an hour, I just try to, it's all general slides, so you'll not be bored. There's no equations, nothing else. I have only few slides about our work. Organic synthesis embellishes drug discovery and material chemistry. What I'm trying to say, the organic chemistry has connect with other branches of science, chemical biology, medicinal chemistry, materials. So the role played by organic chemistry in pharmaceutical industry, it will continue to be one of the main drivers in drug discovery program. This is the message I want to give. It's not working. Huh? Can I use, I have my slider. पहले लेक्चर में बैटरी डाउन हो गई. Achha. No, it's okay. I have. When you go to war, you have to well equipped. What now? Okay. So there are a few slides about the drug. I'll give you some data about the drug discovery program. As you can see, the approved drugs per disease area for 10 years. And if you look at more than 50% of the overall number of drugs for oncology, infection, and central nervous system. So these are, and remaining 50% comes so many things which are here. Okay. Now you have a new molecular entity where synthetic chemists are involved, biological license application. What happened? That was not my slide anyway. Biological license application and biosimilars. If you look at the last 10 years, आप क्या कर रहे हैं? हैं? Something is. Can I use my laptop? Hmm? Okay. 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 So if you look at for 2010 to 2019, there are around 289 new molecular entities, and biology is playing a big role these days. So 89 biological license applications and 27 biosimilars. If you look at top 10 prescription drugs, number 1 is biological license applications. Again, number 2 to 3, you see new molecular entity. But if you look at number 4 to 10, this is biological license applications again. So there are large number of biologics coming in drug discovery program. So is there a challenge to synthetic chemists? The answer is no, because changing landscape and requirements from organic synthesis, only landscape is changing, workload is not going to be less. If you look at the top 5 drugs by retail sales, the data I got from here, the 2 drugs, the biologics, for example, the top seller, like Humira and Keytruda, close to $20 billion and $14 billion, made a lot of money. But about 3 drugs, where the medicinal chemists have played a great role. You can see they are there, but the amount of money it generates is much less than the drugs coming from biologics. So there is a new demand from organic synthesis. For example, you have biological license application drug, but again it has a linker. And these linkers can be synthesized only by synthetic chemists. The role of synthetic organic chemists is not going to be less, even if more and more biologics are coming into the market. And this is what I'm trying to emphasize, that synthetic organic chemistry or synthetic chemistry overall is going to be very, very important. If you look at the natural products, of course, natural products have played a great role in drug discovery program. There are several natural products used as a drug. Sometimes natural products, for example, if you look at here, anti-cancer drug discodermalide, you cannot isolate in large amount to study for the clinical trial and all that. So the only way to procure large amount is by synthesis. So one has to synthesize these molecules. Large number of people have synthesized this molecule, including Dr. J.S. Yadav, which I'll show you the next slide. Not next slide, somewhere after this. So the role of synthetic organic chemist remains same as it was earlier. The only thing is they have to make sure they have to be identified by telling people that we are important, we are doing something for the society. Now, synthesis, we are talking about synthesis. Synthesis is a history. Everybody knows urea was the first molecule synthesized, that is here in 1828. And synthesis of urea proved the ability to synthesize natural products. And from urea till now, organic synthesis has created a history. 1828 urea, and you see a complex natural product, azadirachtin. Azadirachtin, I wrote somehow, I like the molecule a lot. It's isolated from name tree, so anti-infident property. And experimental science remains to be important science. As you can see the quote from Richard Feynman, what I cannot create, I do not understand. So synthesis, or not only synthesis, anything, you make it, then only you understand its importance and its value. Now, since I use the title Creativity in Organic Synthesis, and when you talk about creativity, the creativity starts with the man, Professor R.B. Woodward. The person behind creativity in organic synthesis is he. He had a lab in his basement when he was very young. You don't have to attend all the classes. You can see that he skipped 4th, 7th, and 10th grade, enrolled in MIT in 1933. Okay? And the first paper in 1934, he was only 17 years old. So many things you know about him. He was kicked out of MIT in 1934 because he did not attend classes or did not pay enough importance to the coursework. Again, he re-entered in 1934. Somebody requested him to come and attend it, and then achieved a BS in 1936, PhD in 1937, and he joined Harvard as assistant professor at the age of 20. Okay? And slept only 3 hours a day. Okay? And he's a photograph. The reason I like him, because I got attracted to organic chemistry only reading the biography of R.B. Woodward. I have read most of the articles written by people about him. All kinds of unique character, giving lecture 3 hours, 4 hours, doesn't give lecture without having a wine or whiskey in a glass. You can see that. And it's not that you will become Woodward by doing all these things. When he light the cigarette, people say he never used matchbox or lighter because he was chain smoker. But he was a great mind, and he completed large number of total synthesis. And that is why if you read articles about synthesis, people say it's Woodward era. Okay. Then Organic synthesis is a well-thought endeavor. It is not that just you come and start doing. There is science behind it. Sometimes people understand science. Sometimes people do it without understanding it. But there is a well-thought science. It's not a random walk. You have a starting material. You have a product. And synthesis, unless your synthesis is efficient, nobody is going to pay attention. Most of the top synthetic organic chemists, they talk about efficiency. I'm not going to emphasize whether it's a linear synthesis or convergent synthesis, but it has to be high yielding. Industry people will not look at if your chemical yield is low. Okay? So that lot of people don't give importance, but yield is very, very important for industry. Not picking up. Then Core era came, and he started talking about retrosynthesis. It is not that people did not know retrosynthesis. People did have a planning, but maybe it was not formatted in a nice language where it can be said that this is a retrosynthetic analysis. So finding the most economical paths to complex molecules, I have just given a molecule, very complex molecule. By slow dissection, you can come with a simple precursor. By looking at you, it is just like when I go to any city and I see that electric wire and lamp and the poles. And you may think how these guys come and repair which wire belongs to which house. Okay? The guy does retrosynthesis only. He picks up from top and try to find out which house is connected. So same thing happens when you look at any complex molecule. If you start dissecting it, you will find it is much easier to find a solution, find a starting material for the synthesis. And the work is only because Professor E.J. Core, who got Nobel Prize in 1990. Now, I only wrote synthesis of prostaglandins. See, so many people synthesize prostaglandins. Must be, I cannot tell you the number, but most of the top organic chemists synthesize prostaglandins. And you can see the molecule is very, very complex in terms of stereochemistry. But it's only Professor Core's synthesis is more flexible, economically viable, and this is the path industry uses because my prostaglandins are used as drugs. And that is why the famous Core lactone, as you can see, and this most of the synthesis done by industry, they have Core lactone. These R and all that may differ, that's not important, but they follow the similar strategy to synthesize and the prostaglandins. So there's a great if you look at here, you see the Woodward synthesis is very creative, very nice synthesis, but it's not practical. Industry will not be able to make prostaglandin following Woodward synthesis. But Core synthesis is very, very viable. So what Core has to say, chemist who designs and completes an original and aesthetically pleasing multi-step synthesis is like the composer, artist, or poet who, with great individuality, fashions new forms of beauty from the interplay of mind and spirit. So people who do total synthesis or do any complex synthesis, whether natural or non-natural product, they are more like artists. Okay. You know, it will be incomplete if I don't show the slide of some of the Indian organic chemists. Everybody knows Professor Govardhan Mehta, who is a good synthetic organic chemist. He's not good synthetic organic chemist because he synthesized large number of molecules. He's known because of the strategy he used in synthesis of natural products. And the strategy he used, although this is not a correct thing, you cannot do synthesis without reagent, but some of the things, synthesis without reagent means some of the intermediates. Today you talk about sustainability, green chemistry and other things. You can see Diels-Alder reaction, 2+2 cycloaddition and metathesis. In 2-3 steps, you get intermediate which can be converted into various natural products containing cyclopentane ring. And that was the era of cyclopentane chemistry. So large number of cyclopentanoids were synthesized. Several people did it. Professor Mehta was one of them. And this strategy, he became famous only because of the strategy he used in the synthesis. And of course, this is a review article. You'll see a lot of work. And this is Professor Mehta's paper. You know, Dr. J.S. Yadav, I remember when I was a PhD student, he joined NCL Pune. And he was doing synthesis left and right. Those days, large number of pheromones I could see synthesizing. I used to read most of the papers, you know. And some of the complex synthesis, which he did while being at ICT, Indian Institute of Chemical Technology lab, where you are director. And you can see all these molecules. I was talking about discodermalide, camptothecin, artemisinin, the anti-malarial drug, all those things done at ICT and under his leadership. And all these syntheses make people perfect to understand anything you want to understand. That includes your drug discovery program. I thought if I'm giving a talk, 1 or 2 slides, I should have my work. I also did some synthesis, but I'll just show you one synthesis. This molecule, boronolides, it has anti-malarial property. We synthesized this molecule. Of course, initially we published in 2000, and since synthesis used to go slow because of the constraints, we published the full paper in 2003. The good part is the strategy part. The synthesis is very flexible. In today's world, if you cannot have a flexible synthesis, it is not going to work out. So from readily available material, you can synthesize this, can be converted into intermediate, and which can be used to synthesize all forms of boronolide. Okay? And I found that synthesis is very good, appealing. Although there's no new method developed, but synthesis is quite flexible. The synthesis which I like the most from my lab is this one. I don't care about anything. I get very excited when you synthesize anything in one step. Okay? You add one thing and you get a product. And industry want that. And we could synthesize this kind of molecule depending upon what R you have, is an anti-tumor drug muractacin, and also the length of side chain. What I found in literature, the length of side chain is crucial for biological activity. Some of the short chain homologa flavor constituents in wine. Okay, you synthesize this molecule from sugar, and then you add something, acid base, whatever required, and you get this compound. So synthesis, most of the people here, they are good synthetic organic chemists. There are hazards also in synthesis. And sometimes, not sometimes, most of the time it leads to frustration. And there's a Wikipedia, an extract of this cockroach plant, this natural product. Some student committed suicide, and it seems the stress is connected with the suicide, which I kind of don't approve, we don't agree, because this also gives you training to absorb the stress. And one should absorb the stress, otherwise it is not very good for health. Short synthesis. In today's world, if you do a multi-step, 30-step synthesis, 50-step synthesis, nobody is going to look at unless you are the first person to synthesize it. Okay. And this synthesis, why I posed? Because in one step, this natural product, the alkaloid, was synthesized by Hitchcock. As you can see what he does, he synthesizes this starting material, adds in methylamine, acetic acid, and keep on moving the arrow. And as you can see that this skeleton was synthesized in one step. So short synthesis is very, very important. If you want to develop your career in synthesis, your synthesis has to be very, very short. The other molecule during COVID period, everybody was talking about remdesivir. It's a broad spectrum antiviral drug. Gilead actually synthesized this molecule. As you can see, it's published in Nature and Journal of Medicinal Chemistry. Complexity because of chirality also. Chiral drugs, it becomes more challenging to synthesize them. And one since appeared in 2021. As you can see that in one step, this was synthesized. So this you can say the short synthesis. And since I'm not going to talk about steps, so I'll leave it here. If anybody has interest, you can read it. Because here the chiral center is the phosphorus and this carbon. And that has been done just by using this 20 mole % catalyst and some base and paratolys, sulfonic acid and all that. So today's time is one has to be innovative so that one can synthesize molecule in one step. So what I'm trying to say, creativity in organic synthesis will continue to impact the drug discovery program. Why I say drug discovery program? If you look at most of the top medicinal chemists, they had very strong training in synthetic organic chemistry. I'm talking about worldwide. You take any big pharmaceutical company, that will be the case. And this will happen only when a chemist has a broad understanding of properties and conformation of molecules, extensive knowledge of reactions, and stereochemistry. When all these things you are good at it, whether you are done a synthesis or not, you will be able to achieve the goals. If you look at the Nobel Prize, you know the history about Barton. The first paper for which he got Nobel Prize is published in Experientia. And this is again the conformational issues. And I believe the conformation, if we did not know the conformation, we may not have done so many things in the organic synthesis. So this contribution is very, very important. Reagents, we talked about reagent. Today, if you don't know the boron chemistry, half of the organic chemistry will not be done, especially asymmetric synthesis. So sodium borohydride reagents and things like that. So Professor S.C. Brown contribution and Professor Wittig contribution about the Wittig chemistry, very, very important. Metals in organic chemistry. These days, a lot of people are doing metal chemistry. And if you look at the coupling reactions, the Heck, Negishi, Suzuki, they got Nobel Prize in 2010. Palladium catalyzed cross-coupling in organic synthesis. This has played a great role in the development of drug discovery program. See, industry is not disconnect with chemistry. Whatever you discover here, it actually gets translated there in the industrial lab. Similarly, Graf-Stock, for metathesis reaction, they got Nobel Prize, and that has also played very important role in organic synthesis and indirectly drug discovery program. It's not working. The third thing I talk about is stereochemistry. Today, if you synthesize, if you do a racemic synthesis, nobody is going to look at it. Without chirality, nobody wants to look at any molecule. And stereochemistry is very, very important. And one slide I have just to show you the importance. You can see the molecule cartinostatin. It has this, you look at this chiral center. It has nanomolar activity if hydrogen is alpha side. If it goes to beta side, this becomes inactive. You bring in carbon-carbon double bond, make the molecule flat at this side. Again, it regains nanomolar activity. What we are trying to say, the stereochemistry plays very, very important role. And I'll just show you one or two slides about our work. And then before that, I talk about Nobel Prize work done given in asymmetric synthesis. Sharpless, Naori, and Knowles, they got Nobel Prize in 2001. Sharpless for sharpless epoxidation chemistry. Today, if you look at the sharpless epoxidation chemistry, you don't find any challenge. Not many people may be using it because we have other methods, better methods. Because you have to add, you need allylic hydroxy group, and then you do epoxidation, then remove the hydroxy group. So basically you are wasting 2 steps. But remember, time is the most important thing. 2001, when nothing was known, that played a great role. Industry used a lot, and that is why this reaction became very important and he got Nobel Prize. Nauri and Knowles, who was in industry, for hydrogenation reaction, ruthenium and rhodium catalyzed hydrogenation reactions. The time when I was doing PhD, I think Dr. J.S. Yadav can tell the correct thing. Menthol, I think India was exporting. But after this discovery, I don't think camphanolide product process will be economically viable. I don't know the history, but definitely, I see. But Japan actually came in a big way, and I think this becomes economically viable. So what I'm trying to say that it has played an important role in drug discovery program or in a chemical synthesis. And after 20 years, again, these 3 people got Nobel Prize for asymmetric organocatalysis, because asymmetric synthesis has played important role in drug discovery program. And in our lab, I just show you a couple of slides before ending the talk. Long 2006 for enantiocyclic propagylation reactions. Okay, and we got 99% E, and the 99% E is only because of DEM diphenyl group, and that's why we use this ligand for large number of reactions. Then after several years, Vishnu Maya carried out same thing, intramolecular reaction. As you can see that you do the addition, you zip in with any acceptor, electrophile, and you can synthesize this kind of isoindolinone and other kind of heterocycles. And we've used in synthesis also. This becomes a very nice method to synthesize chiral compounds. 5-membered was very important because not many methods were known to synthesize this kind of chiral compound. 6-membered, of course, there were methods, but 5-membered number of methods were less. So this kind of reactions also become very, very useful. Then we developed this catalyst for aldol reaction. The time we developed ours was the 3rd catalyst and more practical one because it works in aqueous medium and also several steps can be done in one pot, and turned out to be very good catalyst for enantioselective aldol reaction, commercially available, very well used in synthesis. And we never did any computational work, but somebody did work only on our catalyst and reactions, published a paper in 2014, and the model was supported by this DFT. And Berkesel published a paper, large-scale synthesis of zinc catalyst. And there's a reason I brought this slide. One journal I used to read a lot before coming to India in 1999, joining IIT Kanpur, is Organic Process Research and Development. You all will laugh it because I am fond of reading experimentals, and any experimental written in journal, you can 100% believe in it. So he synthesized our catalyst in large scale because he was not able to, he said our method was not very clean in the sense to cleave the N-Bog group. We used formic acid, and when they were using formic acid, they were getting some clean, some not clean, cleaved product, and so they used dilute HCl. But when we repeated our work in our lab here, after several years, we didn't have any problem. So the culprit was formic acid procured in Germany or in India. So, so basically he replaced formic acid by dilute HCl, and he told me he made this one 80-90 grams of this catalyst and used in synthesis a lot. And this catalyst is used by Aldrich also and very useful. There's a review article, a lot of applications are there. So what I'm trying to say that I just want to convey that synthetic organic chemistry is very, very important. Creativity is important. A lot of the people question what is originality and creativity. See, something could be original. Anything you do creative, it is much more important. And as the title says, sustainable innovation, it has to be sustainable. And unless your work is sustainable, industry is not going to look at it. A lot of people publish fancy reagents, industry just throws in the dustbin. A lot of people complete total synthesis with very poor yield, industry throws in the dustbin. So synthetic organic chemist or any chemist, they have to repurpose themselves to the need of industry, to the need of the society, so that the funding continues. And drug discovery is highly connected with the organic synthesis. With this, I like to close it. Again, I thank past and present students, and all my work has been supported by DST CSIR. And thank you for patience. Thank you. This is
I think you agree. It was a fantastic lecture. You can entertain only one question. Later on, you can grab him in tea time. One very important quick question or suggestion. Both clear. Eh?
Yeah.
Chalo, हाँ.
How is machine learning AI is going to influence
Well, see, whatever you say, a lot of people develop software, this and that, but still you have to flame dry the flask, do the reaction, and all those things. AI will not do it. Of course, a lot of new methodology, technologies have come in. But still, I can give you an example. There are several companies. One of the names San Antonio in Texas. The name of the company, near San Antonio, the name of company I'm not able to remember. To discover drug using computational methods. It's very difficult. Again, as you see, you have to make molecule. Computer can guide you and help you, but cannot discover a drug. Even combinatorial and all that, it can expedite it, but it has not helped that much as it, this is my, I don't know, maybe Rajmanar can tell when he gives a talk about more about drug discovery. It's too complex. A lot of time you need intuition, which you cannot rationalize, but you feel it's going to work or not going to work. And that comes based on experience and some third order knowledge or something like that.
Okay. Now, in the nutshell, computer cannot replace our human brain.
Yeah.
That's what it is. One time, I'll tell you, one particular reaction, we got it accidentally. Then we kept on putting AI and all those things. Ultimately, it worked for maybe an hour, and ultimately he said it's too late in the night. I will take some drink and sleep rather than solving this problem. Why it said like this? Funny way. Because they have no solution. What we got accidentally, unprecedented. So it is quite common that way. The things which human can think they can. Of course, this is a tool. AI, ML, whatever you call it, it's helping.
Yeah, it can help it, but
still there is a disconnect between toxicity and our receptor. So that's why it is difficult.
You have some.
Anything else?
Oh, AI question everybody asks these days. No, it's a good. No, it will help. It will help. I'm not saying that. But in the end, somebody sent a video, or somewhere I read in Twitter about the school education system, the teaching methods, like, he said still you need that. If you don't draw a molecule, 6-membered chair, and only you look at the video, it becomes very difficult to draw it in exam. So I think maybe I am a classical organic chemist, so not able to think. But,
Okay, thank you. A lot of talk earlier also, artificial intelligence, now it has become more prominent because the more data is available. Earlier, that much data was not available. Now, more data automatically they are picking up from the literature and they are able to predict something. So it is all depends upon data, and data what we collect here may not be there. So that is also is. Anyway, Dr. Vinod has given very comprehensive. So all synthetic organic chemists who are ever here, so you will not lose a job, okay? That's what he's trying to say. If any drug discovery, you need a synthetic chemist, okay? Maybe landscape may change, maybe biological or whatever it is, but there also you need a linker or all kind of thing. Even your vaccine cannot be without doing adjuvant linker and all those. So organic synthesis will continue as long our universe is there, okay? That much I can tell you. And they won't be without a job. So you are in a good business. That is one thing. So I'd like to thank, we have something from, so I'd like to thank Dr. Singh coming, and I don't know whether you have visited that statue of Keynote.
Thank you so much, sir, for the insightful lecture. And we are extremely sorry for the technical glitches. Now I invite Professor G. Mugesh to chair the next plenary lecture.
Talk. Dr. Rajamanar holds an MSc in chemistry from the University of Madras and a PhD in organic chemistry from IIT Madras. He did his postdoc at the University of Zurich. He began his career as a scientist at Speak Science Foundation in Chennai before joining Sun Pharma. I think he joined Sun Pharma in 1993. So he has over 250 patents, 38 publications in various international journals. Interestingly, his team has developed processes for 200 pharmaceutical drug substances, including novel synthetic routes for many of them. His work mainly focuses on therapies for allergy, inflammation, respiratory diseases, oncology, diabetes, obesity, immunological disorders, and also addressing critical unmet medical needs. Dr. Rajamanar currently he holds a professor of practice at Doon University, Dehradun, and he was honored with the J.C. Ray Oration Lecture at the Indian Institute of Chemical Biology. That was in 2023. And he is also recipient of the Sir U.N. Brahmachari Award. He has held several prestigious positions. So he was a chairperson of the Research Council at CSIR IICT, Hyderabad, chairman of the Academic Planning and Development Committee at NIPER, Kolkata, member of the Research Council at CSIR NCL Pune and CSIR IICB Kolkata. He also serves on the board of governors at NIPER Mohali and CSIR expert committee member for drug discovery research. There are many more recognitions, so which I would not like to give elaborate introduction. I take this opportunity to invite Dr. Rajmanna to deliver his talk.
Very good morning. Thank you, Professor Magesh, for the nice introduction. Thank you, Professor Jadhav and Vishnumaya, for inviting for this conference. I am privileged. I'm an organic chemist, but the last more than 3 decades, I'm in industry. I thank Professor Vinod Singh for the excellent previous talk. So I will not be talking much of the chemistry. I will be talking of what industry looks. When you talk of innovation, I had seen just now Professor Indrandan Modi's remarks, it needs to bring value to the last common man, affordable medicines. That's what it is. So anything we say that there should be economic growth, there should be an impact in the society. That's the innovation. And sustainability is that what is that innovation created in the society? That's also important. It's not that we do an innovation today and it doesn't hold for next generation. So that's also important aspect look into. As an Indian farmer, we had done great. So that reflects that the innovations made as on date, we are providing not only the Indian community, the global health outcomes. 20% of medicines are made from India, which is being consumed by globally. 40% Americans takes Indian medicines, 33% in Europe, 60% in vaccines and all those things. That's enormous in terms of what we have produced and what is the kind of success we have created that basically comes from chemistry. The chemistry has to create a molecule in a manner in which it can become an affordable medicines. Then comes various things. And you also have, in terms of what is the kind of wealth that's being generated, we expect about from 60 billions to 120 billions by 2030, and probably by 2047, another 400 billions. That's the kind of wealth which would be creating. And there is a lot of scope what as an organic chemist can be adding value to the society. So what is that? I mean, when we do certain things, we always think we have accomplished something extraordinary. But in industry, it's not that we do things in lab that's going to translate, because every day you are doing several things. You do at 10 milligrams to 100 milligrams to 100 kgs to 1 ton to 5 tons and things, depending on what is the market size. And you need to measure each of the activity what I am doing. Because in lab, 5 ml using nothing. And if I translate that lab into commercial, that 5 ml becomes 5 tons. How do I move the 5 tons of material in the lab? I mean, in the commercial scale, which is really a different way of looking things. If I use 5 ml, should I use 5 ml? What does it mean when I go to the scale up? What does it mean I do on 5 ton scale? So you need to measure that each of the things, activities, in terms of the efficiency of the process, in terms of the hazard, in terms of detection, in terms of analytical, in terms of quality. Everything needs to be measured. Unless you measure and you quantify and you say that this 5 milligram scale is sensible to go into commercial scale, then only you have to go into the commercial scale. You cannot do by telling that I have done 10 gram scale and I'm having a product. It doesn't make any sense in terms of commercial viability. I will just go to the another slide. We say that we have done an experiment in lab, and what is that it translates when you go into the scale? How many of those experiments really you are able to transfer the technology first time right? It's not that I have done 10 grams, I'm going to 1 kg, I'm having 100 problems because you don't know how to take out, you had use a spatula to take out the material, but there is no spatula in industry. You need to transfer that material and dry that material and get the material out of the whatever the centrifuge, whatever the various instruments which are used in the commercial scale. So the success depends not only on the chemical reaction, but all the you need to be ensuring that at least 99% of your experiments gets translated at a commercial scale, unless it's extraordinarily difficult. And then the second thing, what is the kind of yield conversion? Is that I'm able to capable produce more than 90% every times? Or I'm going to happy with a range of 80 to 90%? We can be happy, we can be still, it can be commercially successful. But what we have to see that, is this way of doing thing, are we going to produce a material which is going to reach into the market as a medicine? That's another question what you need. So at a chemist hand, you see that 80 to 90% doesn't make any difference in terms of the cost of the material, in terms of quality of the material. But in terms of the other aspects of the materials, it can have a detrimental effect, which we will see in a few minutes. But the other aspect, that's the technology at a commercial scale, business competitiveness. Most of the time when you go to the place where you say that this is the medicine available for 10 lakhs, and we will be able to produce at 5 lakhs. But what we see, a medicine available in the market, actually it will be less than 1% as a drug substance. If you are able to produce less than 1% of the drug product what is available at commercials, at the market sales, you will not be able to, because the medicine comes after several years of research, and you need to incorporate all those costs of expenses when you are marketing the product. And it's not all the products result into a drug. Maybe if it is 1% or 2% of the innovations becomes a drug, that's great. So you need to make those money. So you need to be very careful that when I say that I'm making the money, at what cost I'm making the money, the product, and where it's going to produce any commercial success or viability to the any organization or to the development of the nation. So I just said, when you talk of the drug substance, who is your next partner? Your customer is the drug development man, formulation. When we say that he has produced 3 batches of using your material, you have produced fantastic material, it's commercially affordable in terms of the cost of drug substance. But when you say that the first batch, yes, 1 hour, 5 hours, 10 hours dissolution data. This what dissolution means, the drug bioavailability over a period of time. And this is the specification. Here it's meets the specification, meets the specification, meets the specifications. As a chemist, as a scientist, we are all happy that we have made a product which meets the specification. But as a business, what is it meeting the specification? Is this specification is sufficient enough for you to make a product which is going to be sustainable or viable? Sustainability in the sense that when you say that this you have made 100 batches, I had shown that when you make 100 batches, what is the batch which you produce? We said 80 to 95%. That means the 80% material, sometime it has failed and you reprocessed. That means your process is not consistent. That's why, or you don't know what is the criticality of the process where you are supposed to control. And that changes, you may be producing a material in terms of quality which is extraordinary, but in terms of its crystal habits, might influence in terms of this dissolution profile. If I say that, if I analyze this product as a capability index, what it can generate, a business value to the company, this is what it is. Before going to that, what the FDA looks in terms of a drug product. Previously, it used to be quality, it used to be assay, in terms of 3 aspects, they say that you have to identity, purity, assay. That used to be definition of a quality. But today, FDA says drug product free of contamination. No impurity. It should be free of contamination. You should define whatever it is impurity to a level of certain percent. And it's also reproducibly delivers the therapeutic benefit promised in the label to the consumer, reproducibly benefiting the product attributes. So how do you define that? Do I produce a material in the previous? Is it reproducibly produced? No, but which is within the specifications. And my FDA tells that it needs to be reproducibly delivers the therapeutic benefit promised in the label to the consumer, that's the patient. If we look at the process capability of this in terms of sigma, that means if we take 100 batches, what is going to be the failure rate? And if I see that the first batch has more than 3 sigma, it's still going to be commercially viable. But if I see a second batch, at one of the time point, it doesn't meet the 3 sigma. That means it's going to be commercially unviable. Whatever defined specification it is meeting, but it's not going to be commercially viable, because within the 3 batches, we're able to see a substantial difference, which is very high sigma level, and then sigma level coming down, and sigma level coming down. So but that means such a kind of process, even though we have fantastic material, best of the material, best of the cost, but it's not going to deliver the product in terms of therapeutic benefit to the patient, it's not going to be of use. So you need to see that what is that material I am producing and what it is capable of delivering in terms of a final product. If I don't understand that aspect, that means we have failed together. So when you have a sigma level of 2, it's a non-competitive process. That means we are not going to make any money. If I am having at least 4 sigma, then I am able to compete. Even with 4 sigma, if there is one failure in maybe more than 100 batches, then you need to investigate what is that failure reason, kappa, and you need to be scientifically evaluate and then tell, this is my problem, this is how I am going to control. So if I don't understand that, doing that process, doing that science is not going to be great in terms of industry. One of the product which we are looking in early 90s, gabapentin. It's a very simple molecule. There was nothing great about the chemistry, but there was something important. The Pfizer patent tells that if you want to produce 100 grams of 100 kgs of a product, you need to evaporate 5,000 liters of solvent. It's a huge energy. I mean, you need to distill out water and 5,000 liters and weeks. And if you want to produce 100 kg, it's more than a week time. So productivity is so poor, and the energy requirement for producing 100 kg is huge. Then your management comes, I want 500 kgs in 1 week production. We are not able to produce even 100 liters. Then when you say that 5 kl liter water to be distilled out, no, we are not telling that you distill out water, but you find a solution how that should be avoided. That's a process demonstrated by someone else, and he may be doing that. And because he is an innovator, he may be making something in terms of profit. But as a generic product, we will not be able to distill out 5,000 liters for producing 100 kgs and then taking 1 week time. And you know that distilling out 5,000 liters and then asking gabapentin to remain gabapentin without lactam is also difficult. So these are the biggest problem. How do you solve this problem? What we saw is that sometimes you need to be unconventional also. So when we do a Hoffman degradation, the gabapentin, the amine comes from the amide. You treat with alkali and halogen, and you do the degradation. And there are several other impurities apart from your gabapentin, comes about 5-10%. That is part of the solution in terms of the gabapentin. So we said how to isolate pure gabapentin. So we thought we can convert back to lactam. But what we had seen is that by just heating the reaction mixture more than 100 degrees at greater than pH 8, it gets converted to lactam. If you go and ask organic chemists, an amino acid under alkaline conditions making a lactam, how is it possible? It's a very clean process. Very clean conversion Within 1 hour at 100 degrees, gabapentin in alkaline condition converts into lactam. It separates as clean liquid. Drain out water with impurities and you have clean product. And you reconvert this with concentrated HCl and you make a gabapentin. And there your HCl is only 2 volumes. You distill out HCl and you make gabapentin hydrochloride pure product. And you just add water for 170 kg, 85 liters of water and adjust pH to 7.4. Gabapentin crystallizes neatly. There is no 5,000 liters of water removal required here. The solution, what you need to be is that how do you say circumvent distilling of 5,000 liters of water to produce, and we are able to produce 105 kgs in 6 hours. You don't need to distill, there is no energy, there is no 5K reactor, means it's very big. There are so many operators needs to be working, steam supplying all those things for weeks together. That gets solved. And this is the process which every industry uses to produce gabapentin today. The other product was, the citalopram. When you look at citalopram, there is a cyanide, displacement of bromine with cyanide. And more the product you want to convert into a product, this product turns into desmethyl and becomes less yielding process because more and more impurities gets formed and that becomes very difficult to remove. Then what to do that? Because there is always a demand and requirement. How do we solve this problem? Then what we did is that can we use something to facilitate like cuprocyanide? And then when we use, then we are able to demonstrate this reaction much better from 54 to 94%. And later on, we had seen that the same process has been used by Buchol to produce the cyanide, biphenyl cyanide. So where you are not able to produce in a consistent quality, now we are able to produce a consistent quality. This was a requirement, otherwise we were able to, having a 1 sigma process and asking to produce tons of material is not going to make. So that's the biggest challenge we had in terms of making this product. The other product was the companies would have the patent, extension by asking your racemic molecule to making the chiral escitalopram. And you see that this product making the innovator process 100 kgs produces 38 kgs of end product and requiring about 200 liters of solvent and 99% of chiral purity. So whatever the technology one would use, they have used word, an ester, an active ester to produce the citalopram, acetalopram, making the mesylate and mild base and then forming the tetrahydrofuran ring. So whatever variation one would do, they will always say that it's a doctrine of equivalent. If you have used an ester, you have used an ester which we have told that an active ester, whether you use mesylate, tosylate, benzoate, nosylate, anything, which becomes a doctrine of equivalent. You are not out of my patent. So then you have to make that product a non-infringing route. What we asked is that, is it another way of making the furan? We asked whether can we make an alcohol to ether and then ask it to cyclize. So we made a phenyl ether, active phenyl ether, and treated with a base. It's a clean. Now we are not using an acid. Our chiral purity is whatever it is there, it is there. And now we are able to make from 100 kgs, 54 kgs of our product. So if you talk of this 100 kg of an ester, the derivative, and you get 54 kgs of the product, we get about 20% excess yield. And we are able to scale because our solvent required is 100. So instead of making 3 batches, you can make in 1 batch the same quantity. And you have excess of the product, which is part of your outcome. There was another product where we did the imatinib, where Professor Yadav is also instrumental, first time launching in India by Sun Pharmaceutical Industries way back in 2000. And nobody filed a patent because there was a polymorph patent. So we had a polymorph how to make, and which was worth of more than $300-400 million business if you are able to file the first time in US, and 6 months exclusivity can be this kind of a cost money. And what we did is that how do we look into process? When we looked into process of the innovator that requires about 41 liters of solvent and 5 kgs of intermediate, and our process requires about 1/4 of the material, and we just use 7 liters. When you say that you wanted to do on a commercial scale, you cannot be using 40 liters of solvent for 1 kg of output. We are only using about 1/8 of the requirement, so that you are able to produce a material 8 times much larger quantities and a better quality of your material in terms of overall output. So most of the times when we say that what cost one needs to make your product, if you look at Mother Nature, if you say that what is the cost of sugar, table sugar or glucose, and we see well-defined chiralities, and you get for 225 rupees, and per carbon cost, if you say that 180 by 12 number of carbons, if you assume that 15 carbon equivalent, and we can assume that per carbon cost is 15 rupees. But when you say that when you want to make pregabalin, what is made is 6,000 rupees. If I calculate per carbon cost, it's 452 rupees. There is one chirality, but there are several chiralities. So whenever we say that we want to make a product, we need to ask ourselves, if I convert the product in terms of molecular weight divided by assuming everything is carbon, then what would be that cost? In that case, in case of sugar, it's only 15 rupees. In terms of the normal sucrose, it's about 2.1 rupees. And that's the cost we are paying. But when you say that we want to make a drug, I'm making 5 lakhs. What is the molecular weight? If you see Pavipiravir, it's only about 157, but still it's 20,000. Is it, innovative process? Is this a sustainable process? It's not at all. Most of the process what we are producing is non-sustainable. If you have an efficient process, all of the molecules can be relooked into what is going to be the simplest and the best. What Professor Vinod Singh said, one-step process. Is it possible to get one-step process? That's the kind of a thing one needs to look into. If you say that we want to make 2-phenyl propanol, what could be the various synthetic strategies? Because it's not that we jump into somebody has reported I would like to make it. There is a possibility you can convert into acetophenone, Wittig, and hydroboration, then modified Wittig, and then reduction. There is a phenacyl chloride, methyl magnesium, and reductions, and Darjan's glycidic. There are several ways. You need to identify what is my carbon efficiency, what is my waste, what is my yield, number of steps. What is the best route? We need to really engage with the what way synthetic process we would like to demonstrate, and how best it's going to be, how commercially viable, how environmental control would be there. So the other possibility is that we can also have the sulfonyl ylides and then converting this. The best could be that propylene oxide is available, benzene is available. In one step, you could still make a product of 2-phenyl propanol. So you need to really engage yourself, think what best one can look into in terms of making a best product of cost. What is that? We said that we are, in terms of globally supplying medicines, 20% of the global requirement and 40% of US requirement. But what is that we are getting? We are getting our revenues last year of 60 billion. But that is nothing. When we say that 1.6 trillion and we are talking of 60 billion, we are of one digit. And we are third in terms of producing volumes. But in terms of our revenues, the growth, economic growth is not there to an extent what we are what we are making activities. We need to be much more looking into that. What is this innovations and sustainability? Because what we are doing is sustainable. We did fantastic innovations. We are able to create a global health, affordable medicines, all those things. But sustainability depends. If you are not able to make something out of that and only produce and produce and produce for peanuts, it's not going to be sustainable innovations. We need to be much more than what we are doing today. If you look at companies like, Professor V.K. Singh said the Ketroda, last year it was about 31 billions, one product. We are 3,000 companies, 60 billions in 1 year. One company, one product of one company, 31 billion. Half of Indian business is one product of one company. Who is sustainable? Is that 3,000 companies making 60 billion or one product of the one company making 31 billion? That's the sustainability. Is it that we are not able to think to that level? No. We can think as much to the extent of what others are doing. And you see, there is another company, Novo Nordisk, Eli Lilly, Sanofi, these companies. These 10 companies all are more than 10 billions, each one of their product, not the company, each one of their product. And these two products, Novo Nordisk, Eli Lilly, they are going to make 35 billion, 31 billion next year, each of those products. So that's what we made as a country in 1 year, one product. So that's the kind of a novel innovation can bring in terms of the economic growth. So if we are not able to design things in such a way that what is that we are expecting in terms of innovation, scientific contributions, it's only producing the molecule, meaty molecules or generics, and say that we are producing affordable medicines is not going to be no more sustainable. We need to be much more doing innovations in terms of making very different way. And even then, the companies who are making innovations, it's not that everybody does make money, because you have to invest on R&D efforts, and the output should be more. If you see the Ambien, their output is 15 billion per molecule compared to what they invest 5 billion. They invest 5 billion in R&D, but they make 15 billions in the molecule which is coming out. But in terms of Sanofi, you see they spend about 7 billion, but they get about 3 billion. So that's not going to be, economically viable over a period of time. You need to be also be saying that what is that I am spending money and what is my return on investment. So there is one company, the Novo Nordisk in 2023, their company revenue in terms of the market capital 410 billion last year. It's much higher than the whole country, 400 billion. The one company market capital is much larger than GDP of that country. So that's the kind of economic growth they could demonstrate. They're 80% of business in diabetes. 80% of their business in diabetes. They are the leaders. And we can also be leaders. But we need to make an innovation. If you don't make an innovation, but we cannot be leaders. If you are a follower, you cannot be leader. You are going to be follower forever. So we need to change that, and you need to become a leader in terms of doing things very differently. So we embarked in terms of making some of these things in diabetes. What is the problem in diabetes? Insulin resistance, insulin secretory dysfunction, and hepatic glucose overproduction. So we tested one of the molecule what we produced. And we also compared our results in terms of standard of care. Like we have Ozempic, that's a semaglutide, Trulicity, this is one of the best product at that point in time. In animal models, we see that definitely performance at much lower doses compared to 14 nanomole. 6 nanomole per kg produced in terms of insulin secretion, in terms of triglycerides, in terms of HbA1c body weight, much different than the standard of care products. So that means we see that there is a product which we are competing, at least in animal models. Seeing in animal models means nothing. We have seen in animal models. What does it translate into human? Okay, before that you ask that question, you need to test any of the product in terms of several of the off targets. So we tested more than 100 of those off targets at 1,000-fold. We say that our product is about 80 picomoles, and we tested 1,000-fold of concentration. It is selective. And we tested in hertz, SIPs, and various other things. And we found that this molecule is clean. So then we can go into the human trials. What does this human trials tells to us? Okay. So when we did, the single ascending doses, the first in human, with one single dose, if we give the product today and we check the person body weight, we are able to see about 2 to 3 kg body weight loss. We injected 1520 micrograms, that means 1.52 milligrams, and 2000 2 milligrams and 2.52. And on day 8, it's 2.4, 2.2, 3.3. And we measured after 3 weeks, the weight remains more or less 2.2, 1.8, 3.1. So with one single dose, the body weight reduced about 2 to 3 kgs, and that remains more or less for more than 3 weeks. The body weight control reduction around 3%, and you see that the body weight is 103 to 130. This is the range. They are really obese people. And what else we had seen with our product? If you talk about the first generation incretin like liraglutide, which is what they could achieve in 2 months, we are able to produce in one single dose. That requires giving the product every day for 2 months, and here it's one single dose, which can produce about 3.3 kg body weight. We are also able to see triglyceride levels from 163 straight going about 69 milligrams of triglyceride in human trials. So it's a huge in terms of. So what does it mean in terms of products which are available in the market? If we say that fenofibrate somebody takes about 24 weeks, that produces about 29.7 to 38, and we are already there with one single dose. So we don't need months and months and material taking about 500 milligrams. You give about 1.5 milligrams, you produce the same effect. So the next question what we ask, what about its anti-diabetic potential? So the best way is to demonstrate, you take a glucose challenge test. You ask a person to take a glucose of 75 grams oral and say that what is the measure of the glucose uh, excursion. In the untreated subject, there is an increase of glucose levels. And in the treated subjects, there is a decrease of glucose levels. So that means the glucose excursion is controlled by producing the insulin. So you are able to negate the increased glucose levels by producing insulin, because it's supposed to help in terms of the increased glucose levels. Then we asked, is it producing insulin to say that what we had seen in terms of overall glucose reduction? It should have been produced. Surprisingly, what we had seen, there is a reduction of insulin. With a reduced insulin, you are seeing reducing of glucose. That means the glucose is forced much faster than the untreated patient. How do we explain this anomaly? This means within the 4 weeks of treatment, the insulin resistance is overcoming. The person would be producing more insulin than what it is required. So maybe someone requires, the person who is diabetic or obese, they produce 50 times more than what is normal insulin required. And you don't need that much. That's why the person becomes obese. In our case, we see that there is a definite reduction. It tells that you don't need a level of insulin what was produced before treatment, which is becoming because of the insulin resistance. And in this case, the insulin resistance is overcoming. So it's more beneficial to the patient. Your pancreatic beta cell need not have to work so hard. So that's the in principle. We had seen in these patients some amount of HbA1c reduction in 4 weeks, and we also saw that the lipid levels coming down, total triglyceride, total cholesterol, all those things within 4 weeks compared to the placebo subjects. What is the time it took for us? We are competing in terms of the global players, whether it is Pfizer, whether it is Novo Nordisk, whether it's Eli Lilly. We took about 30 months to first experiment to first in human. So the first dose in human happened within 30.5 months. So we all can do that. We need to think and do what going to be something innovations we can create. The more important thing is that we need to dream and discover and live. We think that we are doing everyday innovations, but after 20 years, we believe that whatever we have done, something is not really adding value to the economic growth or societal impact. Unless we do those things, doing things, right things at right time and doing things right is most important. So we need to do at right time so that it adds value to the society. Thank you. Thank you, Professor. Yeah, that's all. Yeah, that's the 4 doses.
Thank you. I think we'll stop here. Please join me thanking Dr. Rajpannar for the excellent talk.
Thank you.
Thank you so much, sir. I would like to invite Professor G. Mukesh to felicitate Dr. Rajaman. Now I request Dr. Vinay Srivastava from Cadila Pharmaceuticals to felicitate Professor G. Mukesh. Now, before we proceed with session 2, we have a short tea break. But before that, everybody is requested to gather near the reception for the photo session. After that, you can have the tea break. And the exhibits of Cadila Pharmaceuticals are also displayed outside the seminar hall. We request you to visit there. And we request all of you to please join us back sharply by 12:15. Everybody, please take your seats. Welcome back, everyone. We now begin with session 2. For this, I would like to invite Professor Tapas K. Kundu, JNCASR Bangalore, and Professor Alakesh Bisai, ISER Kolkata, to chair this session.
So after a fantastic start by Professor Singh and then Dr. Raj Mannar, here next we will have an eminent organic chemist, chemical biologist actually, Professor G. Mukesh. He is a professor in IPC, Inorganic and Physical Chemistry Department, Indian Institute of Science. And he's also the Dean and Division of Chemical Sciences in IIC. I'm well aware about his work and he's a fantastic speaker. You will soon know that. Just to give his brief introduction, Professor Mukesh did his BSc, University of Madras. MSc is Bharatidasan University and earned his PhD from IIT Bombay in 1998. Following his doctoral studies, he undertook postdoctoral research at the University of Göttingen, Germany, and subsequently at the University of California, Berkeley, under Dr. Casey Nicolau. Since joining in IAC 2002, Professor Mukesh has established a research group focusing on the chemical synthesis of small molecules and nanomaterials of biomedical applications. His work has significantly advanced the understanding of thyroid hormone metabolism, antioxidant activity in mammalian cells, and the development of molecular probes for detecting reactive oxygen species. As I told you that he is more an chemical biologist than an organic chemist, and it's right, transition he has made in his career. Throughout his career, Professor Mukesh received numerous awards and recognition. And one of them is, of course, the Shanti Swarup Bhatnagar Prize in Science and Technology in 2012. Infosys Prize in Physical Sciences 2019. He's the fellow of all the Indian academies. And as I said, that he is an excellent speaker, excellent research supervisor. Many of his students has taken very good positions in the country, including my own institute, Jawaharlal Nehru Center for Advanced Scientific Research. So, Professor Mukesh, welcome for your lecture.
Thank you. Thank you very much, Professor Kundu, for the nice introduction. I take this opportunity to thank the organizers for inviting me to give this talk. Today, I'm going to talk to you about small molecules as redox-active artificial enzymes. These are small molecules, but they can substitute enzymes in the cells. One particular aspect I will be talking to you today is how synthetic compounds can be used to redox regulate in mammalian cells. When we talk about redox regulation, one molecule that comes into our mind is hydrogen peroxide. And the hydrogen peroxide plays a central role in redox processes, and particularly in aerobic life. And one fundamental reactions that we need to understand is the conversion of molecular oxygen to water. Right? We all know one of the most fundamental chemical reactions that takes place in human body. And it requires 4 electrons to convert molecular oxygen to water. But in this process, as you can see here, 1 electron addition to molecular oxygen generates superoxide, 2 electrons generate hydrogen peroxide, the 3rd electron generates hydroxyl radical, and the 4th electron generates water. And in this process, it generates superoxide, hydrogen peroxide, and hydroxyl radical. These are known as the reactive oxygen species, inevitable, must be produced in the human body, but they need to be controlled in a way that health is maintained. And there are enzymes involved in this. Also, there are enzymes that takes care of this reactive oxygen species. For example, superoxide dismutase that acts on superoxide, glutathione peroxidase act on hydrogen peroxide, catalase also acts on hydrogen peroxide. That's the reason when you open a hydrogen peroxide bottle with bare hands, without a gloves, that's what you see on the skin, right? The skin proteins get oxidized by hydrogen peroxide. If this is the case, how hydrogen peroxide is present in human body and how they are controlled. And these are the enzymes that act on that catalase, superoxide dismutase, glutathione peroxidase, and peptides like thioredoxin, glutaredoxin, peroxiredoxin, and so on. So today's talk, I will be talking to you on one specific enzymes that act on hydrogen peroxide. Before that, we must understand how hydrogen peroxide play a dual role, and it is a double-edged sword. Less amount of hydrogen peroxide will inhibit the redox signaling. More amount of hydrogen peroxide is going to introduce oxidative stress. And as you can see, hydrogen peroxide oxidizes cysteine on protein to initiate redox biology. Alternatively, it can also be converted to water by cellular antioxidant proteins. When hydrogen peroxide level increases uncontrollably, hydroxyl radicals are generated, and they react with metal ions, and irreversible damages to biomolecule takes place. So when we look at these reactions, this side is the oxidative stress, where hydrogen peroxide react with metal ions like iron 2+ to generate hydroxyl radical, or it can play a more important physiological role that is called the redox biology, where hydrogen peroxide oxidizes the protein thiol to get sulfonic acid that initiate the signaling processes. So we are looking at 2 sides of the same species. And in human body has about 5 to 6 grams of iron, and only 1 milligram is lost per day. We know when we put iron in the atmosphere, it reacts with water and oxygen, and a process we know very well is rust. It's a rusting. And our body has oxygen and our body has water, but there is no rusting takes place because the iron is controlled very well. But if it leads to rusting, that leads to diseases, and we talk about Alzheimer's disease, Parkinson's disease. Many neurological, neurodegenerative diseases are basically rusting of iron in the brain. And that generates reactive oxygen species. Therefore, iron in biology is tightly controlled. But in recent years, the role of iron in oxidizing many biomolecules, although iron is very important, hemoglobin, myoglobin, without iron, we will not be able to survive. And iron can initiate certain processes, pathological processes, and that is now emerging as a process called ferroptosis. I don't know how many of you heard about the term ferroptosis. And it's a recently discovered iron-dependent, oxidative, non-apoptotic form of cell death. It's a regulated, and that's due to accumulation of lipid peroxidase in cell membranes. This is due to dyshomeostasis of iron metabolism that lead to lipid peroxidation, insufficiency of glutathione, and a particular enzyme that triggers ferroptosis is GPX4, glutathione peroxidase 4. And that initiates lipid peroxidation, lead to cell death, and that is known as lipid ferroptosis. And today, I'll be talking to you about ferroptosis and how GPX4 modulates ferroptosis, how it can design small molecules that can functionally mimic these particular enzymes. Ferroptosis has been implicated in several pathological conditions, including cancer, ischemia, reperfusion injury, immunity and infection, neurological disorders. And particularly, I will be focusing on the neurological disorders. And the ferroptosis play a key role in that. So prevention of ferroptosis by GPX4, it's a very, very special enzyme. And there are many isoforms in human body. GPX1, which is present in almost all cells. GPX2, it is in GI tract. And GPX3 secreted by the kidneys into the blood, mainly in the bloodstream. GPX4 is widely distributed in different tissues, GPX5 and GPX6. So these enzymes are selenoenzymes. They contain selenocysteine in the active site. At GPX7 and 8, they contain cysteine instead of selenocysteine. This is how the lipids gets oxidized, and polysaturated lipids, and they get oxidized to lipid peroxides, and they generate lipid radicals that initiate ferroptosis. But this process is inhibited by GPX4. And GPX4 is a very unique enzyme which converts a peroxide to an alcohol on lipid. And this is the only enzyme that can do this job and repairing the lipid, which is oxidized lipid. So we were interested in these enzymes called glutathione peroxidases. So they reduce hydrogen peroxide to water or organic peroxide to alcohol by using glutathione as the cofactor. That's what the name glutathione peroxidases. And the GPX deficiency directly induces an increase in the oxidative stress, and overexpression rescues the cells. And what is unique about glutathione peroxidase is that it contains selenocysteine. When we talk about selenium, people think selenium is toxic. And our human body has 25 selenoproteins, and without which the body cannot function. For example, thyroid gland cannot function without selenium because the selenoenzyme, iodothyronine dehydrogenase, is the one that activates thyroid hormones in the thyroid gland. So what is special about selenocysteine is the pKa of selenol. So the pKa of selenol is 5.2 in proteins, which is much lower than the pKa of a cysteine, which is 8.3, that is again lower than the pKa of a serine residue in protein, which is 13. And this gives a dramatic catalytic advantage, catalytic and dramatic nucleophilicity to the selenocysteine in the protein. So therefore, these are very unique enzymes, 25 of them, just 25 proteins in the human body that encodes for selenocysteine. So we have developed several GPX mimetic compounds, starting from epsilon, which is very well-known antioxidant molecule. We modified that, making new types of compounds. So one particular compounds that we were interested in is analogs of epsilon, similar to epsilon, but I'll talk about some of the biological activity of this. And this particular compound, epsilon, has been in clinical trial for many, many years. And I think maybe hundreds of clinical trials that happened with epsilon. And human trials suggest rescue drug could be safer treatment for bipolar disorder. And this is one of the some of the recent activities. And epsilon has gone from identification as a potential bipolar treatment to human trial in about 2 years, incredibly fast for any drug discovery effort. And epsilon is part of the NIH clinical collection, a chemical library of bioavailable drug considered clinically safe, but without proven use. Even now today, people are trying to identify what could be this used for, epsilon. So we have what we thought, we looked at why epsilon fails in many of these clinical trials. What is the chemical background, the chemical property of epsilon, which is making it not suitable as a drug. And what we found was the presence of an amide, cyclic amide moiety here. You can see it's called selenazole moiety. When a thiol reacts with the selenazole moiety and it opens up the selenium-nitrogen bond, so therefore it can inhibit a variety of cysteine-containing protein in human body, including serum albumin and many other compounds. So it's irreversibly binds to all known cysteine-containing proteins. So the first idea came to our mind was you block that type of unwanted reactions. So what we did, we modified the structure and we came up with a small molecule, which is also a selenazole, doesn't have the carbonyl group here. That is one modification that we have done. And it is basically a fine balance of chemical reactivity, biological activity, and the toxicity that we kept in mind. And what we did, we removed the carbonyl group and we introduced a methoxy group in the ortho position because we wanted to make the cycle intact. We don't want that open chain because when the selenium comes out during that biotransformation, it can lead to toxicity. So to reduce the toxicity, we put an additional group that forces the selenium to form the cycle. And this is the catalytic cycle of this particular compound. I'm not going into the details of this. And this functionally mimic glutathione peroxidase. It functions as an enzyme. And we have done a lot of cellular studies and so on. And finally, we showed that this actually work in mammalian cells as an enzyme. And we wanted to modify that further because keeping the ferroptosis in mind, which is we are GPX4 mimetics, because the compound one is GPX1 mimetic. How do you move from GPX1 to GPX4? All of them are selenocysteine containing enzymes. What modifications you do to make a GPX4 mimetic? And we looked at the function of GPX4, it acts on lipid peroxides. So by introducing hydrophobic moiety, can we target these molecules to the inner membrane of the cells where the lipid peroxides are produced? And we introduce a longer chains, as you can see here, 1 carbon, 2 carbon, 3 carbon, 4 carbon. We reached up to 12 carbons, and the dodecyl moiety attached to the epsilon are attached to the compound which doesn't have the carbonyl group here. So when we carried out the GPX-like activity and we found that there is a large variation in the specificity of peroxide, and that's important. One of them reacts more with the hydrogen peroxide, and the one which has a hydrophobic side chain, it reacts with the cumin peroxides. The activity of the hydrogen peroxide is low. That's what we see in the GPX1 and GPX4. The difference between these two is the selectivity to a particular peroxide. One is selective to lipid peroxide, other one is selective to hydrogen peroxide. So what we did, we have done further experiments in particular cell lines, which is prone to ferroptosis, which is HT1080. And this is a ferroptosis prone cell lines, and there is a RAS mutations that allow the iron to dysregulate in the cells, induce ferroptosis. And what we did here, we treated the cells with RSL3, and this is a compound that inhibits GPX4. And when we inhibit GPX4, you can see this, all cells are dying here. And there is no cell viability here. It's complete death of the cells. But when we treat with the compound which has a dodecyl group, you can see that it completely rescues the cells from ferroptosis. And again, compound 1 also rescues the cells from ferroptosis, but epsilon is not able to rescue the cells. And this is where I talked about the difference between the well-known antioxidant epsilon and the compounds. What we did, we just did a simple modification. We removed the carbonyl group and we introduced a methoxy group. And this makes a complete different type of reactivity to the molecule. And this behaves like ferrostatin, which is ion chelator. And the reactive oxygen species, lipid peroxidation were assessed by standard methods. And we found that compound 1 and the compound 5 works very nicely in HT1080 cell lines, which is a ferroptosis prone cells. We also looked at the cell death pathways in the presence of elastin. So elastin is the molecule that actually blocks the system Xe minus, and system Xe minus is the one that takes glutathione, which is involved in the glutathione biosynthesis. It takes cysteine inside the cells and transport glutamate outside the cells. It's an antiporter. And the cysteine is converted to cysteine, that reduced form that makes glutathione, which is cofactor for GPX4. So therefore, by inhibiting the cysteine uptake, you can actually block the GPX4 activity. It leads to the cell death. Again, you can see when you add elastin to the cells, a complete cell death, and none of the cells survived. And when treat that compound with compound 5 and compound 1 is not that effective, but you can see compound 5 is highly efficient in preventing the cell death induced by elastin. So we have done multiple inducers, ferroptosis inducers. I'm not going to list all of them. Quickly, I will move to the different type of cells, what we did, how does it work in primary neurons, because neurons are much more sensitive to ferroptosis compared to the HT1080, which is cell lines. We isolated cortical neurons, and very special type of experiments need to be done. These are isolated from 15 days old mice, and that is important because those neurons are suitable for neurodegenerative studies. And we isolated that and studied the effect of our compound. You can see untreated cells, the neurons are healthy, and we have treated that with SCR, that is siRNA, scrambled one, no effect. But when we treated the cells with the siRNA specific to GPX4, it knocked down the GPX4 gene. So therefore, no GPX4 expression in the cells that lead to the cell death, as expected. And you can supplement that with compound 5. So that means these cells do not have GPX4 enzyme, but we are supplementing that with the compound. So that can completely substitute GPX4. Therefore, it completely mimic a GPX4 activity in the cells. And the compound 1 is not effective, epsilon is not effective. As you can see here, the Western blotting, there is a decrease in the GPX4 level that is supplemented by compound 5. And this was very interesting. And we also did RSL3 inhibition, elastin induced ferroptosis in primary cortical neurons. As you can see here, RSL3 again lead to complete cell death. Elastin leads to complete cell death, but both of them are rescued almost 100% by using compound 5 here. And compound 1 is not effective, and epsilon is not effective. So what I would like to say here is that they all have similar structures, epsilon, compound 1, compound 5. But what is the difference? The small change in the side chain. We are making it more specific to lipid peroxides. So they are able to work on lipid peroxides, reducing them to the alcohol. So what we did next, we have actually taken the lipids which are responsible for ferroptosis, and we performed the reactions outside the cells. And in fact, when we use a glutathione and the compounds, you can see with compound 5, with these two lipids that we have taken, the activity is much higher for these compounds. And they actually functionally mimic this enzyme. Catalytically, they mimic the enzyme. And by reducing hydrogen peroxide, lipid peroxides to lipid alcohol. And we were able to characterize this lipid alcohol completely by NMR and mass spectral techniques. And what we show here is that this compound is the active form, selenol, that reacts with the peroxide to generate selenic acid and reduce the lipid peroxide to lipid alcohol, just like the GPX4. And then the selenic acid can be converted to selenol by glutathione using the internal glutathione. It can also use other thiol like thyrodoxine and so on. And both the lipids that we have taken, it fully mimic the ferroptosis conditions. And what we did, let us change the inducers. We have done ferroptosis assays with elastin, RSL3, Fin56, FinO2. There are many inducers known in the literature for ferroptosis. What about hemine-induced, and why it is relevant? And in the case of stroke, for example, whether it is ischemic or hemorrhagic, particularly hemorrhagic stroke, heme gets released. And because hemoglobin gets hemolysis, and it releases heme that induce ferroptosis, And what we have done, whether our compounds can prevent heme-induced cell death in primary neurons. This is a hemine, and epsilon rescues the cells little bit, and ferrostatin rescues the cells, N-acetylcysteine rescues the cells, necrostatin prevent the cell death. That means the heme-induced ferroptosis or the heme-induced cell death is not 100% ferroptosis. It is apoptosis, necrosis, and ferroptosis. And it's very difficult to design a compound that can block all three cell death pathways. And that's what we are looking at it. And when we also want to design drugs as a potential candidate for stroke, we should also look at how the platelets function. And that's where the GPX3 isoform I mentioned, it is in the plasma, and it's in the bloodstream, and which prevent the platelet aggregations. So we studied hemine-induced ferroptosis many years ago, but now we started looking at how the inhibition of the prevention of platelet aggregation by GPX4 mimetic could also be important for designing drugs. And we looked at the pulmonary embolism, and in this process, what happens? The platelets get activated, and then they aggregate with each other and block the blood flow, and blood clot is formed, and this blocks the oxygen transport lungs and to the tissues. And therefore, we looked at the platelet activation by the small molecules. Multiple experiments we have carried out: platelet addition, ROS generation, calcium levels in platelets, ASK phosphorylation, ERK phosphorylations, many assays, but all of them lead to platelet aggregation. So I'm going to tell about only one assay, skip all these experiments. So this assay tells us that the GPX mimetic prevents platelet aggregation in collagen-induced platelet activation, thrombin-induced platelet activation, and also ADP-induced platelet activation. All cases, you can see this compound 1 is very effective, and when we compare that with compound epsilon, is not effective at all. You can see 5 micromolar concentrations. You can see that the platelet aggregation is inhibited by 80%. And this is significant. And in the case of thrombin-induced platelet activation as well, you can see 80% inhibition of the platelet aggregation that takes place. Can we translate this to animal experiments? And what we did, we established an animal experiment for pulmonary embolism. What we do, we treat the animals with collagen and epinephrine, which induce embolism, which makes the clot formation in the animals. And you can see these are number of animals that we have taken. 20 animals, and 80% of them survived with the compound 1, and 5 milligram per kilogram of body weight. And you can see this epsilon is not that effective compared to compound 1. And this was very encouraging. And now we would like to do further experiments to show that whether GPX mimetics can be used to treat these type of diseases. Of course, further studies in human and all is required. This is the lung section of the animals. After treatment, you can see the collagen epinephrine and collagen epinephrine compound 1, and almost full rescue of the animals that is seen here. And this is the thrombi counts. And this is the time we thought we'll collaborate with somebody working on mesenteric arteriolar thrombosis. This is an important process. So we collaborated with Professor Debaprada Das, is in the Institute of Medical Sciences at BHU. And they have an excellent assay system to measure the blood flow in live animals. And what they did, they have, this is the vehicle control, and this is the compound 1 treated mice. And they followed many parameters like the percentage of the patent vessels. Patent vessels are the one that is susceptible to thrombosis. And you can see here, this is the formation of thrombus. And this is the time to occlusion and the thrombus growth rate. All the cases, we found that there is a significant effect of this compound. You can see this is the without compound, and you can see with compound, most of the thrombus formation is reduced. And the most important experiment that we thought that gives more information about the safety of these compounds, the bleeding time. And in most cases, what happened, aspirin, people take aspirin, right, for blood thinning agent, and it breaks the clot and so on. So when the mice were treated with aspirin, it actually dissolves the clots, but it increases the bleeding time. And it leads to a lot of bleeding. You can see here. And the bleeding time is about 800 seconds here. But these compounds, compound 1 or the epsilon, do not increase the bleeding time, but at the same time, it blocks the clot formation. I think such type of compounds are important in using further in clinical trial and so on. So this is my last slide. So what we are trying to do here, we studied ferroptosis using compound 5. So the compound 5 blocked the cell death induced by ferroptosis by catalytically converting the lipid peroxide to lipid alcohol. So therefore, the damaged lipid is repaired. That is one side of the story. On the other side, what we did, we took compound like 1, and that blocks the platelet activation and platelet aggregation. That blocks thrombosis. So this is the one we have done animal studies as well. It blocks thrombosis. Now, if you want to treat diseases like stroke, whether it is ischemic or hemorrhagic, that represent a complex interplay of multiple pathophysiological mechanisms, including energy failure, excitotoxicity, inflammation, and oxidative stress, leading to neuronal damage and dysfunction. So what we thought, it's important to combine this function with this function. So that's to achieve something like this. So what we believe is that synthetic analogs which mimic multiple isoform GPX enzymes, particularly GPX1, GPX3, and GPX4, appear to be promising therapeutic candidates. And we do have now compounds which can block hemine-induced cell death, which is mix of ferroptosis, apoptosis, and necrosis. And we just filed a patent on those compounds. Maybe next time I can talk more about it. And with this, I thank my former students and the current students who are working on ferroptosis development of small molecules as GPX mimetics. We also work on nanozymes, nanomaterials, which function as antioxidant. I didn't have time to talk about it today. And SCRB, Ignite Life Science Foundation for funding. And thank you all for your attention.
Thank you. Is there any question? Yeah.
One of your modifications, you are mentioning that when you added the alkyl group, it becomes kind of a lipid peroxidation specific. Is it the molecule itself becomes, insoluble in aqueous, so that's why you see the difference?
Yeah, what we do, solubility reduces when we introduce the long chain.
Correct.
And possibly they are able to reach the inner plasma because of hydrophobic interactions.
Maybe it's going and sitting.
Yeah, and what we found is that all these selenium compounds, there is a specific transporter which takes this compound inside the cells. And we do not know what type of transporters are used, but they use transporters to transport inside the cells.
No, sorry, I can have one more.
Yeah.
So suppose, if this becomes a promising drug candidate for clinical, how would demonstrate this is something superior than the existing? Say there is a clopidogrel, there are several other drugs. So how do we say that this is going to be something very different in terms of?
Yeah, clopidogrel, we looked at the platelet mechanism. It looks different. The way these compounds alter the signaling pathway, particularly phosphorylation, dephosphorylation pathway. Clopidogrel, they don't do. We compared with aspirin and 2 drugs we took for comparisons. And those drugs also increase the bleeding time.
Okay. So
And
what you would say
yes,
that, okay.
Yes. So in this case, the bleeding time is not increased. It is very less compared to the other ones. So other ones have antiplatelet effect, but what we find the difference is that the bleeding time, that is a change, and the mechanism by which these compound act is different from those. Here in this case, it is basically the reactive oxygen species.
Does compound 1 helps to dissolve the clot also, like aspirin does?
Yes.
It prevents the clot formation.
It prevents, but also we see, but the conditions that we used were a very harsh condition to, to induce thromboembolism. And when we treat with the higher concentrations, so we do see, because the clot formation is so quick with this treatment, and we see that, there is an effect.
Okay. Okay, last question.
Please. What kind of fluorophore do you use for monitoring?
If it is hydrogen peroxide, we use...
for the ferroptosis.
Yeah, ferroptosis, lipid peroxidation, we use a body P-based dry.
So does that fluorophore goes to a specific organelle inside the body, or is it just goes into the entire cell?
No, it is entire cell. And we can also do mitochondria. Mitochondria specific body P dyes are available if you want to study the mitochondrial ferroptosis. But we mainly want to study the plasma membrane ferroptosis.
So one of the consequence of ferroptosis is that it also increases the viscosity of the cellular medium.
Yes.
So did you also study how much is the viscosity change?
Yeah, we are doing it now. We have a specific probe to follow the viscosity changes.
Okay.
And so I have not presented today, but we have done some work on that.
Okay, thank you.
Thank you, Professor Mukesh, for a wonderful talk. So big hands for that. There is something I have to give him.
Thank you so much, sir. Now I would like to invite Professor Alakesh Desai to begin the next lecture.
Okay, thank you. Thanks for this opportunity. So it's a great start. First one by Professor Vinod Singh, second one by Dr. Thenati Rajamunnar, third one Professor Govindaswamy Mukesh, then fourth one is Dr. Dumbala Srinivasa Reddy, popularly known as DS Reddy. So DS completed his BSc and MSc both from Osmania in 1991 and 93, then joined with Professor Govardhan Mehta in University of Hyderabad, then again moved to ISC Bangalore because when Professor Mehta moved there, then he completed PhD in 2000, then followed by he joined Cosmin Lab for 1 year as a postdoc, followed by 2-year postdoc with Jeffrey Obe at Kansas, followed by he joined Dr. Reddy's laboratory and worked there for 4 years, followed by he moved to Advinas, Tata Advinas, spent there 3 years. Then he thought, after having enormous experience from industry, he thought of joining academia. Then joined in NCL Pune. Then he became director of IIIM Jammu, followed by director of IICT Hyderabad. In fact, he is also additional charge of CDRI Lucknow. At one time, he had been the director of IICT as well as taking charge of our IIIM Jammu and as well as CDRI. So his research has been, appreciated in the various awards and honors. To tell few of them is he's the J.C. Bose National Fellow now. He is already the FNSC, FSC, FNA. He was awarded Santi Swarup Bhandakar in 2015, and CDRI Drug Research Award in 2015, CRI Sarbonj Medal, and many more. So with that, I would like to request DS to start his talk, maybe 25 minutes, followed by 5 minutes for question and answer. DS, stage is yours.
Thank you. Can you hear me? Thank you, Professor Alakesh, for kind introduction. And before that, I would like to thank Professor J.S. Yadav and Vishnu Maya and all the committee members to making me part of this interesting conference. So in addition to doing some science in my lab, I am also heading this institute. That is why I take 2 slides to little bit tell about our institute. Of course, I'm sure many of you are aware because Professor J.S. Yadav sir is coming from the same institute where I'm coming from right now, that is Indian Institute of Chemical Technology. right? So this is what our institute, we established 80 years back. We are working with basically whatever we think of chemistry or chemical technology, we are there. So our vision is to serve society by creating knowledge in chemistry and chemical technology. That's what we have been doing. And whenever necessity, we rose to the occasion. We have been contributing. Look at this, starting from coal, fine chemicals, pharmaceuticals, polymers, whatever listed here, waste to wealth, and recent times, circular economy, chemical biology. We are into this one. That's what we have been doing. And we are 150 plus scientists and 300 plus staff and 500 plus research scholars, and including dissertation student, contract staff. We have about 2,000 footfall every day. So our major focus is, although we talk about majorly focus on chemicals and chemical technology, our major, these 4 areas we work: healthcare, chemicals, and energy and environment. So that's have been our focus. So we have been contributing so much to the country and to the people, to the society. But I would like to give one example. This is linked to Gujarat. So here is one compound called hydrazine hydrate. And also this is also some kind of association with Professor J.S. Yadav. When he was a director, he started this project, and now it is showing the fruits today. And look at this one. This is a hydrazine hydrate. This is very, very important key starting material. And this is global requirement is 2,000 tons per annum today. And India requires about 20,000 tons per annum. So that's where we are right now. Till now, we are depending on China and other countries to get this material. And this is very useful material. It is a starting material for agrochemicals and APIs, and also as part of rocket fuels also they use this material. This is very, very important material. And IICT started about basically about 15 years ago. We started this program, and now, although we started with the bench scale process, pilot scale, then we connected with the Gujarat-based company GACL, and we together we developed, and now that company established a plant which can produce 10,000 tons per annum. Very recently, our Honorable Prime Minister dedicated that facility to the nation, and some time ago, first commercial production came out. That day, I was happened to be there, and we were there, we were invited to that flag up, that ceremony. So that's kind of a great satisfaction for our AICT and CSIR. That's a great moment for us, where we started with the small scale, with bench scale, and now we are seeing that tons per annum, like several tons per annum production is taking place. That's contribution we have been making, right? Of course, we also made many other things we can talk about it, but one example I'll give you. Recent times, we all suffered with COVID, right? So we developed a process for drugs. In addition to that, Covaxin, we closely worked with Bharat Biotech, and we developed a process Raju Reddy is sitting here, who played a major role in that in the process development of Covaxin adjuvant, that TLR7 agonist we call. So we made a few kilograms of that material and gave it to Bharat Biotech. So that Bharat Biotech used that material, and which produced like all the vaccines, not only in India, exported to the different countries. Here in this audience, whoever has Covaxin shot, so I can tell you proudly is that you all have some component of IICT in your body today. So that's kind of contribution we have made. So coming back to my research, and what we do is, in our research, after spending few years in industry, and when I started my career at NCL Pune, so we started to working on total synthesis and medicinal chemistry. These are the major areas we have been focusing. In the morning session, Professor Vinay Singh also told me that many people synthesize molecules and leave it there. So we are additionally we are doing that. We are trying to convert whatever we made molecules, we are converting into medichem programs, trying to do medicinal chemistry because of my industry experience. That's what I'll showcase a couple of things, what we have been doing in our research group. Right here, I'll give you a couple of examples. How are we proceeding? We pick up molecules, and how do we move forward? Look at this molecule called peganotonin. A couple of years back, isolated, and from this particular species, Peganum harmola. That's a plant. It produces from the seeds. They isolated this compound. And why we became interested in this molecule? Look at the structure. It's like a benzodiazepine. Many drugs are there with that kind of similar scaffold, and we call it as like a druggable scaffold. It's like structure looks like a druggable, and we calculate a Lipinski rule of 5. those who have little experience in medicinal chemistry, those who know about Lipinski rule of 5, if it follows that rule, there's a higher success of chances of becoming drug. So that's where we calculated parameters and perfectly fits very well with this Lipinski rule of 5. We picked up this molecule and we wanted to synthesize. And why we want to synthesize? We also have some history. This particular plant has several biological activities like anti-tumor, antiviral, antibacterial, and also some of the neuroprotective effects. So since it's a druggable scaffold, we wanted to make in our lab and also generate library of molecules and move forward. That's what the idea when we started this program. Right? We simply look at this retrosynthesis. Any molecule, although it is not a complex molecule, it looks simple molecule, but if you can come up with a good strategy to make this compound, then you can make a library of compound. That's what we look at. Look at this one. This is a final molecule. We thought we can do this indole-based tricyclic compound. If you have this one, if you cut this double bond in between these two rings, then you can get this final compound. You look at this one, exactly the same compound, right? So that's what we wanted. How do we make this compound? Very simple. This like if you take like a melatonin or like a tryptamine estamide, like estate protection tryptamines, if you take, and you can make this compound, this cyclization. That's what we wanted to make. Very simple synthesis. We can make large number of compounds, new compound, new skeleton. This is called benzodiazepine. Right? How did you do? So we took methoxy indole. We followed the known protocol to make this methoxytryptamine, and we protected with basically acylated. Very simple reaction. It goes this primary amine. And next, we do some protocol we developed. This tertiary butoxychloride we used. Of course, some related reactions are known in the literature. We cyclize this compound. The next step is ozonolysis. Cut the double bond, you get to this material, right? So in few steps, we could make this compound. In 2 weeks, we could make this compound after planning. That's very fantastic student we have right now. So we made this compound. Then when we looked at the structure, the NMR was not matching. Then we got doubt whether we made right compound or not. Then we took crystal structure. It is exactly like whatever we made is right, whatever we proposed or whatever we made is right. And it was that NMR is something is wrong. So although our NMR is not matching, but that was not the main goal for us to make this target molecule. Our goal was something else, to make the molecules and test for biological activity. For that purpose, then we looked at what are the possible things we can think of this like derivatize and make towards drugs. So for that purpose, we use this phenyl ring for different substitutions. Derivatization you can do, ketone you can do alkylation, substitution, anything you can do literally. This is like very functionally very well embellished molecule. You can do many molecules. They are all going to be a novel compounds, at least based on our at least initial search. Right? So with that in mind, first thing is we wanted to fix this structure, whether it is really, what is this, why it is not matching. Then first thing is we thought that we play with this methoxy group. Can we switch different positions of this phenyl like aromatic ring? So we tried to make different methoxy group. All compounds, some of them are commercially available, some of them are made. And we corresponding tryptamines and isolated, and same followed whatever we did in the previous slide, we followed and we made this compound. Look at this one, all 3 compounds we made and none of the compounds are matching with the literature report. So again, that means there is something wrong with this compound as such. So then, of course, as part of our goal, we synthesized about several compounds, like about almost like about 15-20 compounds. We made these compounds. We tested a few different biological tests, and one of the thing we tested next door, that is a CCMB, and Dr. Kiran, we tested antiviral activity using SARS-CoV. So look at this, a couple of compounds became very attractive, 99% inhibition. So now IC50 is also run into less than 1 micromolar. So this actually one of them has 160 nanomolar compound also we have. So right now we identified a couple of compounds and moving forward. And also we just filed a patent. I can tell you when I went for interview, my directorship, one of the criticism was IICT and CCMB close to each other, but they never collaborate. And Professor Tapaskondu may be knowing that one. So we are probably, this is the first patent came out of IICT, CCMB after I joined that group, that IICT. So that's something, again, cherished moment. Sir, Professor Yadav sab, aapko bhi malum hai. Yeah. Yeah. So I thought if I start, then others will follow. Then that's what I started. Now others also following. So again, we wanted to figure out this molecule mystery. Although we made several molecules, did not figure out. So again, another connection with Gandhi, like Ahmedabad is I visited a few months ago IIT Gandhinagar. So I met Roderick Bates. So I was discussing about this molecule. There is something wrong with this structure. Then he connected me to a couple of other people, Craig Williams and this couple of other people who are good in computational predictions of basically NMR computational predicted structures. They can do that one. He connected me with all those people. Then we found that the structure is something like this. How did we arrive this structure? That is something interesting. You look at this one. There in that NMR, there are a couple of impurities are there. When we analyze that NMR has a couple of peaks which are related to this diethyl. This is difficult to miss, but there are impurities. They could not integrate those compound. That is another one. Another one is when you fragmented mass of this particular compound, that is exactly matching with this proposed structure. So that's how they missed out. This original compound is 335, but 263 is a fragmented base that is exactly matching with this. That's how they proposed this structure. Then they did the advanced techniques for calculating NMR. Hey, it's not working. Click on that one. Click on the slide. Click on slide. Change karo, next karo. Okay. So they came up with a different structure, but the one which is in the first one, so that matched very closely this compound, that whatever the predicted structure I showed you, this one, this is the one, this one. Then what we did is we took this acetylated compound, pyridate cleavage, and again hooking with this diethyl formaldehyde, you get to this compound, exactly matching with the spectra, proton and C13 NMR, everything is matching. So completely different structure. So that is something benzodiazepine, but this is something open form. It's exactly matching. Just recently we submitted, all together we submitted this paper to this Journal of Natural Product, which is under revision right now. This is something completely out of the way it came out. Okay, now next I'll switch it to a different molecule. So this is one of my favorite project in my research group. This is macrocycles, isolated more than 10 years, almost like 14 years ago, when I started in NCL Pune. I saw these molecules. So why it attracted, this is very potent molecule. It showed 100 microgram per kg dosing. They showed 60% pi-edema reduction. So that's very potent molecule when compared to other dexamethasone, other molecules. We have to see like 10, 15 milligrams we need to use. So why it is important? Because inflammation is actually is always good for us. It serves our basically protective function for us, for all of us. But if it is uncontrolled, it can lead to several diseases. You can see the listed diseases here. And all these diseases can be like every organ, every part of our body can be affected by inflammation. There are so many diseases it can be linked with the inflammation. If you can come up with a good druggable compound with which can control inflammation, then there will be a good chance to success in moving forward. Particularly these macrocycles are taking much attention. So we became interested on this molecule. Just to give you a summary of this whole project. So our group has been working for almost like now 14 years. We have been continuously working on this one. We published patents, filed filed patents, published papers. In addition to that, a couple of other people like Chandrasekhar group, Sarabia group, elsewhere the group, couple of people also working. That's so attractive, this molecule. And I can tell you that this major project, 2 SCRB projects and 4 PhDs, and also 7 papers and a couple of patents from this single molecule. That's we have been contributing. Still we are continuing. I'll give you update what we have at this moment today. So here is the 2 molecules, solamonamide B and solamonamide B A, 2 molecules. You see the difference between these 2 molecules is the hydroxy here. Otherwise rest all same. So we wanted to make both these molecules starting from this olefin microcycle. How are you making? See, look at this one. B, if you can do Wacker oxidation, you can do selectively, can move towards benzylic position, you can get this compound. Similarly, if you do dihydroxylation, and followed by oxidation of benzylic hydroxy group, you can get to the solomonamide. That's why these two molecules are identified, like basically the strategy we followed to make this compound. How do we make this compound? Then we wanted to use this HEC cyclization, intramolecular HEC cyclization to get there. The acyclic precursor can be made from corresponding dipeptides and one this acid, which like 3 chiral centers. That's what we wanted to make. That's a key starting material we need to make. We need to worry about. For that purpose, we started with the D-methionine starting material, and we used some of the protocols to get to this particular compound. From there, you see that the difference between these two compounds is one side is hydroxy, another side is olefin. But what we want is this compound. We need to switch between this hydroxy andyl to olefin and olefin to carboxylic acid. That's what we need to do. For that, what we came up with this idea is, can we use basically we converted into phenylselenide derivative. We used ozonylysis. Ozonylysis we know that we use for cutting double bonds, right? Oxidation of double olefins we use. At the same time, in this process, one of selenium get oxidized. When you bring it to room temperature, that get eliminated, you get to olefin. So we call it as like ozonylysis can be used for. We all know that breaking olefins, but we can also use it for making olefins. That's something title we gave that one, breaking and making olefins simultaneously using ozonylysis. This is something fantastically worked with us, and we generated good methodology out of it. After making that product, then we hooked with the dipeptide. We made this acyclic precursor. Then HEC reaction, although I'm showing you the final optimized yield, we struggled a little bit in cyclization. We got this cyclized product. Once we are cyclized product, the next job is just to make sure that whatever we made is right, what stereochemistry right. For that purpose, we protected this this compound that basically kind of making a restricted rotation. We made this compound so that we crystallized, and all the everything is matching with whatever we predicted. Once we are that there, the next step is installation of serine moiety and followed by benzylic Wacker oxidation can give you the product. So for that purpose, we tried, serine installation happened, then Wacker oxidation using palladium chloride, copper chloride gave us desired compound. Our next step is remaining only deprotection of both Bach group and benzyl group. Only these two are we need to remove. For that purpose, we used, initially we used palladium, charcoal, hydrogen methanol condition we used, followed by deprotection gave us this compound solomonamide B. But after doing this one, the spectral data do not match again. This again after 4 years of work, then a student was almost like a frustrated stage. So then we looked at the analysis of this whole structure again, what could have been gone wrong. So they used for analysis Murphy's method. That is a very well standardized method for fixing stereo centers of amino acids. So there is no question of going wrong in serine case or in case of the alanine case. There is no question of going wrong. And only possible is going wrong is here stereochemistry of this compound. We'll do the NMR carefully, and there could be some problem with the stereochemistry. And 3 chiral centers. 3 chiral center means 2 to the power of 3. That means 8 compounds, 8 possible macrocycles we need to make. So these are the 8 compounds, right? So 8 compounds we need to make. If I ask 4th year student to make 8 compounds, then he definitely will not like me and he will go mad at me. So then I requested another student who joined at that time in my group. So together we asked them to make this compound, not only for fixing the structure, our goal is also to generate libraries and take towards medicinal chemistry. That was ideal. That was the idea of going forward in the queue. So I'm showing the final slides of this synthesis. So we went this D-methionine derivative. We did some of the aldol reaction, and finally we get this acyclic precursor. Once you know acyclic precursor, then cyclization, macrocyclization using HEC reaction, you get this olefinic macrocycle. Once we are olefinic macrocycle, make sure that whatever we made is right, crystal structure. Then at this stage, serine installation, again, using routine chemistry we followed, and Wacker oxidation gave us desired compound, and removal of protecting group gave us the final compound. Everything is matching perfectly with this particular isomer. The difference is the stereochemistry here. Rest all everything is same. The stereochemistry was assignment was the wrong original structure. That's what we proved it. Once we are there, then we wanted to make solomonamide A. That is basically with additional hydroxy group. For that purpose, we took this common macrocyclic precursor. We used osmium tetroxide mediated hydroxylation. We got this compound. Interestingly, we got a single NMR. We thought we get mixture of compounds, but we got a single diastereomer. Then we used oxidation condition using DMP. We got the desired compound. Then serine installation and deprotection of both the protecting group, benzyl group and Bach deprotection gave us solomonamide A. Again, everything is matching fine. So we assigned structures, reassigned structures for both solomonamide A and solomonamide B. So that was not the end, right? That's where we wanted to get into medchem program. So for that purpose, this is what we did. The next step is we wanted to get into medchem, right? For that purpose, whatever we synthesized, all the macrocycles in our lab, about 40 analogs we synthesized. We collaborated with the CDRI, with Dr. Manoj Batwal. We did carrageenan-induced paradigma model. We found that couple of molecules are very interesting. Simplified molecules became like better than original molecule. So what we did is, this is the original starting point. We revised the structure. We made about more than 30 to 40 analogs we made. We came to the compound. This is a simplified compound. And these are all SIR. We did like basically different strategies we followed to make this compound. Various strategies. We published a lot of literature. And some time ago, we published JOC. This very students who are interested, they can see this paper. This is actually almost like 45 papers and more than a couple of hundred supporting information. There's a lot of information available. I don't think I saw any recent paper which published 45 published pages in JOC. So next is, once we have this identified simplified compound, what next? Our goal is we also to find out the best out of it. Again, this molecule. What is that we are going to do? There is something in our lab we are trying to introduce is, we all know that structure activity relationship, right? So now we want to add one more S, stereo structure activity relationship, SSAR. So that's what we are trying to do here. In this purpose, we made some other projects, but in this context, there are two methyl groups, right? So can we think of making all possible combinations here? Two means like 2 to the power of 2, that basically 4 compounds. Can we make 4 compounds and put them into biological assays and come up with the best compound? This basically lead identification through SSAR. So can we think of doing that one? For that purpose, what we did is we made all the 8 compounds, basically 4 with the double bond, 4 with reduced form. So we made both like 8 compounds. All the 8 possible compounds we made, we subjected them into initial assays, and we found that a couple of compounds are looking good. Then after that, we also picked up these 2 compounds, 2129 and 2127, where they showed good activity in anti-inflammatory activity in PA edema model. Then we did pharmacokinetic study. 2 compounds showing very good oral bioavailability. Out of them is more than 90% bioavailability. Generally, peptides are not that good in pharmacokinetics, but this compound seems to be a good compound because of cyclic nature and simplified nature of this peptide. Once we have this compound, basically what we did is, after SAR, we identified this compound. During our SAR and PKPD, we identified a compound, 2129, as a potential compound to move forward. Then we are doing various assays with our colleagues at IICT, Dr. Balaji and Dr. Sisla. They are working with us. There are different models are there. Psoriasis model, ulcerative colitis models are under progress. But we do have very initial results here. And this is so basically, DSS is a dextran sulfate sodium that they use for model development for this colitis. And what we have, again, colitis is again, people who doesn't know about this one, these are serious problems, like basically bloody diarrhea, abdominal pain, constant urge for defecate. Basically, you want to go for toilet immediately. That's kind of a problems there. This is a serious problem. There's very few medications are available. And according to some numbers, in 2023, the prevalence of ulcerative colitis is estimated to be 5 million cases around the world. That's something numbers what we have. And this is a model we developed at our IICT, Dr. Balaji and a group. They developed after, like, we picked up these particular doses, like both compounds 2127 and 2129, 25, 50, 100 mg. So the three doses we picked up, and we wanted to measure all the parameters of ulcerative colitis model. So I just show you one result here is, this is something interesting to watch out. And one of the measurement is they measured colon length. So colon length, they measure like basically whether it can come back to original starting point. This is a normal one. If the disease one, it goes to like this. After 3% DSS, after developing model, it goes like this. So these are all our different doses, different compounds. You look at this particular compound, 2129 100 mg per kg. It is almost getting close to like original colon. So that's basically kind of, we have very positive results in vitro, in vivo, and beautiful structure SAR. Where we started, original starting point, we started with a simple structure assignment and basically redesigning the original structure and SAR in the beginning, then followed by SSAR, serial structure active relationship, and we have simplified compound which is optimized, including PK parameters, and now finally we are getting into animals. Hopefully we are moving forward in this direction. This is something started from very basic to now we are going towards application, and I think that's what Professor VK Singh wanted people to do the similar things. I'm in that direction we are looking at. So last few minutes, I'll connect with this conference. Also, we talked about some of the drug discovery, but I'll also connect with like sustainability, towards sustainability development. How are you going using process development? I'll give you one example, which is in my research group ongoing. This one particular molecule we are interested, this lifitograst, used for dry eye syndrome. So basically, it is like you get tears in your eyes. These days, it is going to be very common, and also it's going to increase burden also, dry eye syndrome, where because of our digital platforms, like all computer, most of the time we are all phones. So this can cause into major problem. This is expected to increase market for this lifitograst. This is a marketed drug. It's approved in 2016. And there are 3 intermediates which are important for making this particular compound. Fragment 1, 2, 3, these three compound. We are interested in fragment 1. We looked at the cost, and of course this is one source. In another source, we also got quotations also about 1 kg more than 1 lakh. So that kind of cost it was available when we started, when we wanted to synthesize. Lengthy synthesis, and in addition to that, they also use transition metal complexes, including even as part of Sonagashir also they use, and expensive starting material. We wanted to simplify this make this compound in a simple route, as simple as possible, as scalable. So for that purpose, what we did is we took very readily available material. We used this magnesium chloride and methyl THF formaldehyde. What we get is we get about 35%. This is the best we could from this optimized condition. And then we did aqueous HCl treatment. We get both the compounds. Then you use a very old technique. We use the sodium bisulfite techniques to separate one adduct, and we starting material recover, and finally recover it back, and you make this compound. So basically 95.6% purity and more than 50% starting material recovered, and we scaled up to 100 grams this compound. Again, that's something we did in our laboratory. And finally, after making this aldehyde, then sodium chloroacetate, sodium hydroxide gave us this particular compound. And finally, sodium acetate treatment gave us benzofuran 6-carboxylic acid in good quantity, which has about 98% HPLC purity. Still, we need to work on this one. But again, this we did without much experience in process development with the students, PhD students they did. And we are talking to some of the people whether we can take it forward. This is what we are. And at the same time, again, in the 20-gram batch reaction we did, and also this 100-gram batch reaction also we did formylation reaction. Although we have only 35% at the best optimized condition we have, but this is what we could do our best at this moment. And and I collaborated with Dr. Venkata Mohan, who is good at LCA, that life cycle assessment, and the sustainability analysis. So we found that based on his inputs also, we changed some of the solvents like THF to methyl THF and a few other conditions we marked. And in fact, we showed that this also reduces this carbon footprint, our route, when compared to existing route. This is again trying to get into that one. This is another new area, people we need to watch out in the industry. It's going to be futuristic step. Although it is not a mandate this moment, in future there can be a mandate from the government. We need to calculate this LC analysis and also how much carbon footprint is going to be there in our globe. So this is what we have been doing. What we do in our group is we work on simple organic synthesis, but we connect with people who who can make value for those compounds what we make. So all my tenure at NCL, IACT, or other places wherever I worked in Jammu. So all the time we collaborate with people and we create value for the molecules what we make. So more than that, whatever we have leads or semi-leads, hopefully see the light of world in the future, in the near future. More than that, I am satisfied that students who worked in my group, they get at least oriented towards application oriented, where we can something we can feel good about what we do synthesis. How can we apply to our problems? That's what we have to do. So this is what I have a great research group, and particularly who are in the out of this box. These are all students who helped me in my research group. So I always think that, again, I also suggest to people who are starting their career, first students, they can really make or break your career. I think I was lucky to get good starting points, starting students, and they made my career today what I am today here because of all my students. And these are some current research group at IICT. Last few years, we are shuffling between Pune, Jammu, Lucknow, Hyderabad. Now came back to Hyderabad. Hopefully, we'll continue to do some of the work what we have been doing. And finally, I would like to thank you all for your patient listening.
Thank you, dear sir. So, in the interest of time, I'll take only one question. Yeah, please.
Animal model, acute model for carrageenan pyoedema. In another case, you have used DSS-induced colitis. So what is the mechanism working? Is it a cyclooxygenase pathway or something else?
I don't know exactly, sir, but what we use in the beginning is that is for quick readout, acute model. When you use it, you can filter the molecules.
Cyclooxygenase pathway.
Yeah.
But we don't know.
We don't know at least.
Which pathway it is working.
Right now, we don't know which pathway it is going. Maybe I'll talk to you if you have some ideas, then we can discuss on this one. We don't know at least which mechanism also, we don't know. Right now, only phenotypic, whatever we have this one.
No, it's working very well.
Right.
But somebody, if you want to approach industry, what is the mechanism? So then they would be able to decide to compare to which molecule we should compare, where we should position, how it should be positioned.
So in the case of initial screening, carrageenan screening, we used dexamethasone.
Correct.
So that's what I remember, that dexamethasone
correct.
we used. Again, we don't know the mechanism at this moment. Again, probably we need to pull out the targets and we need to work a little more on it. So right now, whatever we have is, this is what we have right now. Yeah. Thank you.
I'm sure there will be many more questions, but I request you to catch up the speaker who is here till tomorrow morning. So let's have a big round of applause for Dr. D.S. Reddy, please. Oh yeah, Jadhav sir ka question hai. Oh, okay.
So no,
So tomorrow early morning he is leaving, so you have to catch today. Okay. So by evening. Thank you, dear sir.
Thank you.
So, great
Thank you
talk.
so much, sir. I request Professor Alakesh Visai to felicitate Dr. D.S. Reddy with Charkha. Thank you so much, sir. Now, before we proceed for the lunch break, there is an important announcement. Now I request Professor Sriram Gundimeda, Head of Department of Chemistry, IIT Gandhinagar, to felicitate the session chairs, Professor Tapas K. Kundu and Professor Alakesh Bisai, and give them a token of thanks. The parallel poster presentation will be from 2:30 p.m. in B block. The judges for the poster presentation are B block. Yeah, parallel poster presentation. The judges for the poster presentation will be Dr. Janakiram Vaitla, Dr. Navinita Sadhu Khan, Professor Ravi P. Singh, and Dr. Sandeepan Haldar. Ladies and gentlemen, we will now take a break for lunch, and I request all of you to return back by 2:30 p.m. sharply for session 3.
वर्चुअल वाले में कौन है अभी जो वर्चुअल वाले में कौन है तुम लोग बैठे हो वाले में हम यहाँ पे भी मल्टीपल जगहों पे कैसे बैठे हैं
Hello, Professor Cozy. Are you able to hear?
Yes, I'm here.
Okay, thank you so much. Welcome to SIDD 2025. So soon we'll be starting your session. I'll just inform you in a bit. Yeah, thank you.
Can I share my screen?
No, please, please wait for some time. I'll tell you in a bit,
Okay.
because
But
we are
I would
waiting some
like to try in order to see if everything is in order.
Okay, okay, fine. I think then. Yeah, so kindly, I'm proceeding, like handing over to the technical team. Kindly share, yeah. Our technical team, Mr. Sudeep Kumar, he is handling, so he's taking.
Can I share?
Yes, ma'am. Yeah, kindly share that. Yeah, yeah, please.
Can you see it?
Yes, yes, we can see that your name, but slides we couldn't see.
Okay. Oops. Can you see it?
Yeah, yeah, we are working on it. Give me a few seconds. I'll let you know.
David, je pense que je vais avoir un problème avec ma connexion. Ok, merci beaucoup. Merci.
Hello. Hello. Good afternoon, ma'am. Can you hear us?
Yeah, I can hear you.
Hello.
Hello, Professor Koji. Did you share that? Share your slides?
Yeah, but I think you cannot see anything.
Kindly first click the share button and then entire screen. You have to click share. Then
Can you see something? Hello? Hello?
Yeah, Professor Koji, but we are not able to see your slides.
I asked a guy to come because I mean I have some problems in here.
Yes, because here we are our technical. Hello, Professor Koji, can you please log out and then again log in?
Well, I try, but I mean, I cannot do it. I mean, when I am sharing, nothing is happening.
Oh, I see. So our technical team says that from here everything is fine. So, okay. Okay, please wait for a bit. Hello.
Sound us.
Oui. Désolée. J'essaie de...
Oui, alors est-ce que vous êtes parvenu? Alors.
Yes, we can hear you, your voice.
Okay, we try to share.
Alors, écran.
So from our side, we have given permission to share.
C'est lequel que vous...
C'est celui-là.
Celui-là derrière. Alors.
Can we request you to share your PPT? Oh, now we can see that.
Can see it?
Yeah, we can see you.
Okay. Donc c'est bon.
Alors c'est bon, alors du coup. Yes.
Parce que je vous avais dit, je regarderai après.
Voilà. Alors,
Now, now I have to put it in full screen. Oops. Can you see it? Can you see my screen?
No. Yes, we can see you.
Okay.
Not the screen.
Is it okay?
Professor, we can only see you.
You cannot see my screen?
No.
Oh, God.
Okay, you will need to share entire screen.
Yeah, we tried. Zoom. No.
Alors, qu'on ne voit pas du tout. Il y a des PDF.
C'est peut-être moyen.
Non. Normalement, c'est tout simplement cliquer partager et vous devriez voir maintenant le document. Alors, normalement, c'est bizarre, je ne vois pas votre document. Le document que vous l'avez ici,
Et-
vous l'avez. Est-ce que vous pouvez me dire où vous l'avez mis?
Ah, il est sur le bureau.
Le bureau, d'accord.
Ah,
C'est celui-ci.
non. Ah, voilà, pas. CIDD. Ah, oui, c'est dingue ça.
Peut-être ça colle à ça, parce que comme il n'est pas sur le bureau,
Il y est. Ah, ben maintenant, c'est S. Ah, pardon.
c'est ça, alors attendez.
Voilà, c'est celui-là.
D'accord. Oui, pas vu.
Ici. Oui.
C'est ça. Et
Professor Cozy, alternatively, you may share your slides.
Yeah, but I cannot share. You know, I have the technical guy who isn't here, but it seems that there is a problem.
Through email, we are asking.
Hmm?
through email.
Oui,
We are asking to share you through email.
So, I mean, which mail?
I'm sending.
Alors, est-ce que dans ce cas-là, on va balancer. Tac.
Email shared.
Okay. Well, this one? What is this?
Alors, on peut glisser peut-être sur le...
Je comprends rien de ces conneries. Did you send the mail?
Alors, on peut peut-être glisser sur le... Comment ça s'appelle? Sur le... On peut glisser le document.
Où ça?
En fait, il est sur le bureau.
Oui, et je le mets où?
Vous glissez ça sur le... Alors,
Donc j'enlève ça.
oui, alors, celui-là.
Oui, c'est celui-ci.
Voilà.
Et je le mets où?
Voilà, vous glissez ça dans le chat.
Oh. Oh, purée.
Did you already share the slide?
We have a technical problem. We can share the document. Ok, ça marche pas. Alors, tac.
C'est en bas, parce que ça ne m'a pas marché là. Ça, vous cliquez là.
Oui.
Là. Non. Ça.
Ah oui, d'accord.
Mais ça ne marche pas.
Alors, on va envoyer cette adresse-là, en fait.
Alors, comment il faut faire?
Apportez votre messagerie.
Kindly share your slide through email we have sent.
Please, one moment.
Alors, on va faire, vous cliquez ici, tout simplement. Tac. Voilà.
OK.
Et donc là, du coup...
Non, celui-ci.
Glissez ça là-dedans.
OK.
Voilà. Et il faut l'envoyer. Voilà, tout simplement.
Ah, purée, ça ne marche pas.
Qu'est-ce que c'est que ça? Alors,
Tentez.
voilà.
Ouais, moi, je ne sais pas si ça va fonctionner. Hein.
Alors, vous pouvez répondre aussi à...
Did you have it? Did you get it?
Checking.
Do you have it?
Not yet.
Ah. À mon avis, ça ne va pas le faire l'affaire, ça.
Received.
Receive it? Oh, good. It's a PDF, so you have to put it in full screen if you can do it. Je comprends pas que ça marche pas.
Ah oui, je sais pas. Je vois ça avec... à la fin, sur votre poste. Est-ce que... à quelle heure, là?
Can you see the slides?
Yeah, but it's not full. Oh, great. Yeah. I got it.
You may tell us when to change.
And I cannot change it.
After every slide, you may say that next slide.
But I cannot change the slides. Hello?
You won't be able to change.
No.
But after presenting one slide, you may say next slide.
Okay, good.
A very good afternoon to all the esteemed dignitaries, distinguished.
I cannot hear you. Excuse me, I cannot hear you.
Hello?
I cannot hear you, please.
Hello?
Yeah.
Ma'am, can you hear us?
Yeah.
Hello? Hello?
Ils sont fermés.
Du coup, le micro est fermé, effectivement.
The microphone is off.
Okay, we are
Oh,
here
okay.
to
Yeah,
introduce you,
yeah,
ma'am.
I can hear you.
Can you hear me now?
Yes, yeah, yeah.
We will make a brief introduction of you. First of all, good afternoon from Industrial University and welcome to the SIDD conference. I'll briefly read your profile. Dr. Janine Kosi, Emeritus Professor, Organic Chemistry, ESPCI, Paris. She obtained her PhD in chemistry from the University of Reims before pursuing a postdoctoral fellowship under the guidance of Professor Barry Trost at the University of Wisconsin-Madison. In 1990, she was appointed as a professor at ESPCI Paris Tech, where her research has been dedicated to the total synthesis of biologically active natural products, including anti-cancer agents, antibiotics, anti-inflammatory compounds, and molecules affecting the central nervous system. Additionally, her work encompasses free radical and photochemical reactions. She has served as a consultant of Rhone, Pollens, Rhodia, and L'Oreal, co-founded the startups Acanthe Biotech and CDP Innovations. Dr. Janine Kossi has held key positions of organic chemistry, serving as president of the Organic Chemistry Division of the French Chemical Society and the Franco-Japanese Chemical Society. Her accolades include the Zinc Fish Prize, CNRS Silver Medal, Grand Prix Achille Labelle. She was knighted in the National Order of Merit and the Legion of Honor. Recognized by the pharmaceutical industry, she received awards from Novartis, Boehringer Ingelheim, Eli Lilly, Abbott, AstraZeneca, and Bristol Myers Squibb. A member of the IUPAC, she received on the Pierre Gilles de Jeunes Foundation and CNRS Scientific Council. She is also an editor of the Organic Letters, the New Journal of Chemistry, the European Journal of Organic Chemistry, and the Journal of Organic Chemistry. With this, we welcome her to the plenary session. Her talk title is Powerful Tools for the Synthesis of Bioactive Heterocycles Towards Sustainability. Ma'am, you can please start your talk.
Thank you so much for the introduction. And I would like to thank the organizing committee for the invitation. It's really a great pleasure to be part of this program. I'm sorry about the technical problems, but I hope we will be fine afterwards for the talk. Please, next slide. Next slide. I cannot go for, yeah, thank you. What is our portfolio? We are involved in multidisciplinary projects with physicochemists, biologists, and physicists in order to do the vectorization of drugs toward the tumor. In order to decrease the secondary effect. But before doing the vectorization, you have to synthesize the compounds, and we are involved in the total synthesis of natural products and complex molecules with which are bioactive. And in general, when you are in the synthesis of complex molecules, you are in front of synthetic problems. And it's why we are developing synthetic methods and enantioselective reaction, rearrangement, organometallics, and now enzymatic reaction. Next slide, please. So now we have new requirements in chemistry. We need to use abundant and renewable sources with low toxicity. We have to develop catalytic processes which are selective, step economy and atom economy. And we have to limit the waste. Next slide, please. So if we are in the if you want to use transition metals, you have to better to take this part of the diagram, the left-hand side, and to use iron, for example, manganese, zinc, nickel, copper, cobalt. But you have to avoid these transition metals that are really rare, which are on the right-hand side. And you have to avoid ruthenium, palladium, rhodium, iridium, gold, for example, and platinum. To avoid that doesn't mean that you have not to use them, but if you can avoid them, it will be much better. So next slide, please. So at first, let's consider, for example, the compound which is represented in here, ML3000. ML3000 is an anti-inflammatory compound, and it is a non-steroidal anti-inflammatory compound with no gastrointestinal ulceration and no bronchoplasm side effect. And in fact, if you want to have an anti-inflammatory that is really active, you have to be very careful about the inhibition of COX-2 and 5-lipoxygenase, and you have to have a right balance. And it is the case of ML3000 because, I mean, the EC50 for the inhibition of COX-2 is 0.21 micromolar. And for the 5-lipoxygenase, the EC50 is 0.18 micromolar. You can see that this product is constituted by a 5-membered ring. You have a pyrrole and 2 aromatics groups, and also a side chain in the alpha position of the nitrogen with the carboxylic acid. Next slide, please. So, I mean, Merckle has synthesized this compound from a very cheap starting material, this diol in here, and in 8 steps, they could have the formation of MS3000 from this bicyclic compound, this insaturated, this enone in here. But I mean, this company has only an overyield in MS3000 of 0.5%. So if you want to make some profit about to sell this compound, you have to increase the overyield. So how can we synthesize this molecule? Next slide, please. Next slide, please. So we have envisaged to synthesize MS3000 from the same intermediate as Merckle, this bicyclic enone. And this compound should be coming from this enamine, where you have this cyclic enamine that can react onto the ester, which is activated probably by a Lewis acid. And this product should be coming from this acetylenic amine. So how can we transform this acetylenic amine to the corresponding cyclic enamine? Next slide, please. Well, probably the thing that we can think about is that if you are treating, for example, this acetylenic amine by some metal, a silver, gold, or platinum, you will have a complexation of the triple bond, and then the amine can attack this activated triple bond in order to get the formation of this enamine. But I said before, you have to avoid this kind of transition metal. So what is the second hypothesis? Next slide, please. Well, probably if we have this acetylenic amine on the right-hand side, and if we have a thermal reaction, maybe we will have an in reaction. So the abstraction of the hydrogen in the alpha position of the amine, and then the amine can attack this acetylenic in order to get the formation of this halide. And then after a hydrogen transfer, probably we can get the formation of the enamine. So we try to see if we can have this kind of transformation in a thermal conditions. Next slide, please. So if we have synthesized this acetylenic amine, and then we have heated this compound at 200 degrees, and we obtain the formation of the cyclic enamine with a yield of 45%. It's not great, but the reaction is working. Next slide. But if we are adding next slide, please. If we are adding one equivalence of pivalic acid, now we can decrease the temperature to 130 degrees, and the yield is increased to 80%. So we knew that this kind of thermal reaction is working. Next slide, please. So, I mean, we obtained the formation, and now we have to know if it's an in reaction. So we prepare this compound with 2 deuterium in the alpha position of the amine. And if it's an in reaction, probably we will have a deuterium in on the double bond. So we did the transformation. Next slide. We heated the compound, and next slide, please. And we never observed the formation of a compound with a deuterium on the double bond. So it's not an in reaction. Next slide. So probably it's only the reaction, the addition of the amine onto the triple bond that lead to the formation of this cyclic enamine. So now, as we knew that this kind of transformation was working, we tried to apply this kind of transformation to the synthesis of Ml3000. Next slide, please. So, I mean, if we want to have the formation of Ml3000, probably we have to synthesize this cyclic enamine that will be coming from this acetylenic amine. Next slide. So we started from a very cheap starting material. In 2 steps, we could get the formation of this chloride, and then after condensation with this butylaldehyde, probably we had the formation of this aldehyde under the phase transfer condition and under basic condition. And then after a reductive amination using this glycine, we can get the formation of this acetylenic amine. The yield is pretty good. And then we have to transform this compound to the cyclic enamine that will be the precursor of the bicyclic enone that was obtained by Merkle. So, next slide. We took this acetylenic amine, treated this compound by pivalic acid at 130 degrees, and we never observed the formation of this cyclic enamine. But, next slide, we obtained directly the formation of this intermediate that was obtained by Merkle with a yield of 80%. So probably we have the activation of the ester, and we have the formation of the cyclic enamine intermediate, but now it reacts directly on the ester to get the formation of the cyclic enamine. So now we have to transform this bicyclic compound to Ml3000. Next slide, please. So, I mean, at first we tried to introduce this second aromatic group with a chloride. So for this, we used a Suzuki coupling reaction. We treated this enone by NH in the presence of this triphycamine, and we obtained the formation of the corresponding enol triplet. Now, by using a Suzuki coupling reaction, by using palladium 0 in the presence of a base and this boronic acid, we obtain the formation of this bicyclic aromatic, this aromatic compound. But, I mean, next slide. But by doing this, the yield is really poor, 5 mol%. So that means that we are not doing much better than Merkle. But the students make a very important observation. Next slide. I mean, when we were looking at the crude NMR, I mean, the product was clean. But then after purification, when it started the purification, on the column, you can see yellow, red, and then black. And we have something that is black in the flask. So that means that we have a polymerization. So, I mean, the compound that we have synthesized is not really stable, but ML3000 is very stable. Why? Well, because you have a side chain in here with a carboxylic acid, which stabilizes the pyrrole. So probably instead of introducing the aromatic at first, we have to introduce a precursor of this side chain. Next slide, please. So the thing that we did, we took this bicyclic ketone, and then we treated this compound under basic condition in the presence of ethyl oxalate, and we obtained the formation of a diketone, which is enolized. This is very good because we need to use a Suzuki reaction in order to introduce the second aromatic group. So we treated this bicyclic ketone in the presence of NH, in the presence of the triflicamine. We obtained the formation of the triplet, and then we did the coupling reaction with this kind of boronic acid, and we obtained the formation of the desired product. And now, next slide, the yield is much better, 85%. So now, in order to get the formation of this MS3000, we have to reduce this ketone in here that was done with tosyl hydrazide, and then by reduction, we get the formation of the methylene at this position, and after saponification, we got the formation of MS3000 with an overyield of 76%. Next slide, please. So in a very few steps, from a very cheap starting material, we obtain the formation of MS3000 via this bicyclic intermediate. So 8 steps, and now the yield is much better than for Merkle because the overyield now is superior, is up to 40%. Next slide, please. So, I mean, if you are looking at the drugs on the market, the most predominant substituent in the drugs is a benzene ring. But there is some problems with the benzene ring because you have problem of solubility, bioavailability, toxicity, planarity. Next slide, please. And I mean, if you are a medicinal chemist, has noticed that if they have the FSP3 that is increasing, they have a better chance to get the formation, to have the product on the market. So what is FSP3? Next slide, please. SFP3 is the number of CSP3 towards the total number of carbons. So what does this mean? Next slide. That we have to escape from flatland. So how can we escape from flatland? Next slide, please. Well, I mean, to replace a phenyl ring by a bioisoesters. And the bioisoesters of a phenyl ring are a cyclopropyl, a cyclobutyl, a bicyclopentyl, a cubyl, or an adamantyl. Next slide, please. So Sanofi came to us with 2 problems. That means they wanted to have, for example, molecules and to introduce a cyclopropyl group at the last stage of the synthesis of the compounds in order to get the formation of these cyclopropyl compounds. But they wanted to have a robust reaction, chemoselective, easy to handle, transposable to a process, and by using a very cheap catalyst. The second problem that they came up was about isoquinolone. I mean, they were really interested in this isoquinolone, and they have some leads because for their biological target, but they wanted to increase the bioactivity of this compound. And they say, we would like to replace this phenyl ring by a strain ring, such as, for example, this cyclobutyl. So next slide. So let's go back to the first problem. How can we synthesize these cyclopropyl compounds? Well, we can probably obtain this kind of product from a molecule which is functionalized, for example, by an halide in the presence of this cyclopropyl grignard. You can do this kind of transformation by using a palladium. But we try to avoid palladium and to replace this palladium by some transition metal that are not rare, so not expensive and not too toxic. Next slide, please. And the best condition that we found were the following. If you have, for example, an alkyl iodide in the presence of a grignard, cyclopropyl grignard, if you are treating these compounds by cobalt ACAC 3.5 mol% in the presence of TMADA and THF, you can get the formation of the coupling product. Next slide. So the reaction is very general. You can use different primary iodide. And for example, this primary iodide with a benzene group, you can introduce a cyclopropyl with a yield of 86%. The reaction is really chemoselective. If you have a iodide with an ester, you can get the compound with the ester still in place. For the cyano, that's the same. You can have also the formation of a cyclopropyl cyano compound with a yield of 81%. And even if you have a phenyl with a bromide, the reaction is also chemoselective. As you can see, the bromide is still in place, and you can do some other coupling reaction with this phenyl bromide, and the yield is around 78%. Now, I mean, the reaction is also working on secondary iodide. Next slide, please. For example, if you have a 4-iodopiperidine, you can introduce a cyclopropyl with a good yield. Even with a C3 iodide, you can have the formation of the cyclopropyl. Sometimes, I mean, if you are using other transition metal, you have an elimination. And in this case, the reaction goes very smoothly, and you can get the formation of the desired product with a yield of 85%, as well as for the 3-iodo-pyridine and also for the acetidine. You can see that the yield are also very good. Next slide, please. So, I mean, for the second problem, they were really interested, Sanofi was really interested by the synthesis of isoquinolone. And in fact, this compound has very nice biological activity. For example, for the first one, this compound with a phenyl group is an antitumoral agent, and it is a tyrosine 1 inhibitor. The other compound, the second one, is an antitumoral agent also, and it's an inhibitor of PAS. And for the third one, it's an immunosuppressive agent, which is also an inhibitor of tyrosine kinase. So, as I said previously, all these compounds are constituted by a phenyl and also an heterocycle, and they wanted to replace this phenyl by a strain ring, such as a cyclobutyl. Next slide, please. So we have to find a way to obtain this kind of compounds. So this cyclobutapyridone should be obtained from the cyclobutenyl hydroxamate in the presence of an acetylenic. What is the hypothesis? Next slide, please. Well, I mean, the hypothesis was the following. If you are treating the cyclobutenyl hydroxamate by a metal and a base, you will have probably a concerted metallation deprotonation that will lead to the formation of this 5 metal cycle in here. And then this intermediate could complex the acetylenic to lead to the formation of this intermediate, which after carbometallation will lead to the formation of this 7-membered metal cycle. And then after reductive elimination, cleavage of this NO bond in here, you will have the formation of this cyclobutapyridone, regeneration of the metal and the base. Next time. So, I mean, we try to do the CH activation. And at first, we choose to use ruthenium dichloride paracymane dimer in the presence of the acetylenic, in the presence of a base, and copper acetate, and see if we can get the formation of this kind of cyclized product. And in fact, next slide, the reaction was not working. Well, we tried to use another catalyst, such as the rhodium. Next slide, please. So we treated the cyclobutenyl hydroxamate by the ruthenium CP star Cl2 dimer in the presence of cesium acetate in different solvent, methanol, chloroform, DMF, DCA. We have 100% of the conversion of the starting material. Next slide. But we never observed the formation of this compound. So it became very frustrating. But here you can see that it's an intermolecular reaction. So maybe it's not favored. Next slide, please. So we have envisaged to synthesize this cyclobutenyl compound in here, bicyclic compound, from this cyclobutenyl hydroxamate, where you have the acetylenic, which belong to the cyclobutenyl hydroxamate, and see if we can get the formation of the cyclized product. Next slide, please. So We treated this cyclobutynyl hydroxamate by the rhodium CP star Cl2 dimer, cesium acetate in methanol, next slide, and we obtained the formation of the cyclized product. Next slide, please. The reaction is very general. You can have different group on the acetylenic, such as a phenyl. You can have a phenyl with a bromide. You can see that the reaction is really chemoselective. You can have also an aromatic with a methoxy group in the para position or in the ortho position. Here, for the compounds with the phenyl in the ortho position, you have to multiply the quantities of the catalyst and the base by 2. But still, the yield is pretty good. It's a moderate, but we can get the product 44%. Next slide, please. We can introduce an heteroaromatic, such as a pyridine, but the nitrogen has to be protected by chloride. Here also, we have to multiply the quantity of the catalyst and the base by 2, but still the reaction is working. You can introduce also a silyl group, a methyl, and the yield are pretty good. Next slide. And you can have also different substituent on the 4-member ring, such as, for example, a benzyl or an isopropyl, and different lengths for the tether, 3 to 1, 2, 3, and the yield are also still acceptable. So we make a library of 35 examples, and Sanofi has tested these compounds, and 2 of them were proved to be much superior to their compounds. So, I mean, it was an improvement about the bioactivity by introducing a strain ring. Next slide, please. Another way of obtaining, increasing the bioactivity of compounds is to synthesize macrocycles. And for myself, I think that macrocycles are an underexploited class of compound. You have only 2% of macrocycles on the market. Next slide, please. And in fact, if you are considering this compound in here, which is a farnesyl transferase inhibitor on the left-hand side with antitumoral activity, you can see that the EC50 is 5,000 nanomolar. But if you have the closed form on the right-hand side, the EC50 is only 0.1 nanomolar. Next slide, please. Another example, if you have this compound with a check kinase 1 inhibitor with chemosensitizing activity, the EC50 is around 4,000 nanomolar. And for this, the closed form, the EC50 is 10 nanomolar. So how can we increase the bioactivity of this isoquinolone? Next slide, please. Next slide, please. So probably the thing that we can do is to synthesize a macrocyclic isoquinolone of this type. How can we have the formation of this macrocyclic isoquinolone? Next slide, please. Well, probably by having this N-methoxy hydroxamic acid, which are on the right-hand side. And if you have the activation of this aromatic CH bond, probably maybe we can have also a cyclization. Next slide. So the hypothesis was that if we were treating this N-methoxy hydroxamic acid by a metal and a base, we will have probably a concerted metallation deprotonation that will lead to the formation of this metallocycle, which will complex the triplet bond intramolecularly to lead to the formation of this kind of intermediate. And then after carbometallation, we will have the formation of this 7-mandatory metallocycle. And then after reductive elimination, cleavage of this NO bond, we will have the formation of this macrocyclic isoquinolone, regeneration of the metal and the base. So we try to find the best condition in order to have this kind of transformation. And next slide. When the thing that we did is we treated this compound in here by the ruthenium paracymene dichloride dimer in the presence of a base with copper acetate in chloroform, and by very slow addition of this starting the compound in here, this acetylenic compound onto the catalyst, we can get the formation of the macrocyclic isoquinolone. So you can have, for example, a 12-membered ring with 2 oxygen in the macrocyclic, cycle. You can have even a 20-man bedroom. You can see that the yield is not bad, 78%. You can have an oxygen and a tosylamine, 71%. You can have also an ester in the macrocyclic derivative. And we can have, for example, a 14-man bedroom with a yield of 75%. We can have also the formation of this macrocyclic compound with an amide. The yield is only 35% because the starting material is not very soluble under this condition in chloroform. We can also scale up the reaction. You can see that you can run the reaction on 1.3 grams, and you can see that your yield in the 13-man bedroom in here is around 58%. Next slide, please. So we were also interested in the synthesis of danoprevir. Danoprevir is a highly selective and potent inhibitor of hepatitis C virus. And in fact, this compound was synthesized by Array Biopharma, and then it was licensed to Roche. You can see that you have a 5-man bedroom, 2 cyclopropyl groups in here, and you have also 1 stereogenic center at this position. And in fact, in order to get this part of the molecule with the mean protected by a buck and this carboxylic, this ketone, amide, I'm sorry, and double bond, they were synthesizing this compound, next slide, please, from this unsaturated amino ester. And the thing that they did, they do the hydrogenation of this compound by using this rhodium catalyst under very high pressure of hydrogen. And under this condition, they obtain the formation of this amino ester with a yield of 99% and an enantiomeric excess, which is very good, 99%. But as I say at the beginning, if you want, it will be good if you can avoid to use this very expensive transition metals, such as, for example, rhodium. And by working with Proteus, which is a company that belongs to Sequence in France, we show that, next slide, please, that if you are replacing this rhodium catalyst by using an N reductase and a buffer condition in the presence of this cofactor, NADH, we can get the formation of this amino ester with a yield of 99% and also a very good enantiomeric excess, 99%. You can see that this reaction is very chemoselective, as the double bond, the terminal double bond, is not reduced. There is only the double bond which has the ester and the amine that is reduced by the this N reductase. Next slide, please. We were also interested in the synthesis of capromyelin, which is represented in here. This compound is constituted by piperidine. You have an amide at this position and also a second amide at this position, an heterocycle. And in fact, this compound is an anti-aging compound, but now is repurposed and is used for the management of weight loss in cat and dogs. And the thing that is really important for the bioactivity of this compound is the control of the stereogenic center with this benzyl group at this position. How can we synthesize this molecule? Next slide, please. Well, probably from this keto ester in here. And if we are able to reduce this beta keto ester to this hydroxy ester by controlling the stereogenic center for the hydroxy, probably we can control the stereogenic center, which is the quaternary stereogenic center. So how can we do it? Next slide, please. Well, at first we use the S-binap ruthenium dichloride under also very high pressure of hydrogen. And we have to heat the reaction at 50 degrees. The yield in the expected product is 77%, and the enantiomeric excess is not really great. It's only 60%. So what can we do? I mean, we try a lot of chemical catalyst, optically active chiral catalyst, in order to do this kind of transformation. But each time, either the yield is good or the enantiomeric is good, but not both. So, I mean, we were really at one moment inspired by the work of Professor Yadav in India, And thanks to this work, we were solving the problem. Next slide, please. So we were using carrots. And if you are treating this beta-ketoester by carrots, you can get the formation of this hydroxyester with a very good yield, 91%, and the enantiomeric excess is 91%. So here also, I mean, the reaction was done in water for 48 hours at room temperature, and we know that the enzyme that is acting in this kind of transformation is Docus carota, so it's a ketoreductase. And then we can transform this hydroxyester to capromyelin. Next slide, please. So, I mean, I show you that we can replace, sometimes, it's not always the case, but if we can do it, we have to do it. We can replace the very rare, expensive, and also toxic metals by some very simple activation. For example, to get access to Ml3000, instead of using, for example, gold or silver or palladium, we are only using proton and heat. In the case of the Kaplan reaction to get cyclopropyl compounds, we could replace palladium by cobalt, which is very cheap salt. For doing the synthesis of capromyelin, instead of using ruthenium, we were using enzyme. For dolutegravir, instead of using rhodium, we can use enzyme. The only thing that we didn't succeed in the CH activation is to replace the rhodium and the ruthenium in order to do the CH activation and to have the cyclization in order to get the formation of this isoquinolone or the bioisosters of isoquinolone, such as for introducing this formanderdring or to have the macrocyclic compound. Next slide, please. So now I have to thank all my group for their work. I mean, particularly all this person in here, Damien Bellotti, Christophe Meyer, who is our director of CNRS, Thomas Sayed, Jean-Philippe Krieger, Amandine Guerino, Claire Andersen, Domingo Gomez-Pardo, and Romain Lacherez. Domingo and Romain were working with the carrots, and also all the collaborations that I have with companies, Dominique Le Suisse, Gino Ricci, Julien Boutet, and Pierre Gilles. And I would like also to thank the ESPCI, the CNRS, and the Minister of Research for the financial support, Sequence, Proteus, Janssen Pharmaceutica, and Sanofi also for their generous financial support. And I would like to thank you for your kind attention. Thank you so much.
Thank you. Thank you. The floor is open for questions.
I have a question related to ML3000. What made you to think pivalic acid should facilitate this cyclization?
Well, I mean, the pivalic acid, well, probably you have the activation of the triple bond by the acid, and then you have the attack on the amine on the acetylenic.
But doesn't it protonate the amine?
Well, I think you have an equilibrium. And it's why at one moment when you have the formation of the cyclic enamine, I mean, you have the equilibrium that is drawn toward the formation of the enamine.
Okay, thank you.
If there are no more questions, let us thank the speaker. Thank you, Dr. Kausi.
Thank you. Thank you. Sorry for the technical problem.
We are sorry about that too. We are closing your slide and deleting it.
Okay.
Thank you.
Next, we have Professor Lutz Eckermann from George August University, Göttingen, Germany, To introduce the speaker, I invite Dr. Nidhi Gaur.
Hello, everyone. It's with great pleasure I want to introduce Dr. Lewis Ackerman, who is full professor at Göttingen University, at George Augusta University, Göttingen. And he has pursued various leadership roles, including the role as Dean of Faculty of Chemistry and Director of Institute of Organic and Biomolecular Chemistry. His research focuses on development and application of novel concepts of sustainable catalysis and in particular emphasis on electrocatalysis and bond activation. In 2009, he founded and became director of Polar Research Institute of Sustainable Chemistry uh, and this showcases its full commitment for advancing the research. And thank you so much, Professor, for giving the talk here.
Yeah, thank you very much. Can you indeed hear me?
Can you share your slides?
Yes, I will share my slides. Yes, of course. So can you see the slides?
Yes, we can.
Wonderful. So thank you very much for the good afternoon, first of all, and thank you for the invitation. So let me just maybe give you first a short overview of what we're interested in. That includes electron and photocatalysis and asymmetric chemistry by machine learning. And today I will try to focus on some applications of these. And if you take an analysis of what has been used in where chemistry is used, it can be for food, it can be for health, it can be for materials. And one of the game changers was the introduction of cross-coupling reactions, as we've seen in the previous talk so nicely. For example, the Zucchi-Miura cross-coupling reactions is used a lot in production. And that is a change from 1984 to 2014. And however, you need to prepare the starting materials. You have waste products, and therefore we and others have been interested in using CH bonds as latent functional groups. You can reduce the number of reaction steps. So do one step instead of 4 or 5. We will see a few examples. And this can be enabling also in sustainable drug development, as the topic is today. Okay, so I would like to give you a short introduction, how we came into the game of CH activation, and then a few aspects include non-precious metals, asymmetric catalysis, and electron photochemistry, I guess. So at the outset, we became interested in how these reactions work and our different mechanisms. And in contrast to these well-precedented ones where you change the oxidation state of the metal, where you go from, for example, ruthenium 0 to ruthenium 2, like in the work of Lewis and Smith and Kokuchi, Shatani, and Murai, we identify a complex that was stable, a ruthenium 2 complex, this bis-carboxylate complex that is stable. It doesn't form a hydride, it doesn't need ruthenium 0. And therefore, it's tolerant of many things. And this is a summary slide. Many reactions, including arylations. You can make drugs. You can modify peptides. You can modify nucleosides. And many groups have contributed to this. This is not a complete list. This is not necessarily updated, but there was a worldwide interest. And also Professor Kossi has nicely shown some examples from her work using these ruthenium bis-carboxylates. Now, I think one remarkable feature that I would like to highlight is, first of all, the functional group tolerance, which you can see here, which is important for drug-type molecules. And another aspect that we studied was that you can change from the very typical ortho-selectivity, if you have any Lewis basic functional group, that you can go to the meta-selectivity. Now, all of this we have discovered over the last 15 years how you can change this, but let me show you some examples to biorelevant molecules. So along the studies on this peptide modification, we could show that you can make these oligopeptides featuring, of course, the peptidic backbone with many stereogenic centers. And some 6 years ago, we could show that indeed you can do this on SPPS, so solid phase peptide synthesis. And this slide sort of includes a number of challenges, one of which is that, of course, you have many functional groups. You have this NH backbone, which is acidic, which could kill many other catalysts. You have some functional groups, so you can have an arginine here finally in the product. But also, of course, this is on a cross-linked polystyrene, and therefore the swelling properties, diffusion, and so on are very important. So this was a proof of concept that indeed you can make oligopeptides by CH activation on solid support. Now, another challenge we wanted to address is, can we indeed use this for real drugs? And I guess the topic of this symposium is sustainable drug synthesis. And we have shown this. In particular, I would like to share with you a few examples with our colleagues from industry, where we use these ortho-functionalizations recently with Daniela and Stig, two postdocs, joint postdocs, in which we could show that you can do this ortho-amidation reactions. And this is all reactions that we have done with this ruthenium system that I've introduced you before in the in situ generated ruthenium bis carboxylate system. You can have all kind of difficult drug type molecules, acid handles. You can have here the cyclobutane, which is tolerated by the versatile ruthenium catalyst. Now, why is this important? Because it reduces the number of reaction steps that you would need to make these compounds de novo. So these are the numbers, the reaction steps shown here, calculated by our colleagues at AstraZeneca, if you were to make this molecule in the most modern and best way today. And for example, for the atazanavir, if you would need to make this molecule, you would need 14 steps, whereas the ruthenium catalyst can do this reaction in one step. You can introduce PROTAC motifs, as you can see here. So the PROTAC, the typical one, you can have streptavidine conjugate interactions. And all of these reactions you can do in one step. So you reduce the amount of reactions, you reduce the amount of undesired byproducts, and thereby in one step you can change many physical and biophysical properties of these new drug molecules. Now, one aspect that I emphasized before is that the ruthenium is the only metal that can change for an alkylation, for example, for CC bond formation in a robust fashion, to go from ortho to meta with the same substrate. So not introducing a special kind of template design auxiliary. And this is a long mechanistic investigation we did for the CC bond formation with stereogenic handles, computation by these Fukui indices or kinetics and EPR. Okay, and these reactions give the by ortho CH activation the meta product. Now, why exactly that is sort of exemplified here, but it's specific to ruthenium. We and others have not found any other metal that can do this job. You can do these reactions if you want also at room temperature, if you use photochemistry. This is shown here. But also here we were interested in, and I should mention that Mike Greeny also independently worked in this to make our ruthenium meta CC bond formation photochemically available. Now we wanted to go the next step, the transition and translation from bench to bed. And again, we teamed up to show here that we can do, and this time with Lucas from Strasbourg, a postdoc, a joint postdoc, we could do all these meta-alkylations. For example, here the cyclobutane can be introduced. We can do this on complex structures. And it's not limited to the cyclobutane, but again, now with a meta-selectivity on marketed drug compounds, real-life molecules, for example, with good yields, you can introduce, for example, this primary, tertiary alkyl group, cyclopropyl, cyclobutyl, and so on. And thereby, in one step again, we can change the physical chemical properties like solubility, log D for these molecules. Okay, this shows you the power of CH activation in real life molecules to do this in a sustainable fashion. Now, recently, as has been already indicated in the last talk, we became interested not only introducing, let's say, cyclopropyl and cyclobutyl, but maybe introducing these by incorporation of BCB, so a strange small molecule. And the design was based on the ruthenium meta-functionalization, but for the first time taking these BCB molecules for meta-functionalization. So this you can see here. You can make a number of compounds now that have directly the cyclobutane, including here in a 3-component reaction. You can do these reactions on gram scale, and you can diversify it to do sort of first a meta CH activation, and let's say a ruthenium or any other metal that you would like for sequential meta and ortho functionalization to increase the structural complexity and have some FSP3 rich molecules by remote CH activation. Now, we founded I was the founding director of the Wöhler Institute for Sustainable Chemistry, and we were particularly interested in replacing some of these metals that are shown here, not only for the cost of goods, not only because they are relatively rare, but also if you make a drug, you need to remove them down to 10 ppm level. So this is the data from GSK that shows you if you use rhodium, ruthenium, palladium, even silver oxidants, you should remove these metals for 10 to 15 ppm, not to bias the bioactivity of these compounds. So indeed, we have a long-standing program, and today I would like to show you a few select examples of using most of these metals. This is the price of the metal here from an earlier review from us. This is the price for iron per ton of the metal. So as you can see here, manganese and iron would be very desirable. There's no limit because it's anyway already part of the enzymatic procedure. And therefore, we showed that manganese 1 can indeed be used to do this linchpin alkylation reaction some time ago. You can do this. And these manganese 1 catalysts do not want to do any cross-coupling. So for example, such an aryl iodide is tolerated. You can tolerate an azide. And indeed, you can use this to make metabolically stable macrocyclic peptidic structures where you have this sort of as a linchpin. This is an X-ray structure of the CH-activated complex. So here we have the manganese. It's not magnesium, so it's manganese, and therefore this is a very tolerant to carbon. Yeah, everything okay? That was a back. We have a very stable manganese carbon bond, and indeed, this allowed us to do these reactions also in aqueous media. This is shown here. Here in this study with our colleague from the University Hospital, Professor Conradi, we made these cyclic peptides as anti-cancer agents. So this is the catalyst where you have sort of the manganese carbon bond here, making in aqueous media anti-cancer agents. And if you want, you can also use this manganese catalyst to make fluorogenic amino acids, which can be used as, and this is work together with from the University Hospital in Scotland, Professor Vendrell. We could stain these with this new fluorogenic tryptophan derivative. Now also the 3D metals can be used to change bioactivities in one step, and that was actually one earlier work in which we introduced directly a methyl group, as you can see here, now with a cobalt catalyst. Now these are orthoselective reactions, not like the ruthenium meta-selective. This is based, all of what I've shown you here today in this collaborative effort is based on high throughput engineering. These are the plates you can see. And again, in these colors, you see number of reaction steps. So for example, here 12 steps to make this methylated analog. Atazanavir in this case would require 9 reaction steps. And here it can be done in just one step to change the log D, for example, of this drug molecule. I think this nicely illustrates the power of late-stage diversification for drug molecules, not only using ruthenium, but also cobalt catalyst. I thought I share with you a few other aspects of our research line here, and particularly how we can use data science and machine learning for asymmetric CH activation. This was a field that was dominated, no matter if you make a stereogenic center, chiral axis, planar chirality, or helical chirality, dominated by rhodium and palladium catalysts. So these are beautiful examples from Tom Rovis and Tom Ward and the Haltermann type complexes that have been used some time ago, as you can see here. But we were wondering, can we indeed develop stable cobalt-3 or for the first time even iron catalysts to do asymmetric CH activation? Now, key to success was actually the original design of these kind of chiral acids. As you can see here, 8 years ago, we reported indeed chiral amino acids, which groups like Matsunaga and others have subsequently used. But this chiral acid here gave a significantly higher enantiomer induction, interestingly. And I was a bit, ignorant maybe. I thought almost all chiral entities have been used at one point in transition metal catalysis, but this was a compound that had not been used before in transition metal catalysis, but enabled to use a stable cobalt-3 complex for an asymmetric CH activation. I want to rather give you, so this was actually a research we started within a program on using weak dispersive interactions. Here we teamed up, among others, with Stefan Grimme to identify, quantify, and harness weak dispersive interactions. And recently we have combined this with an approach where we acquire data both for multivariate linear regression models, for quantitative linear structure activity or enhancer selectivity prediction models, and ultimately to use machine learning. And this we have achieved in this paper from 2 years ago, where we tested different, like random forest, different models. So here we have identified actually for this cobalt catalysis some new chiral acids that were not known before, to give actually trained by data on high EE for this enantiomer selective reaction, to give a highly diastereoselective reaction. So enantiomer selective here and further a stereogenic axis here, so chiral axis. So 2 stereogenic elements being viable. This is part of a program which we have used in different directions. Also here some recent contribution, how we could actually not only optimize enantiomer selectivity, but also optimize chemical yields in this regard. Now, this is so some recent trend were based on insight by DFT. We try to develop enantiomer selective reactions through weak secondary interactions, and now to use this combined with data science, so AGI informed machine learning. And this here is actually was a first time where we could apply this in this case for electrochemistry, and that leads me sort of to this part where I want to talk a little bit about how we can use earth abundant metals to do electrochemistry. And conceptually, what we showed here as a proof of concept is that we can use either solar or wind energy, and for dehydrogenative reactions do CH activation at room temperature. Whereas if you take a look at the catalytic cycle, which can be very complicated, and if you're interested in this, we can discuss this later. When the catalytic cycle uses protons and electrons to do CH oxygenation reactions, with the only byproduct being molecular hydrogen. Now, this would be a talk on its own right just about electrochemical transformations. If you're interested in this, I would be very happy to discuss this. So we did CO bond formations, CN bond formations, CC bond formations. If you're interested in the setup, we compared the homemade systems with commercially available ones. But in the context of today's talk, where it's more about bioactive components, I thought I share with you one example in which we now could merge this field of electrochemistry with asymmetric catalysis. Now, let me guide you through this here. So we build up here a stereogenic center. We have here a relatively simple cobalt pre-catalyst that is combined with a chiral acid, and in this case, interestingly, this unsubstituted bienol phosphoric acid diester was found to be more enantioselective than any of these Akiyama Terada or Nankai type chiral acids. That was interesting to us. Second, maybe just to guide you a little bit through, we have here a high surface area carbon-based anode material, and we have a platinum cathode to control the overpotential for the hydrogen evolution reaction. We can quantify, also qualitatively by headspace analysis, and quantify the amount of hydrogen is formed. And typically these reactions have a Faradaic efficiency for the hydrogen formation of around 70 to 80%. This proved to be broadly applicable, and we reported here 6 enantioselective cobalt catalyzed electrochemical transformations. You can build up a stereogenic center. You can build up 2 stereogenic centers with high EE and high DR. You can have atropo selectivity to build up this chiral NC axis, and you can do desymmetrization of these meso compounds, these phosphoramidites. And from a conceptual level now, all of these reactions make hydrogen. I could give you some more examples where this could be even more interesting. But this hydrogen you form here would be available either as a energy storage material, or you could utilize it for paired electrolysis for further hydrogenation reactions. Now, in contrast to this aspect, I would like to emphasize that the cobalt shows here complementary selectivity to nickel. So if we do this reaction, this kind of approach now with a chiral Salox, or based on the Salox ligand from Carsten Bollm, we developed some new ligands, again based on informed machine learning, identifying descriptors for these ligands, feature analysis. We can come up with a model with 80-20 splitting that allows us to design some new chiral ligands. And if we do so for nickel, now with these oxanobornine derivatives, you can do the annulation to the 6-membered lactam, whereas indeed the cobalt catalyst gives you the 5-membered derivative. Now we can understand this in greater detail. We did both some data science as well as some computation. And if you do computation, again, this long-standing interest in weak secondary interactions shows you that there's a non-covalent interaction both between the substrate and the ligand. So therefore, the ligand design is very important, as well as with this aromatic group, the additional methyl group exerts CH pi interaction to stabilize this diastereomeric transition state model. Now, this is broadly applicable, but from a conceptual level, this gives you a complementary selectivity, and this is actually 2025. I'm sorry, so this is a very recent paper that just appeared on the web. You can see here we have the absolute configuration. You tolerate the number of functional groups, and all of these reactions typically proceed with high enantioselectivity. So this was a first example where you can now use nickel for asymmetric electrocatalysis. In the remaining few minutes, I would just like to highlight that we're also interested in photocatalysis. Actually, I did my diploma thesis a long time ago in oxidative photo-induced electron transfer. And again, I showed the slide before, we are interested in earth-abundant metals, but I think the king of the metal is somewhat still iron. And I give you just a talk in its own right, how we have used iron in many different variations. Yeah, this is a very exciting research area because not only can we do here, for instance, cyclovoltammetry, detailed kinetic analysis, NMR and EPR spectroscopy, but also MIRS power, of course, becomes an increasingly important role here. So we have a long-standing interest in using iron, not for cross-coupling, but for CH activation reactions. And so iron is dirt cheap, if you want. It is, there's no trace metal impurity regulations. This is some older work starting from more than a decade ago, where we could show that we can have here peptides that enable sp3 functionalizations. I should also mention that other people are interested in work in using iron catalyst. sp3 arylations, methylations prove to be viable. We can also do a diester selective modification of olefins. But so let me pick out a few examples of this research program. We can do allylations, arylations, alkynylations, alkyne annulations. Maybe here this is still interesting, but it's just exemplifying the challenge. So you can do these arylations on non-aromatic CH bonds, okay? But in all of these systems, actually needed some, and this was a little bit overlooked, DCIB. And the DCIB is dichloroisobutane, and it's actually relatively expensive. I was myself a little bit surprised by it, but it has basically almost the price of palladium. So there's an analysis we did, this DCIB, that has USD dollars per mole, almost the price of palladium acetate. And that was a little bit neglected by the community, because if you replace now palladium by iron, but you still have this costly oxidant, that is actually an issue. And we wanted to address this, and actually we did so by electrochemistry. Another issue we wanted to address is actually the fact, is it possible to use iron for asymmetric CH activation? I've shown you this field was dominated by this beautiful rhodium complexes, palladium. And we were able to contribute some stable CP star cobalt complexes. But iron, so far, the only example that proved to be viable is the one that I've shown you here. Despite of this beautiful work on rhodium, palladium chemistry. And I think conceptually, this was really illuminating for us. So this is a reaction, you have an iron catalyst, similar to the conditions I've shown you before, but you need an NHT. No other ligand basically gave really high isoselectivity. But what people typically would do is modify proximal to the metal. So the metal would coordinate here. And we wondered, whether it would be possible to have weak dispersive interactions in the chiral pocket in a remote position. And this is what you see here. So if you increase the size of this R group, the yield is going up, okay, but the E, from lousy, not useful, you go up to the first time to high enantioselective organometallic CH activation. And this was based, we did some early computation. We had a transition state model that was also based on kinetic experiments, where we could show that the reaction is first order in indole, second order in styrene. You can have more examples if you want to. Of course, organic chemists like to have more examples. But recently we could improve the system further. So with this remote functionalization, I should point out this was done by two excellent PhDs, Joachim and Daniel, who were in this priority program on weak dispersive interactions. And based on this work, so Joachim came actually from EPFL in Lausanne to us, we could now team up and show that with this same type of ligand, again, remote, if you want dispersion energy donor groups, adamantine groups, you can set up this stereogenic center and at the same time with an iron catalyst have an axial chiral NC, so CN axial chirality that is stable here. So here we did also some DFT computation now on an advanced level with Shinran. We did also some Milchbauer studies. And now we could indeed again identify pi-pi interactions as well as CH-pi interactions that stabilize this discrimination. There's sort of selective discrimination. So here you can see you have EE in the 90s, also here across the board, and high DR, so high enhancer selectivity and diester selectivity. Now with iron to make not only one but two stereogenic centers. Now, this was an effort towards enhancer selective iron catalyzed CH activation, and last but not least, not only there are often some chemical oxidants, but or these Grignard reagents. Grignard reagents are okayish, but often they compromise the functional group compatibility, and this we could actually address last year by doing these reactions with light. So this is now photochemical transformations where we have a stable iron complex that can do this kind of olefination reaction. And the key feature is that now you can do these reactions at room temperature with light. And as you can see here, you can tolerate functional group such as a nitrile or an ester in any remote position. And if you compare this with the previous Grignard system, you go from lousy yields or no yields to high chemoselectivity. Now, with this being said, it's a lot of information, but let me be clear. It's everything I talked about today is only one reaction, and it's CH activation. We use different metals to address some aspects. So orthoselectivity, we could indeed get meta, also power selectivity using ruthenium chemistries. We don't want to use precious metals. I showed you some example where we could use manganese, cobalt, nickel, or iron to do CH activation with earth abundant metals, but all is CH activation reaction. Full selectivity control, so namely enhancer selectivity, was largely limited to palladium and CP rhodium systems. And indeed, we could show that as a proof of concept, CP rhodium could be replaced by CP cobalt systems, but also that we are able to do enhancer selective CH activation for the first time with organometallic iron catalyst. And particularly today, I wanted to focus a little bit on applications. So the late stage diversification on real life drug molecules could be possible, both on scale, but also for diversification of bioactive compounds. And I've showed you a few case studies in which we used ruthenium and cobalt catalyst, also manganese, to make cyclic peptides. But basically where you can build up this structural complexity in one step instead of up to 14 or 15 steps, as it was shown on the slides. Last but not least, I hope we could contribute also with electron photochemistry to utilize renewable forms of energy. That we gained some new mechanistic insights, and hopefully I could present you with some novel reactivity and selectivity manifolds. With this being said, I'm just the ambassador of the group. I have the pleasure of talking about these things worldwide and present or virtually. I'm really grateful to all the co-authors, co-workers for their great experimental contribution, and particularly their intellectual input to all of these projects. This is a picture, I think, from last year. I'm grateful for our collaboration partners, a few of them I mentioned also Mike Neidig and Joanna for the iron chemistry recently. I'm grateful for our collaboration partners, both also in industry. I talked about some projects here with AstraZeneca. I'm very grateful for the European Union for some funding within the ERC scheme. And last but not least, I think in the last part, I wanted to emphasize I'm also grateful for the Deutsche Forschungsgemeinschaft, so the funding agency in Germany, because they allowed us to really dive into understanding and harnessing weak dispersive interactions, particularly for asymmetric catalysis. And recently we get funding to do this also utilizing data science. So with this being said, thank you again very much for the kind invitation to speak here. Thank you for listening. And if there are any questions, I would be happy to entertain them.
Thank you so much, Professor, for a very nice and engaging talk. So if there are any questions, yes.
Hi, Professor Ackerman. Thank you so much for a nice talk. So I was wondering if you have noticed the chiral induction, any dependence you have noticed on the particular metal, that some metals are better at chiral induction?
Yeah, very good question. There's some, the audio. I mean, the metals are complementary to one another, and I think for us, best case is actually here. If you use here the nickel catalyst, indeed you get this 6-membered lactane. If you take under otherwise identical reaction conditions a cobalt catalyst, you get the 5-membered system, both with high E. I think each metal sort of has its own, of course, right, and they are not easily, exchangeable, let's say.
Thank you.
But we did also some work on asymmetric ruthenium catalyzed CH activation, but I think, conceptually, I think these 3D metals are very exciting to us. Yeah, thank you for the question.
So is there any more questions? So if there are no questions, I heartily thank Professor for accepting the invitation and giving such a useful talk on bond activation. Thank you so much.
Thank you very much. My pleasure.
Our final speaker for this session is Professor Varinder Agarwal from the University of Bristol, UK. To introduce the speaker, I now invite Dr. Poulomi Sen-Gupta.
I feel deeply honored and privileged to introduce Professor Varinder Agarwal, the Alfred Kapper Pass Professor, Department of Chemistry, University of Bristol, UK. Professor Agarwal is a distinguished organic chemist renowned for the pioneering work in asymmetric synthesis. He earned his PhD under Stuart Warren at the University of Cambridge in 1986 with a thesis on stereo-controlled synthesis with phenylthiomigration. Professor Varinder Agarwal has held several prestigious editorial and advisory roles. He was a member of the International Committee of Organic Chemistry of Sulfur and the International Committee of Heteroatom Chemistry. He served on the editorial board of Organic and Biomolecular Chemistry and the International Advisory Board of Chemical Communications. Additionally, he has been on the International Advisory Board of Chemistry and Asian Journal since 2006. The editorial board topics in stereochemistry and the advisory board of Asian Journal of Organic Chemistry. With this, we welcome him for his talk, Synthesis with Boron at the Helm. Welcome, sir.
Good morning, or good afternoon, I should say. First of all, can you hear me okay?
Yes, we can hear you.
Perfect. Thank you very much. Okay. Well, It's a pleasure and an honor to be with you, even if it is remotely. I've recently been in India and had a very pleasant experience in various places in Bhopal and in Kanpur. And it's a country that's very dear to my heart for obvious reasons. Now, I'm going to tell you about some chemistry that we're involved in, and it involves chemical synthesis. And in chemical synthesis, you start with a starting material, you add a building block to it, you carry out functional group interconversions before you add further building blocks to reach your target molecule. And we, like many other people around the world, are interested in how to go from starting material into final product more effectively and more efficiently. And our idea is to try and engineer methods that allow this complex series of transformations to be conducted in essentially a one-pot operation. And the way we think about doing this is to exploit some of the beautiful and fundamental chemistry of boron. Boronic esters, having a vacant p orbital on boron, are electrophilic, and so they react with nucleophiles to give boron 8 complexes. Now, if that nucleophile, depicted here as an organometallic, has a leaving group attached at the same carbon, then the intermediate boron 8 complex can undergo 1,2 migration, expelling the leaving group to give a new boronic ester. And that new boronic ester can be used again and again in an iterative fashion until the final target molecule is created. This chemistry has some similarity with what happens in nature. Nature takes these thiol esters and carries out numerous acylation reactions, reductions, eliminations, catalyzed by the beautiful polyketide family of enzymes, creating this vast array of natural products. So, iterative chemistry is not just the remit of nature, we can envisage it as well in the lab. Now, in order to realize this overall process, we need to address 3 important issues. We need ways of introducing boron into the molecule to make our starting material. We then have the growth phase where we carry out this iterative chemistry. And then we have a termination phase where we convert the carbon-boron bond into what we really want at the end, because it's unlikely that boron is the final target molecule. And fortunately, there are lots of transformations that you can do with organoboron compounds to other functional groups. And it is interesting that actually each of these 3 areas are important areas in their own right in organic chemistry. But you'll see the power of this chemistry when all 3 are combined together in a natural product synthesis. And I'll show you a couple of examples as we go through. Let me begin by focusing on the growth phase, the reaction shown here. Now, in order to do that, we need to develop a nucleophilic reagent that's got a leaving group attached. It's chiral, it's got to be configurationally stable, and it should be available in both enantiomeric form. Quite a tall order for a single reagent. But we were attracted by the chemistry from Dieter Hopper, who showed that carbamates could be deprotonated with strong base in the presence of a chiral diamine ligand and trapped by electrophiles in high enantioselectivity. The intermediate here, the lithiated carbamate, looked to us like a chiral organometallic with a potential leaving group attached. So could we use Hopper's lithiated carbamates with boronic esters for asymmetric homologation of boronic esters? That is the question we asked ourselves over 10 years ago, and this turned out to be correct. Here's the reaction again, and I'm going to first of all show you the scope of the chemistry with respect to the carbamate that can be employed. The R group can be a simple methyl group, small group, high yield, high in enantioselectivity, but it can go all the way up to T-butyl, high yield, high in enantioselectivity, although in this case we resorted to using, instead of the carbamate, this alternative reagent, the triisopropyl benzoate ester. Now, the chemistry tolerates some functional groups, but of course it's limited by the strong base conditions employed. So it'll tolerate silyl ethers and alkenes. But in the substrate scope in terms of the boronic ester, on the other hand, is much broader. Not just alkenes and silyl ethers, but now you can also bring in esters, azides and nitriles. And this shows that the boronic ester is more electrophilic than these other potential electrophiles. So it tells us a little bit about reactivity as well. Now, when we have methodology, we like to apply it in synthesis. And we chose as a target molecule baulamycin A. And we chose this for a number of reasons. It had just recently been isolated in the labs of David Sherman, and it had very interesting biological activity. It's an antibacterial agent in which it inhibits the bacterial biosynthesis of sidophores. Sidophores are molecules that bring iron into the cell. Bacteria don't have other mechanisms for metabolizing iron, but it needs it, so it generates these sidophores to bring iron in. We don't employ such biochemical pathways, so by inhibiting this pathway, it would be unique to the microorganism. Furthermore, there were no published syntheses of this molecule at the time, and so we embarked on its synthesis. And we envisaged a disconnection across this carbon-carbon bond that breaks the molecule into two roughly equal fragments, fragment A and fragment B, and we envisaged their union through this lithiation-borylation reaction to not just make this carbon-carbon bond, but also control the stereochemistry of the alcohol that's generated. Fragment A contains two boronic esters, which can be made through diboration of the alkene, which itself, being a homoallylic alcohol, can be made through a simple alloboration reaction. Fragment B, I've masked the ketone as an enol ether because that enables us to disconnect it across here and go back to this boronic ester, because boronic esters can be converted into alkenes using Zweifel olefination. And I'll tell you about that reaction in a minute. This boronic ester can be made through our assembly line synthesis using these building blocks. So now let's have a look at this in the forward direction. But before we start, I just want to make one further change, or to highlight one further change, that instead of using the carbamates, we're now using the triisopropyl benzoates more routinely. And that's because these are slightly better leaving groups. And as molecules become more hindered, you want to have a faster 1,2 migration to stop the boronate complex simply going back to its constituent parts. Furthermore, the corresponding stananes of these building blocks are crystalline. That allows us to have enantiomerically pure blue and red reagents. And it means that when we use them iteratively, we don't generate diastereomers. So with that preamble, fasten your seatbelts, as I say, to enable us to launch our assembly line synthesis. Because we first of all come in with the blue reagent, and then we come in with the green chloromethyl lithium reagent, then the enantiomeric red reagent, chloromethyl lithium again, and finally the blue reagent is added. After 5 sequence homologations, we do our first purification, and this boronic ester was obtained in 64% yield as a single diastereomer and single enantiomer. We then need to we then need to do our Zweifel olefination. So boron has done its job of enabling that iterative chemistry to take place. We then carry out the Zweifel olefination, and the chemistry is shown here. The reaction of the lithiated enol ether with the boronic ester gives an 8 complex, which on treatment with iodine forms an iodonium ion that then triggers 1,2 migration to give this beta iodo boronic ester, which on treatment with methoxide attacks boron and affects an elimination across here to give the alkene. That completes fragment B. Fragment B was then deprotonated with secondary butyl lithium in presence of spartine and combined with fragment A. Fragment A contains 2 boronic esters, but this chemistry is exquisitely selective for the less hindered primary boronic ester over the secondary boronic ester, and that's partly a consequence of the highly hindered nature of this lithiated tib ester. So it reacts at the primary boronic ester, and then after oxidation, we form the alcohol, as you can see, in high yield and high diastereoselectivity. From here to the end, we simply have to remove the mom ethers, phthalal ether, and enol ether, all of which are done using aqueous HCl. And that completed a 10-step synthesis of borlomycin A with full stereo control, both relative and absolute. However, we discovered a problem, because when we compared the NMR of what we made with the natural product, it didn't match. Now, we were confident about the stereochemistry that we had installed, and so we assumed that there was a mistake in the natural product structure determination paper. But which of the 7 stereogenic centers shown here had been misassigned? Well, that story turned out to be even more interesting than the synthesis, because in the end, it wasn't one of the 7 stereogenic centers that had been misassigned. Rather, 5 of the 7 stereogenic centers had been misassigned. And what I want to do now is show you how we arrived at the correct structure of borlomycin A. Here's the proposed structure of borlomycin A. And like the synthesis, you can break the molecule down into the same 2 fragments for structure determination. These 2 fragments are far enough apart that they barely speak to each other. So let's first of all focus on fragment A. When we compared the NMR at H1 prime, we noticed that the coupling constant in our synthetic material was 3.5 Hertz, whereas in the natural product it was 7 Hertz. That immediately told us that the stereochemistry here was almost certainly incorrect, and rather than being syn, it should be anti. Of course, we didn't just focus on this particular example. We looked in the literature at related examples, and there was a general pattern that emerged. For fragment A, then, there are 4 possible diastereoisomers. And what we did in collaboration with Craig Butts was we calculated what the NMR should look like of each of these 4 diastereomers and compared them with the natural product. That's quite a big job because the molecule is flexible. You have to compute the different conformations and Boltzmann weight their populations. But we did that, and we compared it with the natural product, and we looked at proton-proton couplings as well as NOE distances. And in orange here, I've shown you the proposed structure for the natural product, where fragment A has syn-syn-syn stereochemistry. What we're doing here is we're comparing with the natural product, and when the chi-squared reduced value goes to 1, it means you have an identical structure with a natural product. So you're looking for numbers that are close to 1. And from this, you can see that actually, if you focus on proton-proton couplings, anti-anti-syn is very good, but so is syn-syn-syn, which was proposed. But when you include NOEs, then anti-anti-syn wins hands down. So this was our predicted structure for fragment A. Anti-anti-syn was made through a phosphoric acid catalyzed elaboration reaction, chemistry developed by John Antilla. This worked extremely well, high yield, high diastereo, and enantioselectivity. selectivity. We then carried out a Morkan hydroxy-directed diboration reaction using simply cesium carbonate and B2-pin2, and again, that worked very well indeed. That completed fragment A. Fragment A was then coupled with fragment B and deprotected as before. But once again, when we compared the NMR with what we made with a natural product, it didn't match. The good news, however, was that this area here matched almost perfectly. This area here did not. And so we had to consider the stereochemistry of this polymethylated region. Now, we did consider doing the computational analysis as before, but tell you the truth, that was hard work. And we wondered whether there was a quicker way of arriving at the structure of this region, a way that exploited our stereo-selective, stereo-specific homologation chemistry. Let me show you what we did. Here's the homologation reaction that we have employed, and then we come in with the green reagent, chloromethyl lithium. We then used a mixture of blue and red reagents, 72 to 28, that gave us a 72-28 mixture of these diastereomers. That means that when we integrate the NMR and we see 72, we know that we've got the syn isomer. So NMR integration is telling us about stereochemistry. Homologation once more, and then finally we come in with a further mixture of blue and red reagents to now give us a mixture of 4 diastereomers. And the numbers in parentheses in black are the numbers that we calculated we should get if there are no matched and mismatched effects, and the numbers in red are what we measure, which show that there are no matched and mismatched effects in operation. And by the way, these numbers are not chosen at random. These numbers are chosen to maximize the difference between the 4 diastereomers and to ensure that we can see the smallest one by NMR. We wanted to have that at least 10%. These are the 4 possible diastereomers for fragment B. Fragment B was coupled with fragment A and deprotected, and now one of these compounds should be baulamycin A. Let's hone in on carbon-21. Here are the 4 peaks for carbon-21, and when we compare the NMR of this mixture with the natural product, we found that the tallest one matched the natural product. And so we came back with pure syn isomer and fragment A, combined them together, and now all the data matches with the literature That's the proton, carbon NMR, alpha D, and the rest of the data. And here are the 5 stereogenic centers that had been misassigned in the original structure determination paper. Now, I'm often asked, what is the limit of these homologations that you can do? Well, I don't know the answer to that, but I do want to show you what we have done. We've applied this chemistry to the synthesis of hydroxythioceranic acid. Hydroxythioceranic acid is the component of the cell wall of the lipid coat of the TB bacterium. And there is interest in developing vaccines against the TB bacterium, which would need this lipid molecule. So we have considered its synthesis using our assembly line synthesis. And once again, I've color-coded the carbons so that you can see where they are in our starting materials and where they end up in the product. So this pink carbon here in this aryl boronic ester ends up as carboxylic acid. So once again, we launch our assembly line synthesis where we come in with these reagents. These reagents grow the carbon chain 1 carbon at a time, sometimes just controlling the chain length with a CH2, but otherwise controlling the stereochemistry as well. After 15 sequenced homologations, we arrive here. We then need to introduce this long alkyl chain, and that's done once again using our homologation chemistry. So we have now done 16 sequenced homologations. And from here to the end, we have to protect, oxidize, and deprotect, completing a short synthesis, once again with full stereo control of this complex molecule. It's hard to do a step count, so that's why I've indicated X steps, but 13% overall yield is a significant achievement. This chemistry of introducing methyl groups along the carbon chain enables us to address some fundamental questions about nature. Why does nature do it? What is the role of these methyl groups? Nature could, after all, equally well do all of its chemistry using acetate metabolism, but it doesn't. It chooses propionate metabolism. Why? Because when you look at natural products, they are full of carbon chains containing methyl substituents. Well, in some nice review articles from Reinhard Hoffmann, he suggested the role of the methyls is to control conformation, because these methyls, when they're 1,3 related on a carbon chain, want to avoid this destabilizing syn-pentane interaction. And that destabilizing syn-pentane interaction is worth round about 3.5 kcals per mole. And this is an interesting example from the labs of Dale Boger. This is an anti-cancer compound that contains a region here that recognizes DNA and a warhead, an alkylating agent, a potent alkylating agent that destroys it. These two are connected together by a linker, but that linker turned out to be important as well. It is not innocuous. When he removed one of the methyl groups from this linker, the activity went down almost 50-fold. And that's because the conformation of the recognition arm with the warhead is no longer well controlled. If you remove one of these methyl groups, then both conformations become equally accessible. So we asked the question whether we could use methyl groups on their own to control the conformation of molecules. Because of course in nature, there are many other functional groups that are present. Well, our analysis is that if we could generate this alternating syn-anti-isomer, it ought to adopt this linear conformation, because the methyls want to avoid this syn-pentane interaction. Furthermore, if we could generate the all-syn isomer, this should adopt a helical conformation, because again, the methyls want to avoid this destabilizing syn-pentane interaction. And we have unique methodology that enables us to put molecules like this together. So let's first of all have a look at the alternating syn-anti isomer. That is assembled by bringing in pairs of red reagents and then pairs of blue reagents. These reagents are brought in one at a time, each time controlling chain length, each time controlling stereochemistry. This molecule was beautifully linear. Here's the X-ray structure of the molecule with a linear backbone with methyl H, methyl H, methyl running across it. It's easier to see in this particular way, but if you actually make a space-filling model of this, you see how little space there is for a methyl substituent 1,3. And you can see why there's a destabilizing interaction of 3.5 kcals per mole. So the alternating syn-anti adopts a linear conformation. What about the all-syn? Well, to make the all-syn, we come in with red reagent and then a blue reagent, alternating each time. Each time they're added, they control chain length, and each time they control stereochemistry. Now, this molecule was not crystalline, but it contains a boronic ester that can be easily oxidized and derivatized, and the paranitrobenzoate shown here was crystalline. And here's its X-ray structure, and I've highlighted in green the backbone. And now you can see the backbone winding its way around in a beautiful helical conformation. So these methyl groups really are acting like levers, pushing and pulling the carbon chain to avoid this destabilizing syn-pentane interaction. Now, I should say that the main challenge in this chemistry is not controlling stereochemistry, it's controlling chain length. Because if you imagine doing 9 sequenced homologations, the potential for getting a mess out of this is really high, because it's going to be impossible to separate under and over homologations if they're formed in any significant amount. But when we analyze our mixture really carefully, we hardly see more than 2% over and under homologation. That means that each step is going with greater than 99% fidelity. So why does this chemistry work as well as it does? Well, here's the reaction that we're doing. And the way we do it is we add an excess of the lithiated tib ester to the boronic ester to ensure all of the starting material is converted into the boronate complex. Of course, that leaves behind some excess reagent. That is done at low temperature, and then we increase to a moderate temperature, -40, at which point the lithiated tib ester begins to decompose. The boronate complex is still sitting here. It requires a much higher temperature before it undergoes homologation. That means that the homologated compound is never generated in the presence of excess reagent. That's all gone through self-destruction. And we don't see under-homologation by using an excess of the reagent to ensure all of the starting material is converted into the boronate complex. So in this way, we're able to do single homologation with very high fidelity. And we've likened this chemistry to a molecular assembly line. And in a molecular assembly line, you take a simple starting material, you add a blue reagent and then a red reagent and a blue reagent. And these reagents control the stereochemistry, and the stereochemistry controls the overall shape of the molecule. And now here in Bristol, we're trying to realize this animation in reality. We are generating or creating a robot to automate our homologation process so that it can be conducted in an autonomous manner without human intervention. And that will enable a broader set of molecules to be made and could assist in structure-activity relationships that are, of course, important in drug discovery. So that is the story I wanted to share with you, story of lithiation borylation, chemistry that we've likened to a molecular assembly line. And although I've only shown you how we generate these secondary boronic esters from primary carbamates, the chemistry can also be extended to secondary alcohols and the carbamates thereof to make tertiary boronic esters. And I have to say, this is one of the best ways of making these tertiary boronic esters with exceptionally high enantioselectivity. selectivity. And the tertiary boronic ester, of course, can be converted into a host of functional groups. And we're also developing cross-coupling reactions as well. So let me close by acknowledging our support. We've had good support from our national funding agency, EPSRC, and the European Research Council. I especially want to thank very strong collaboration that we've had, a long and deep collaboration with Craig Butts on NMR. But the people I really want to thank are the ones who've done the chemistry. They are a terrific group to represent, and I really want to thank them for their tremendous contributions, not just practically, but also intellectually in the development of our research program. So with that, thank you very much for the invitation to participate in today's meeting, and thank you very much for your kind attention. Thank you.
Thank you, sir. The session is open for questions. Since there are no more questions, we will close this session. On the organizers' behalf, I want to invite you physically in Inaugural University whenever you visit India. Thank you.
I'd love to. Thank you very much.
Thank you so much. With this, we conclude session 3. A heartfelt thank you to our esteemed speakers and session chairs for an exceptional session. We will now take a short break for tea and networking before reconvening for session 4, featuring industrial innovations from Cadila Pharmaceuticals. There will be a short tea break for 10 minutes.
Hello, hello.
Welcome back, everyone. We'll now begin session 4, a special session dedicated to industrial innovations in pharmaceutical research in collaboration with Cadila Pharmaceuticals. This research will highlight transformative advancements in drug development from an industrial perspective. We are honored to have Dr. Debendra Mohapatra from IISER Brahmpur and Dr. Vijay Singh from Indrajil University as our session chairs. Our speaker for this session is Dr. Bakulesh Khamar, Executive Director R&D at Cadila Pharmaceuticals Limited. I now invite Dr. Vijay Singh to please introduce our speaker.
Good afternoon, all of you. So, the Dr. Bakulesh Khamar served as Executive Director, Research and Development at Cadila Pharmaceuticals since 1996. He earned his MBBS in 1975 and Master of Surgery in Ophthalmology in 1980 from BJ Medical College, Ahmedabad. His research focused on the cataract formation leading to the development of the medical device. This innovation earned his Kol Rangchari Award from the All India Ophthalmic Society in the recognition of his significant contributions to the medical research innovation product development in India. Dr. Khamar has honored Swadesh Samman Award at the Swadesh Conclave in 2022. This award was presented by Dr. Jitendra, Union Minister of Science and Technology, Ashwini Kumar Chaubey, Union Ministry of Consumer Affairs, Food and the Public Distribution. With this, let's welcome Dr. Bakulesh Khamar, sir. Sir, please. Thank you. How do I change the slides?
Okay. Good afternoon, everybody. And thank you for inviting me. I'm an odd man out amongst the scientists of repute. So, as a coordinator for research and development at Cadila, I oversee whatever product development processes are there. And in next couple of minutes, I'll take you through what we have done for successful outcomes. Okay. So everybody knows that knowledge is a facts information which we study, we imbibe through journals, books, etc., etc. Okay. While innovation is something which has not previously been existence, and it's a process by creating value, by applying whatever we know to create novel solutions for a meaningful problems. And this is particularly true in pharmaceutical industry because solution has to have a problem which is meaningful for the mankind. Okay. So when you look at a patents, because it's a buzzword now, last year India filed 100,000 patents, so it's increasing trend. So innovation, I mean, if you compare the two, there are some parallels. Something which has not been previously in the existence, and that justifies novel. But on the top of novel, it has to be non-obvious, and that differentiates innovation. So it is one level higher barrier, and on the top of it, it has to have industrial application. Innovation can be disruptive, like the first Apple iPad, or it can be incremental, like subsequent phones. In pharmaceutical industry, the key is a problem which can be unmet medical need or need for a better solution. It can be product or a process. And then you have to have a solution, and you need to identify whether you are able to solve that problem. You can identify a problem, but whether you are able to solve it in a finite resources. And most of the challenges lies majority in Indian counterpart in this part and relatively on the smaller part. In the pharmaceutical industry, it requires a teamwork of a people with knowledge in various subspecialty of science, and it involves biology, chemistry, formulation development or pharmaceutics, and it involves preclinical and clinical studies, and this requires special skill sets. And you have to manufacture the product before it can be commercialized, and it requires a special skill sets. And to have everything coming to the market, you need a regulatory team. This is a usual cycle, and everybody feels that if I'm successful here, I can go all the way to the phase 3 and success launch of the product. But the process is met with significant challenges. There are multiple failures at each stage, and it's an iterative process. So once you fail, that does not mean it cannot go forward. So you have to come back and redo it and modify it, and your chance of success goes high from a preclinical texas, from a disease discovery to the preclinical testing. Once you cross that, your failure rate drastically reduces, and it's significantly small when you reach a phase 3, provided you have done proper phase 2, proper phase 1, and that involves multiple phase 1 and multiple phase 2 if you want to get a phase 3 successful. So having done this introductory part, I'll take you through some of the innovative products which we have developed. So this is the first one, which is a Rizorin. It has come out of a public-private partnership between IIM Jammu and Cadila Pharmaceutical. It used the traditional Ayurveda knowledge converted into a modern medicine. It involves rifampicin 200 milligram, piperine 10 milligram, isoniazid 10 milligram. If you know, in India, rifampicin conventionally is 450 milligram. So with the addition of piperine, it has been brought down to 200 milligram. The genesis of that was TRIC2. The scientists at IIM were concerned or curious why TRIC2 is coming in Ayurveda literature multiple times. And then they say that, okay, what does it do? So it induces the efficacy of the product with which it is added. And by doing a lot of experiments, they identify it is a paper, long paper, and piperine of the long paper is responsible for its activity. And then how do you use that knowledge that piperine is responsible for the activity of a TRIC2? And so we joined hand at that point in time, and we identify rifampicin is a drug which needs to be bioenhanced. And so there were multiple studies, and this is one of the studies where there is a 3-way crossover, 3 period. And it showed that in humans also, after all preclinical, that 200 milligram of rifampicin with piperine 10 milligram is equivalent to 450 milligram of rifampicin. So if you look at a blue and red curves, And then the bottom line is 200 milligram of rifampicin. And then it maintains the level at a day 14, day 0, day 14. So if you look at control arm is known with the rifampicin, it induces its own metabolism. So rifampicin level drops over a time. When you add piperine, the levels don't drop. Day 1 and day 14 are the same. And by and large, the problem with tuberculosis is, one of the problem is decreased bioavailability over a time. And so adequate amount of the rifampicin is not in the blood and you don't get a proper cure. So with addition of a piperine, the levels are maintained all throughout the study period. And because it is maintained all throughout the study period, I mean, superconversion rate are also higher at 4 weeks and there's a little higher cure rate. So in nutshell, what it does is it improved compliance because it is a lesser amount of rifampicin in the stomach. It improves the GI tolerance. And one major problem with the rifampicin is that for initial period, people don't tolerate and give up taking the drug. So it's better tolerate, it's less hepatotoxic. Actually, in the clinical study, we have shown that those who have hepatic toxicity because of the rifampicin, when you switch over to rizorin, they tolerate it all throughout and they get cured. It maintains bioavailability over a time. It's not a one time. So, I mean, in a phase 3 study, we have shown that bioavailability of a rifampicin with conventional rifampicin drops down, while with this product, it is maintained all throughout. And it's available at a lower cost. We did not charge for the research and the development cost on the product, so it's available at a lower cost. Then I'll take you through something which is modern medicine. This is a herbal, this is a modern medicine. Everybody knows sitagliptin. So when it was launched, we reviewed the patent literature and said what is protected. And we realized that this is a portion where we can play and we can come out with the patent. So we synthesized a series of compounds, and this compound where we have added phenylalanine showed the activity as good as sitagliptin. So it was a prodrug, and you don't see any concentration of our drug, which is outside NCE, but within the body, it is as good as sitagliptin. So, and these are the example that how it behaves inside the duodenum at various parts of the intestine. And this is a oral sitagliptin. Whichever amount you give is only sitagliptin which comes into the blood. So you can come out of the patent and you have your own compound with a limited risk. And then we did phase 1, phase 2, phase 3 study, and we showed that it is as good as sitagliptin. Sure. It's okay now? Okay. Yeah. Still niche? Okay, okay. So even if when there is a patented product, you can find out a niche and develop a product with a lower risk. You don't have to start with a chemistry all over. So another example is a Sapsevac, which is essentially hit-kill microbacterial. It's essentially a repurposing of a leprosy drug. It was originally invented for a leprosy to reduce the duration of therapy. During the course of everything, we realized that it is very safe, and whatever amount you give, there's no side effect. And we were perplexed because as per the modern medicine and modern teaching, at some point you have to see a side effect which is systemic in nature and which will be devastating and which will reduce, control the amount of drug which you can give. With this product, we have given from 0.1 ml, which was the usual dose, we have given as high as 10 ml at the same time. So huge therapeutic index without any systemic side effects. So we were kind of curious to find out why it does so. And so, I'll just run you through quickly. It's a potent TLR2 agonist, but it turns out it is a poly-TLR antagonist. So if TLR is overexpressed like 4, 7, 9, it downregulates those TLR. If they are normal, it does not have any effect. And on its own, it does not have effect on this. It is only when they are overexpressed it downregulates that. Then it also it has effect on pro-inflammatory and anti-inflammatory cytokine, which is typical in the sepsis. Generally you work on one arm of the inflammation. Either you have a pro-inflammatory activity or anti-inflammatory activity. This product, it reduces both pro and anti-inflammatory cytokines. And this happens only when they are overexpressed. So this is an example of significantly high TNF-alpha and IFN-gamma, both are reduced drastically. But on its own, it induces TNF-alpha and IFN-gamma in normal individuals. Then we looked into what happens to the gene profile, and then we realized there is a significant match with the genes which are overexpressed when in a gram-negative sepsis and when we give MW or Sepsivac. So with that, we went to the next level, because everybody thinks that immunotherapy will take its own time and it will not be quick in response. So we did a small animal study. There's the rabbits which are injected with LPS. They get rise in temperature, and at that point in time, at the peak of a temperature, we gave MW, and if you see red line, temperature comes down. And this it does it because works through innate mechanism. Innate immune mechanism is the fastest mechanism in the body and is responsible for prevention of all the challenges which we face during the lifetime. So whatever we know about immune response being delayed is for adaptive response. Innate immune response is very quick to act, and this is how it can manifest very quickly. Then we went to the next level. We created an animal model of gram-negative sepsis, where animals will die within, survive for some time so that you can have a drug going on, and then they will die eventually. So if you look at it, the way we did it, we treat the patient. We induce sepsis by giving E. coli, then the standard of treatment, antibiotic, to which this organism is sensitive, along with steroid, and then on the top of it, we added MW, and if you see, there's a significant improvement in the survival. And then it also reduces TNF-alpha. And this experiment has been repeated 30 times at 3 different locations with 3 different groups of animals. With that, we went into human. Why we took so much pain was because everybody has failed in the sepsis management. People have tried, multinationals have tried, everybody has tried and everybody has failed. So we were very skeptical why we should be successful. So we took extra precaution. But phase 1 data again showed that SOFA score was dose dependent. There were 3 dose levels, and we showed dose dependent decrease in SOFA score. SOFA score is a surrogate for sepsis severity. And then in a phase 3 study, we showed that there's an absolute difference of 10% in the mortality, and treatment benefit is 55%, and very low number, NNT. If you treat 9 patients, 1 will survive. And there is a significant improvement in SOFA score over time. And the beauty of this was, higher the SOFA score, the efficacy was better compared to the lower SOFA score. The SOFA score less than 7, SOFA score more than 7. And SOFA score is proportionate to the severity of the disease. So higher the severity, better the outcome. Then I'll take you through some biologics which we have developed. So everybody, I'm sure, knows about the rabies and its generation. So originally, Pasteur did it, and there were multiple injections. Then spinal cord to brain, 18 injections, 14 to 18 injections, and then cell culture, which brought it down to 5 injections. But at all throughout, it was an inactivated virus. It is only the substrate which kept changing. But if you look at a science part of it, it is only the G protein which is immunogenic and is also responsible for the disease. And protective type of G protein is also known. Okay. So what we did was, this is a commercial now. It's the world's first 3-dose recombinant protein rabies vaccine. So we prepared that protein in the recombinant way and did all the studies. So if you look at this, Rabipur versus Trebis, it always consistently gives higher seroprotection level. And then we did 17 arm phase 1 study, and then we did multiple dose finding. So 2 dose versus 3 dose and 4 different dosage level, 5, 10, 20 and 50 gram microgram, and then show that everything is identical. It's as good as Rabipur. Then we did a phase 2 and evaluated 2 dose form, 10 and 50, and in a 3 different form, 037 and 03. And again, we showed that it's identical. And then we did a phase 3 study, which is one of the largest phase 3 study for rabies ever done in the world. 800 subjects randomized 2 to 1. And again, we showed that it is as good as Rabipur, if not better. Then I'll take you through some of the formulation tricks we did. So this is aqueous injection of vitamin D. Everybody is familiar with vitamin D deficiency prevalent widely across the world, particularly in India. And then when you want it quick, I mean, everybody thinks in terms of taking an injection. Since it is a fat-soluble vitamin, injections are oil-based. It has to be given deep intramuscular. They are painful, and they release the drug very slowly, and they take practically as much time as it is taken with as if you have taken orally. So there is no significant advantage except that you feel comfortable that you have taken injection, and you get little pain as a part of a proof. So what we did is we remove the oil, converted into an aqueous formulation by using glycopyrrole as a diluent and surfactant and chelating agents and buffer. And if you see that, this is a conventional oil preparation, and this is our preparation, how quickly it rises vitamin D level. But vitamin D is not the active substance. It's a 25-hydroxy vitamin D. And then if you look at, compare with that, there's a level, normal level. And in 2 days, almost everybody achieves vitamin D level, which is normal from, subnormal. And it's maintained all throughout the study period. And if you look at this particular study, oily preparation has not achieved vitamin D levels. So it corrects vitamin D deficiency very rapidly. I mean, you can stop me anytime, huh? Because I'll continue. Okay, so then, is there a timeline? Okay. So this is based on the BMJ article. There are biostatisticians in UK. They published this article with the editorial that we say that if there is a poly pill which contains 3 antihypertensive, 1 lipid lowering agent, and 1 platelet aggregate inhibitor, you can prevent 80% heart attack. So this was in 2003. So our team also read this article, like multiple people in the world. And then we thought that let's try it. And it's a success story. It's available by name of a polycap. So it contains 3 antihypertensive agents: hydrochlorothiazide, atenolol, ramipril, 1 lipid lowering agent, simvastatin, 1 antiplatelet agent, acetyl salicylic acid. And there's a selection process, which one you select, how you select. I won't take you through the details of that. But challenge of developing a polycap was volume. If you look at a 5 ingredient individually, it's a significant volume. And you have to put it in a single tablet or a single capsule with a limited volume. So you have to reduce volume drastically, at least tenfold. Okay. And then when you do that, you have to maintain stability and also bioavailability. Because when you put it in a different ways, either the stability will not be there or bioavailability of not there. And then if you maintain that, hopefully this activity is maintained. Sometimes there is you can preserve this and this, but there is a loss of activity. So we showed it and published in the American Journal that when you do it as a polycap, I mean, you can maintain that. And this was done against each individual. And it was again an innovative design to show bioavailability of 5 active ingredients against each one of them and do it effectively on the same individuals. And then this is suggestive of that, okay, it maintains the same activity and for a blood pressure as well as lipid lowering and aspirin. And this was first published in Lancet for any Indian product, and Lancet published it with its own editorial. So this will only tell you how complex the study was. 9-arm randomized comparative, 50 centers, 2,000 without cardiovascular disease, and active treatment for 12 weeks. Okay, and we measured blood pressure, heart rate, lipids, urine thromboxane. This is for aspirin, safety and tolerability. And then we did TIPS 2, which was a second study, and then TIPS 3. And in the TIPS 3, Wellcome Trust came forward on their own without our soliciting. Actually, they solicited us that we are interested in study. And we did this study, 5,000 patients followed for 5 years in this many countries. And now World Health Organization has put it in the essential medicine list. Okay. Can I continue or stop here? Huh? Okay. COVID. Okay. So, I mean, we had a Sapsivac approved for gram-negative sepsis before COVID came. And then COVID came, and in the early part, we had to close down our factory for 14 days because people got infected with the COVID. And that's where the people with knowledge started talking about, can we have Sapsivac given to us? Though it is not approved, nothing was available. And during that time frame, with the consent, there were 2,500 people within our organization who wanted to have Sapsivac. And there were 1,200 people who wanted not to have Sapsivac. So it became a control, but not a planned control. And because it was a COVID time, everybody was followed meticulously. And then we analyzed the data. If you look at it, I mean, this red solid line suggests who got COVID in the control arm. And blue solid line, how many got the COVID is those who received Sapsivac 0.1 ml once. So there's a significant difference, 6.47 versus 1.95%. And if you look at hospitalization because of COVID, 3.47, but hardly anybody. And so this turns out to be prophylaxis in terms of vaccine efficacy of 74% for getting COVID. And for getting significant COVID is 0%, I mean 100%, and around 90+% for getting a symptomatic COVID. But then there is another group. He says we will give you 0.2 ml, okay? And then they showed that the protective efficacy is 93%, and these are healthcare workers. They are high risk. They were managing COVID, and they showed that it's 93% when you give 2 doses. Then, I mean, this will give you an idea that how it happens. So it protects you for 6 weeks, 6 months. So, I mean, considering innate immune mechanism, no vaccine, no correlation of antigen, no antigen sharing, I mean, we were wondering how long it lasts. So at least for 6 months, they could protect it. And so, I mean, if you look at this is highly significant. You give it to 4 people, and you, I mean, one is saved. Okay. So then from the same group, when they were followed further, the vaccine became available. So they got the vaccine on the top of it. So now those who had got MW plus vaccine, this group, and those who got vaccine alone. And again, there is a difference in efficacy. And then we went into the mechanism. What is the innate mechanism which gives you protection? And this is the first time that anybody has shown that it is NKG2C, which is a memory NK cell, which increases following MW, and it decreases NKG2A, which is inhibitory NK cells. and it decreases drastically. And then there is a ratio of NKG2C and NKG2A, which is responsible for prophylaxis using innate mechanism. And so this is an overall summary of how you can protect yourself in absence of knowledge about antigen from a respiratory tract infections. You don't have to share the antigen, you don't have to have a vaccine, prophylactic or therapeutic, just by inducing your innate mechanism, increasing NKG2C and NKG2, decreasing NKG2A, you can be protected. And NKG2C are known to have a longer life and memory. Thanks. Yeah, baculovirus express protein. So we have a baculovirus expression protein, SF9 baculovirus. Yeah, okay. So that's the same facility we use it. So the reason we went for that, okay, I mean, there's an interesting story behind it. So while working on all those biologics and challenges with immunity, we realized that we can have a good adjuvants. And actually we developed a couple of adjuvants. Some of them were small molecules, known products repurposed. We have not used them in any of our products, but that made us think that if we have a good adjuvant, can we get a good vaccines? And that's how we enter into novel vaccines. That's how we got into relationship with baculovirus SF9 system. So that's a history. Yeah, please. Yeah. So is that a pro drug or is it a new compound? It's a new compound, but pro drug. It's a NCE. By definition, it is a NCE. As an NCE, that compound is not known, and that compound was out of the patent of a month. Yeah, yeah, yeah. But patent does not look into what happens in the body. Patent looks into what is outside. You can sell what is outside. You can't sell what is inside. If I can take any drug and put some ester group on it, put it in the dosage? Yes, provided you are not infringing the originator patent. So you have to look at critically what originator has claimed and what he has not claimed. So that's how we came out with that. This is the area where we can do something, and we did. And then it's not easy. I mean, we had to synthesize 40 compounds using that active site, active moiety. And then couple of them turned out to be as good. Couple of them turned out to be no good. Okay, they did not get cleaved. Yeah. That's actually supported by NMIT program of yes. CSIR. Yes. No mention of yeah. We gave all money CSIR. Yeah. And NMIT. Yeah. And they are looking for some support, some acknowledgement. You didn't even mention about. Okay. So I'll tell you. Okay. I'll tell you about everybody. I mean, so Rizorin CSIR support, Sapsivac CSIR support. I mean, Sapsivac original gram negative was a CSIR NIMITLY program. When COVID came, Sapsivac again was a NIMITLY gram negative. I mean, COVID program. DPP4 was a CSIR NIMITLY program. So 3 out of 5 are NIMITLY program. Vitamin D is our own. Rabies is our own. Yeah, I'm acknowledging. Okay, if you want, I can put it. Okay. Yeah. So. Yeah. So I mean, okay. So for the interest of everybody, so we have been. Yeah. Go ahead. Yeah. Yeah. This acknowledgement is there. Project supported by CSI. Yeah. Yeah. Sure. Yeah. 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 Okay, I'll take one step ahead. Okay, so when we had initial data on a gram-negative sepsis, okay, initial story, we did everything in-house, and we were wondering where do we go and how do we get it going, because it's a high-risk project, because nobody has been successful so far. So we went to NIMITLY, and NIMITLY encouraged us all throughout. And then there was a PGI who was roped in as an academic partner. So PGI was academic partner. Cadila had done initial groundwork and immediately became a funder, come advisor, come monitoring. So everything was done by CSIR Nimitli group. And there were a lot of ups and downs and challenges during the whole process. So as a matter of fact, which you also may not know, there was a special committee created outside the monitoring committee to review the work, and that committee said that everybody has failed and this is high risk, we should drop it. Okay. At that point in time, we were, more adamant, but our monitoring team was also very, that there is hardly any risk. You have gone up to phase 1, phase 2 data is good, let's go to phase 3. And that's how it went to phase 3. Otherwise, it would have been dropped between phase 2 and phase 3. So that's the Nimitli strength. That it, yeah, no, success everybody knows. But what I'm saying is, for people who are going to go, so we have been, beneficiary of ICMR projects. We have been beneficiary of DST projects. We have been the beneficiary of DBT projects. But Nimitli has a tremendous strength. That's what I want to communicate to everybody. Sir, same thing. People like you, if you acknowledge only, we get to know, people get to know. Yeah, so I'm telling it openly. Say Nimitli is a great program compared to all the other 3 organizations from CSIR. The reason being is they not only provide you funds, they help you to take you all the way through. And in between, if there's a correction required, they will do correction also. So say, for example, DPP4 was conceived inside. Again, we went to Nimitli. And original concept was that with the cleaving mechanism, we will have a combination of the product so that we don't only clear diabetes, we also, minimize the kidney complications. After 2 years of work and interactions, they say that this becomes a little high risk. So restrict to whatever you have achieved, don't go beyond it. Okay. So Nimitli, from a funding agency perspective, it is not only funding, it is much more than funding. And for industry, those who are from industry, 50% or more, whatever goes to the academic organization comes as a grant. While whatever comes to the industry to do whatever they can, it comes as a loan. And that loan is again a soft loan. You have to pay after some time. You don't have to pay immediately, and the interest rates are very low. So one of the best programs we have in our country is the Nimitli program. And we have witnessed it. So, the only catch they had it was any company can have not more than 2 programs. So we could not pursue more than 2 programs with them.
Slide, please. Any student? If there is no question, so thank you so much, sir.
Thank you, sir, for sharing these valuable insights into industrial pharmaceutical innovations. I now invite Dr. Debendra Mohapatra and Dr. Vijay Singh to honor Dr. Baklesh Khamar.
Anywhere. That is the beginning, not at the end. So the reason it was not that, because it's very difficult to remember each and everybody who is contributing. See, my one limitation is practically, I'm a spokesperson for everything. But there are so many people within Kerala and outside Kerala who have contributed to this. So it becomes a very long conversion to the end. And there are all challenges. We had regulatory challenges, we had
challenges with population waivers, environmental waivers, all the challenges are resolved by multiple people, and some of them are still not in the category. Thank you.
Thank you so much, sir. I now invite Dr. D.S. Reddy, sir, to honor our chairs for the session, Dr. Debendra Mohapatra and Dr. Vijay Singh. Thank you so much, sir. With that, we conclude the session 4. A big thank you to our esteemed speakers and session chairs for an insightful session. Up next, we have session 5, the Students Special Academia Industry Interactive Session, where young researchers will have a unique opportunity to engage with the leading experts from both academia and industry.
We'll just take 2 minutes to arrange the stage according to students' interactive session. So without wasting much more time, I would like to request session chair Dr. Sri Ram Gundi Meda, HOD Chemistry, IIT Gandhinagar, to come over the stage and introduce our distinguished speakers and panelists of this session. So I would like to request Professor J.S. Yadav, Research Director, Indrashil University, Professor V.K. Singh, Institute Chair Professor, IIT Kanpur, Dr. T. Raja Mannar, Non-Executive Director, Spark Limited, Professor Tapas K. Kundu, Professor at JNCSR Bangalore, Professor G. Mukesh, Professor and Dean, ISC Bangalore, Dr. D.S. Reddy, Professor and Director, IICT Hyderabad, Dr. Devesh Das, President, Branded SBU Cadila Pharmaceuticals, Dr. Vinay Kumar Srivastav, Professor of Practice at Indrashil University and Assistant Vice President, VFS Cadila, to come and take their seats on the desk. Dr. Sriram Gundimeda, the stage is yours. Since we have Dr. Bakulis Khamar also with us, so I request sir to come and take the stage. Can we request all the students to come forward? This session is especially designed for you. Come and take the front seats, please.
You can also call your friends standing outside the hall. Greetings, everyone. Okay, this session is an interesting session which has both academicians as well as industry leaders. The objective of this is directly coming from the students' perspective. The session is aimed at bridging, telling about why translating the science into medicine, drug development can bridge the gap between academic research and the industry. So this session aims to provide students with some understanding of various career opportunities, skill development, emerging trends in pharmaceutical research, drug discovery, and industry academia research collaborations. Finally, the session aims at to understand the transformative power of research in modern education and to come up with a possible reference guide that can help in pursuing research in the right direction. Nothing but science for the society. So again, I repeat, the session serves as a critical bridge between academia and industry, equipping students with insights into contemporary and relevant pharmaceutical research, drug development, and career opportunities. This probably will give you some sort of understanding on multidisciplinary thinking, industry readiness, and translational research strategies. So we hope that this interaction with the experts sitting on the stage will empower you to make a meaningful impact in both academic and industrial sector. So I invite the moderator for this session, Professor Santosh Garpure from IIT Bombay, to take over the proceedings. Thank you.
Good evening. We had a long day, but this is a session which is particularly geared towards the students. And I hope to derive maximum benefit out of this session. We have a very distinguished panel here, and my job is very easy in the sense that I just have to ask the question and let them do the talking. Because what you are here to hear from is largely from the experts who will share their experience. The way we will do it, as what I'm thinking is that I will request each one of you to kind of, answer one of the initial question which I have related to the topic. And as we proceed further, we will also try to take questions from the students and other participants. So what I would encourage you to do, rather than we moving the mic around, you please jot down your question on a piece of paper, share it, and then they will share it with the team. They will pass it on here, and I'll ask those questions to the panelist. If you want to ask the question to a particular panelist, you can also mention that, and I will try to direct that question to the concerned panelist as well. So let me begin by asking the first question to somebody who is right at the center here, Professor V.K. Singh, who has been leading the institutes, right, as a faculty member, worked with the industry, headed an institute. you have looked at science from both the sides, fundamental science as well as translational aspects, which are very important from an institutional point, from the impact point. So the question I had particularly is, can you please share your perspective? What are the current challenges in doing translational research in the academic setup with the industry focus? And this is particularly keeping in mind that students are looking at a career. And from that angle, we would like to hear your initial impressions.
Important. Hello. Aspect is, if you look at from the point of view of challenge, the ecosystem. Lack of ecosystem actually is not there at large number of places. And focus on translation actually started recently for the last few years. So that is one aspect. The second thing, several institutes, they are focusing more on translational work these days, especially IITs, despite having any system for career progression, especially young faculty members. I think there has to be some kind of evaluation process also. When a faculty joins as a young faculty assistant professor, after career progression, you go to associate professor, then you become professor. And people are more interested in publishing papers and that is fine from assistant to associate. But I have a feeling there should be several other verticals. And one of the vertical could be translation. And only this week, a new career progression policy has been actually approved at IIT Kanpur, where after becoming associate professor, you can have yourself evaluated based on research, teaching, and translational. What means that will be your main theme. And such things actually help a lot. But again, this is a career progression for faculty. And I believe if the good ecosystem is there in the institute, I think more people will go for such things.
No, I think it's a great point. We are talking about this is career progression for faculty members, but actually faculty members drive research where students are direct or indirect beneficiaries of that. So obviously focus on the translation by the faculty members will also gear up students to take up more translational work. So I think that's a very, very important point, something that many of our leaders today and potential ones in the future should make note of it. Institute like IIT Kanpur, institute like IIT Bombay is actually working on using these tracks, translation as one of the tracks for the career progression from the point of view of faculty members as well. So I think it's a great point. Let me throw the next toss to Dr. Rajmannar, who comes from industry. And this is basically a question which is there in many of the students who want to take up jobs in industry. Right. I mean, one of the thing that is there is everybody who comes in chemistry, particularly organic chemistry, you ask them, they always say that why I chose organic chemistry because it will get me a job. So what are the skill sets that industry looks at in the student when you are looking at prospective candidate who want to take a plunge in the industry?
Yeah, sure, definitely. I mean, at one point in time, probably in early 1990s, where everyone was looking at the process research, the emphasis was there for organic chemistry. But today, whether you're organic chemist, whether you're inorganic chemist, whether you're analytical chemist, you're related to regulatory affairs, there is a place for everybody. But what is important is that your self-conviction, what I can be a person in terms of adding value. Because in an organization, there are several people, and if you want to be really first time doing your job interview, you need to be fundamentally strong what you know. Don't go and tell that I know everything. If you know 10% of what you read, you tell this 10% I know very well. But you try to answer as much as possible that 10%, because it's not necessary that you need to know all the things what you learn, but you learn several of the things, but you like some of those things and you can express this is my interest, this is my understanding, this is what I can do best. Then you're sure that you will get a job. That's the first step you need to be keeping in mind, because that's how we try to look at evaluate a student when somebody comes. If you are interested, you are telling the truth that this is my interest. You don't tell that I know everything in organic chemistry. This is what I know, this is what I can add value, because it's not that you are going to do 100 reactions in every year. In industry also, there are 10 reactions you are going to do. It's not that 100 reactions you are going to do. So because if you're working on 20 molecules into 5 steps, that means 100 steps. So there are 100 people. So you need not have to know that all 100, but whatever you know, you need to be, that tells you that you are coming with an interest to do the best what you know. So that much of information you need to convey, because your presentation in terms of what you are, what you know, how you are going to be adding value to the group or to the company, to whatever thing is most important. That's what I would encourage students to be, point blank, that this is what I am, this is what my capability is.
I think you are underlining what we keep emphasizing in the classrooms, right? Basics, they're timeless. And it's important to be honest, know what you know, and don't try to bluff your way out. Something which is a timeless advice at every point in time. I'm very happy that industry actually looks at that, because the general impression, I must tell you, for the students is that you need to be smart, and smart doesn't necessarily mean being trying to bluff your way out. So I think that's a great advice from somebody who is an industry veteran, that be honest and then pay attention to your basic. It's not that how much vast thing that you know, but whatever little you may know, you should be able to express it very, very clearly. That's a great message there. Now let me take the next question to Dr. Bakulesh. This is again, students generally are always wondering about the way industry functions. Uh, they are always struggling to find out what are the typical problem statements that they may be working on going forward. And because we don't have many such interactions what we are having today, they are not clear on many occasions what are the challenges that industries are working on today. So the question I have is, what are the biggest challenges in bringing a new pharmaceutical product from research to the market that one faces? And how can young scientists contribute to this particular challenge effectively when they are coming in industry? They try to do something which is already made available outside India, and they try to make it in-house and relatively inexpensive way. So there, what is involved in the process chemistry? Can you identify the processes by which the same compound, well known, patented by so many people, still you can make it non-infringing and cheaper? So there, your creativity and background knowledge helps. Same thing applies to formulations and enzymes. Can you identify different methods of analysis? Can you identify different methods of formulation? Complex beverage. So these are the skills which are generally required for a me too or a generic product. Some of the companies like us, those who have ventured into trying to do something innovative product, there you need a skill set to think differently. Is whatever you have read, whatever you know, is not going to be helpful when you're trying to come out of whatever is meant. So, say for example, say Sapsivac, which was spoken multiple times. So it was gradual, one step at a time. First, if you want to be innovative product development, the first part is you should be curious
And like you said, you should know what you know and you should know what you don't know and where you can go for help. Because everything is not possible in a drug development. All the things are not possible by a single individual who have innovative product coming to the market. And what an academic can do or what a student can aspire is, does he know what he knows? Does he have a flavor to put in one scratch himself and adapt to the environment to do something differently where large number of Indian, PS will say this is not something which you should enter in. But currently the environment is not right for getting innovative products. And it's a whole ecosystem. Like I said, in the academic environment, the ecosystem needs to change. Same way in the industry, in the mindset of people, in the mindset of regulators, in the mindset of government, it has to change that innovation and innovative products can come from India. Like the example I gave you before, if we would have done this activity in China, we would have got products approved in 5 years' time. But because we were Indian and stick to the Indian regulatory environment and the other conducive environment, though it was supported by immediately, though CSR was at our back and helping all the way throughout, we got the product approved until the sitagliptin product patent expired. So we could not reap the advantage of whatever we did. It was all lost to the, essentially to the nation, partly to CSR and partly to Cadila. So all this needs to change. At each level. But as a student, you should have a flavor of doing something differently, thinking differently, and be passionate about it. That is okay. Based on whatever I know and whatever I can think, this is the best foot forward, I will do it. And I'll give a classic example in the novel variant. mRNA vaccine which came up very big in the COVID time. Carico Catalina, if you have known the story of her, she was working on mRNA since long. But then she was going out of the box, she was going against the wind. So funding agencies stopped giving her grants. So she did not receive any grant for a couple of years, but still persisted. And if that was not adequate, she was demoted in the academic organization. Okay, rather than promoting, she was demoted. And then its results were seen only when the COVID came. And the other part of the story is she could not go on her own. She struggled. And as a chance, everybody knows that as a chance, at a printer, she met somebody who helped her out in stabilizing the compound. So you have to have an ability to network. You have to have an ability to go to somebody who knows the counterpart of it and take it forward.
Okay. Great points. From the student point of view, just to summarize what he mentioned, one is that there are two main streams, if I may call it, the process chemistry and then the discovery or medchem as it is called in the Western world many a times. These are the two main tracks. And one more track he elaborated, something he didn't say it in as many words perhaps, is some of you, if you happen to take up government jobs in the pharmaceutical related sector, please clean up the regulatory processes so that we can move forward. And that's a great career opportunity right there, right? Through that. So let me come to again academic,
They also won Nobel Prize for immunotherapy, PDL1 and PDL2. They had the same track. Hanjo, when he filed a patent, he had to get a company which was not a typical pharma company, but Japanese government wanted. If you want to file a patent, you have to have a company file a patent. So a small company filed a patent. There was nobody who was willing to take it forward to develop a humanized monoclonal antibody. And actually, he spent his own money initially. Same story for James Allison. We changed everything. The cancer therapy. He had the same story. He was in Berkeley. Nobody was looking at him. He filed a couple of patents, published in a science journal initially. Wonderful results. Nobody was coming forward. A small biotech company, Medanax, they helped him out for the initial part. BMS, as a matter of fact, everybody takes a pride, but BMS was not willing to look at them. BMS, when they, as by compulsion, they took it, okay, we will take it. They said, identify one person within the organization, gave him $100 million, and say that we are going to not ask you any questions. It is a loss to the organization, and we don't mind spending $100 million for nothing. And that's where everybody comes in picture. So the industry has to take a risk, individual has to
Sure.
take a risk, and then everybody has to come together.
Yeah.
And there is success.
No, I think there are many such examples which exactly. So see, academic research going on to industry. That is one aspect of it. But academicians going on to become entrepreneurs. So let me ask this particular question to Dr. Yadav. Uh, you know. What do you think that are benefits of academicians transitioning into the successful entrepreneurs, particularly in pharmaceuticals?
I'll give you a couple of examples. Many times our survivors, they start doing it. Like you got a government job, easy job, continue. But I'll give you a couple of examples. There was one of my students, I think Batrano Jayaprakash, right? He came from Singapore. Nobody's ready to give the job. Then he has to survive. जैसे किसी को आप साबरमती में फेंक दो तो तैर के बाहर आएगा. Exactly वैसा है, right? Now Jayaprakash started in Hyderabad, and when I went and saw that, it's a huge facility. And now I'm sure he's making more than within a period of 3 years. I think he has touched 50 crores turnover almost. So because otherwise you would have got suppose in IIT they have given scientist C or even you would have been dead. Another example, I think, but business-wise. Business-wise, I'm not saying absolutely. Again, I'll tell you, Dr. Ravi Ramara retired as a director. He was a tough guy. He will abuse also. And then when he was retired, and in those days, there was nothing like today, Bhatnagar Fellows, distinguished scientists are kind of not there. So what I had to do, he got something to continue. Something from here. Then people said that no, they will not allow you to come in because once you are stuck, so they want to plan. I don't want you to come to ICT and all. So he said, yeah, I will start my own. And he started, with a very little grant from here and there. In 95, 96, of course, we were there from backup support. By 2010 or 12, it became a 500 crore company. Wow. It's only survival because I can also survive. Right. And also, it was totally science-based. Like we were working on cancer treatment center. I told Ramara, theoretically, क्यों नहीं बनाते हो? So we were already there. So we did, and we started making them. So there was a problem in natural product because there were a methoxy group. To remove methoxy group one position, you lose 50% of material. That was one thing. And second, whoever works on cancer plant, they have to take active heat.
Right. Right.
Otherwise, they will have all kind of skin disease and all that. So he said, it's a good idea. Then he made synthetically. Of course, I was there. So he made it synthetically. That was far purer than the natural product. Even in the natural product, you go to echinocandin, at least you have to go 5 steps.
Right.
More. He would lose natural product. Then he started doing, and one product gave him 100 crore business. Out of that, 60 crore was in profit.
Wow.
And he got all DSIR awards, SIR award, all these awards. See, again, I'm telling you, it's the idea that comes from there, made synthetically, because as a chemist, he knew all these problems are there. It's an cancer. Another things I get, a couple of examples I'll give, not take much time. Now, Indo-French wanted that some product we are not able to commercialize. So Modi said that, Yadav sab kuch to karo. We are getting very bad name. So I went to Ramarao requesting that we'll give you Mr. Prostar because if you are taking RU486, ladies has a problem. And then we said, why not make a combination can be given, that is safer. Grammar has I don't go for it. Usually it's compound. But I requested him, you don't spend money, you say Indo-French will spend, but at least you agree. So he agreed. But, we made misoprostol, this Mapatra was involved. And we gave him compound. How much profit he is making in that one? 500 crores profit. Okay.
Yeah.
And still he has not paid to IICT.
But that is post-retirement, right? So.
Very training. IICT only gave training. Exactly. But anyway,
Yeah.
so this was 2-3 example.
Right.
Again, I told you, it's not your IIT guy, or correspondent. He was Dr. Mehta first. Because we were classmates from BSc, MSc, PhD already. So he was come from a very lower middle class family. Then he immediately needed money. He got job in Ranbaxi in those days in 72, 73, right? He was doing Ranbaxi. Then we were talking one day, so we said, then we may Jara US actually. So he called me and said, if you stay with me, then go to US. I stayed there. I told Pandey ji, आपको तो इतना knowledge है, सीख ही लियो, अपना क्यों नहीं शुरू करते? Then he discussed his Govardhan Mehta. Then his brother-in-law supported and another finance company supported. They started train under in 76, around that, 75, 76. Today, it is almost 700 crore. Again, he could have got a government job. Could have been happily a professor somewhere. Today, it's giving to the professor. It is empty number of examples I can give you, where because of survival, they started and they have been successful.
Right.
Most successful. And at least, Mahapatra, how many students of mine who have started now? At least 5, 6 in Hyderabad. Someone is making in 20 profiles, someone making this. But at least they are making over 1 crore profit. 1, 2 crore, 3 crore, 1, 5 crore profit.
Wow.
And majority of them, I will tell you that, yes, Reddy once retire, unke student ko job nahi dete ye. So that's good. That's good. So they have survived. Now they give job to IICT. Once you took PhD from IICT, they employ.
Right.
So they have become employed.
Right.
So what by that saying is, if you want to start your own, okay, nobody give you idea what is that. It is you have to survive, you have to have an idea. Then what you do, I will say, actually CSR is very beautiful scheme, but now I think why they have told you. The one thing is you work in industry for 3 years, 4 years, 5 years. Don't look for academic job, otherwise will become usual when the industry comes. So you go and work in an industry. ऐसा नहीं होता है. Then it works. अब आपसे कुछ कैंडिला से कितने बाहर गए और कितने अपना योग इंडस्ट्री खोल दी है. मैं एकेडमी से आया इंडस्ट्री में.
Right. डॉक्टर साहब एकेडमिक नहीं निकलेंगे तो इंडस्ट्री में आएंगे कहाँ से?
Here, one work for couple of industry. Then come out, learn the what you are to say. Your process development or your learn. Industry need and PhD need is different. So you learn. Then come out and start your own. Majority of people are there. Don't copy that industry thing. Don't take industry.
Right. Right.
Start your own.
Yeah.
Otherwise, industry will kill you because you cannot compete with them if you start with their own money.
Yeah.
So, again, even in say lot of our software, they started their own. So they were working for some company. And once they learned, they came out and said, sir, we will make for you. Why? And they started making and they developed that. In Hyderabad, Reddy Lab was one start. And the people who were working with him, they said, okay, why not you go make a meeting? They started making and supplying. Said, why not you go and make me that? Double. Big hour. He said, why not you go and make more? They made, and now, and you reduce normally. They will develop. Because he had a support. I'll give you some money, 5 crore, 3 crore, you start. And product sell to me at this rate.
Okay.
But that's
Yeah.
how
Right.
At least 10 industry came out because of unrelated support.
So basically, you need support somewhere.
They got money, they also got trained, they have the right of vision. I'm sure that is kind of idea. I'm sure they are the famous kind of. Definitely they will have. What I suggest for the student is work with the industry. Maybe you come out and say that, okay, I will make this product. You know, otherwise, any other model, there are so many models where government supports you. Gives you, there are incubation centers, there are kind of things. Again, it was not there, but it's still very. Now you have much better ecosystem. Government gives you loan, government gives you land, government. You go ahead. But the only thing is a clear idea what you want to make. That will not come from out of you. It will come based on the need around you. You observe what is the need around you. Then start. And that need becomes your hand to work in a particular ability. I think I am okay.
Sure. No, thanks. I think some great points from that. Basically, you have to observe and look at what are the challenges at a given point in time, and depending on what your motivation is, you can take up academic positions are not limiting. You can come in, you can have your own industry setup, what examples he elaborated. In fact, just for the understanding of the students, the company you are talking about probably is Avra. What Dr. Ramarao made. In fact, last time I checked, it was about 100 million valuations at which it got invested in the last round when some portion of it. That's what I'm talking about. So company valuation, exactly. So there is one more point there. He started it after retiring. So one of the things that probably we all should look at is not just to stick around in the academic setup and maybe venture out, take the plunge in the unknown and take these challenges, use our science to set up something new. That's another example, another message I think probably from these stories for the academicians as well and for student community as well, because there are many such examples where industry has led to another industry. Your initial job in industry can lead to venture creation of your own, for which there is tremendous support available today from the ecosystem at large. Now, when we are talking about pharmaceutical research, right, it's not one particular side of the story. We heard today through the day that people are approaching medicinal chemistry problems, whether you are a synthetic chemist or a plain pure biologist, right? So a lot of interdisciplinarity is involved in this kind of a research solving problems. So I want to come to Mukesh, who has himself been working on a lot of interdisciplinary ideas, and ask this question: that can you provide a little bit insights from this for the students, that how does one approach multidisciplinary research, particularly in biomedical side, and then even in the drug discovery and development side?
Yeah, good evening. Doing interdisciplinary research in academic institutions, it's quite challenging. And as Dr. Yadav mentioned, things are changing now. It's improving. So there are three aspects that actually prevent interdisciplinary research at many academic institutions. One is, of course, the funding. And second one is infrastructure. The third one is the motivation of the students. All three are challenging. When we consider funding, many of us depend on government funding agencies, and there is no assured funding opportunities. And some of my colleagues, when I ask, why don't you take up something more challenging, long-term projects? When the funding is not available for long-term projects, they would like to take up some short-term projects and the students, the problems that myself uh, faced, motivating them continuously at interdisciplinary areas where no publication is assured. So if they have to wait for 3 years, 4 years to get a publication, certainly they don't want to take such projects. And if the next lab is publishing 10 papers per year, and Professor Vinod Singh mentioned about the number of publications, it's important, but in my opinion, that is killing the creativity by publishing too many papers in a year and incremental work. And students, many of them they want to take such projects so that they can get quick publication. So motivating them to continue. I had a student, PhD student, who is from biology background, and he didn't know how to draw a structure. They said, it's fine. But if you want to take a student from biology, they should be good at biology. And we found one student, and the work I presented today, they entirely based on what he has done in my lab. Whenever I try to say that, let us publish this, and because in chemistry, we try to publish more papers than biologists, most cases. Of course, there are exceptions. He told me, sir, let us not publish, and let us get a full picture of this. And we have not published any paper, whatever I presented today. And that type of students, getting that type of student is difficult. So that is the second part of the students part. And it is possible, in my opinion, and infrastructure facility is the third one. When we have to do interdisciplinary research, we need, for example, at the interface of chemistry and biology, we did not have a refrigerated centrifuge to start the work. And we have to establish all the infrastructure facilities, microscopy facilities, and so on. But I do not know how many of the academic institutions will be able to do, or PI from academic institutions will be able to do that. And these are the three challenges that academic institutions face when we want to do truly challenging multidisciplinary research work. But in my opinion, things are changing, and many are getting into interdisciplinary research. In my opinion, choosing the right problem is the first step. And we can work on wrong problems, or we can work on something which is not going to be more impactful. Choosing right problems and spending enough time on that. And when I started a collaboration with the biologists, we wanted to, because 20 years or 15 years back, we did not know even how to do cell culture. And we made a compound, but we were looking for collaborators to do cell culture work. And it took 1 year, 2 years to do some of the cell culture work. And then we started thinking that we should bridge that gap. And the language that biologists speak and the language that chemists speak are different. And that's where we need to bridge the gap and speak in the same language. And that's very important for the success of collaborations. And for that, I told my students we should first establish an intra-lab collaboration to bridge this gap before going to inter-lab outside the labs. And that's where I started taking students from both chemistry and biology. And to bridge this, these are small gaps, but that makes a big impact later. And now what we do in my lab, the students, chemistry students, they make molecules, and the biology students we have everything in the lab, and they observe what the biology students are doing. And they see some activity and say that, oh, this is the one that is creating a problem. Immediately they give it to the chemistry student, go back and modify the molecules and bring it. And this, I find it very efficient within the lab to do this. And then, of course, collaboration is important, and taking it to animal studies and clinical trials and so on. But in chemistry lab, what I feel is that to fill this gap, we need to have expertise. We need to develop expertise. And for that, we need right kind of people, right kind of problem, right kind of people to work on, and right kind of infrastructure. These three are essential to work at the interfaces.
I think that's a great perspective from the student point of view. I think something that you should pay close attention to. And when you are talking about interdisciplinary research, there are many new areas which are emerging, and that's what brings my next question, and that is for Dr. Kundu. Epigenetics, in a way, is coming up as a very crucial area in drug discovery. So can you elaborate on what are the career opportunities, how industry and academia can come together on this particular aspect, and particularly from the emphasis of what students should be looking at from this domain going forward?
Okay. Actually, epigenetics is which is beyond the gene. So beyond the DNA. DNA is not your destiny. Basically, the DNA modification, DNA-associated protein modification is the final, outcome which you see in your everyday life. And epigenetics, your epigenetic cassette are set by the most important person in your life, and that is your mother. You have got your first cytoplasm from her, one set of chromosome from her. Father has very little importance because he has given you little histones and little DNA. The next thing comes your teachers, next comes your environment. And your any disease, even if it is a genetic disease, that can be delayed by proper epigenetic management. So this thing we have realized much later, because the drug, epi drug has in the market much before even the epigenetics word people understood better. So if I, the cancer drug which has been used, the 5-fluorouracil, basically people, it's an analog of the nucleotides. And then now we know it's a DNA methyltransferase inhibitor. Then the series of drugs start coming, which is histone modification inhibitors. Now the most interesting aspect is any infection, any metabolic disorder, what you are facing, all due to the alteration of epigenetic modifications. Any drug you take, that drug basically alter your epigenetic cassette. So in the pharmaceutical industry now, a new perspective is coming that you need to go for epigenetic evaluation. Metformin. Metformin is the wonder drug, but it's an arginine methyltransferase inhibitor. Aspirin. Aspirin, actually the first parent molecule, we had shown that it's a histone acetyltransferase inhibitor. Then valproic acid, sodium valproate, used for epileptic seizures. It is a deacetylase inhibitor. So now as our understanding of biology, pathobiology is going deeper and deeper, we are understanding that the phenomenon of epigenetics is everywhere. So this is the new era. India hasn't started yet epigenetic diagnostics. It's a full vacant area. Now, why I'm saying epigenetic diagnostics? You see, many cases, before the disease starts, much before we get an epigenetic alarm, for your cardio disorder, for your infection even. So this is a very important area, new area for the chemist and biologist, and it needs an interdisciplinary approach. Because you have to understand the metabolism, you have to understand the modification, you have to reanalyze fluoxetine. The data I have not published yet. It's a antidepressant drug. It induces the acetyltransferase activity and induces neurogenesis. It also induces the dopamine production. But the fundamental reason is because it is activating the acetyltransferase. So it is entirely new area, and both the pharmaceutical companies as well as the young students for both biology chemistry and chemistry biology interface now need to understand the subject. And before we become again follower, especially from this land, we should do a couple of things on our own.
So more doors open up so far as career prospects are concerned. So one should be always aware and cognizant of this particular aspect and be constantly on the lookout. Now let me change gears and ask something completely different, right? I mean, many people don't know that Gujarat probably is home to some of the top forensic universities probably, where chemistry does play a part. But when you come to pharma, forensic also does play its own part, right? So my next question is actually to Dr. Srivastava. What role do vigilance and forensic play in the pharmaceutical industry? Now, this is something very, very important from our student perspective because we keep emphasizing the bookkeeping part of it in the laboratory and academic setup. I'm sure that same is the concern in the pharma industry as well. And then how can these strategies on forensic play or in pharmaceutical industry enhance drug safety, regulatory compliance, and IP protection?
Well, good evening. Maybe again deviated from the subject and lot of research, molecule development, drug development. You all have been listening since morning. The pharmaceutical industry begins from molecule to drug development, then various stages of trials, and then it enters into industry. And then from industry, it has to move in the market. Now for market, there is a lot of problem, which I think he will comment later, who is heading the marketing in Kedla Pharmaceutical. But I can tell you another big industry is about counterfeit medicine in the country. If India has 60 billion US dollar industry, but fair research will not come about the percent of counterfeit medicine in the country. But many literature tells about 5%, 10%, 20%, no idea exactly. But that means you do not require anything related to molecule development, to research, to clinical trials, going into the market, and you can have a good industry. I have seen various places in Delhi, Haryana, Uttar Pradesh, where a lot of counterfeit drugs are marketed almost every day. Not only in India, but it goes to the other country. I mean, the counterfeit medicines are exported as well. So here comes the role of vigilance. And when vigilance will identify in collaboration with the police, whenever such cases are detected by the police, then there comes the role of forensic. We have to do a lot of investigation, we have to do a lot of forensic, be it about the wrapper, be it about the chemical composition, etc., etc. So as the industry goes, this counterfeit will also grow. So for all that purposes, the forensic as well as vigilance has a huge scope in this pharmaceutical industry. So this is an open market waiting for all of you, the forensic industry. Another important role of vigilance and all that, which is related, is pharmacovigilance, where this industry does not close everything once the product has gone into the market. But even for years after years, we have to keep monitoring about its impact, side effect, and so many things. And that part is taken well taken care of by pharmacovigilance. So there also, a lot of role has to be played by the forensic, vigilance, and other agencies. Lot of subject has been dealt here about the chemistry, biology, and everything. What you have to have about one is the knowledge which you gain from the university, from your studies. Say by education, I'm a chemist. But by profession, I'm cop. I'm a police officer. But while entering into the police job, I did not leave chemistry. To the extent that one point of time, I became one of the best explosive expert of the country. To the extent that I have done 120 plus post-blast investigation, and you take me to any blast site, I can tell you who the terrorist is behind in the blast. So this is forensic, and this is chemistry, I should say. And I got the opportunity because of this teaching chemistry with the police work. I got the opportunity to serve the President and Prime Minister of India for 15 years. As an explosive expert, I used to move with him. The government of India recognized my work of forensic to the extent that some of the blasts which took place outside India, and they wanted expert opinion, I was nominated to do the investigation. So this is all chemistry. Forensic is nothing but a scientific approach of investigation. Be it viscera, be it a murder, be it any kind of crime, where the scientific approach is required. So this field is quite open for all of you. Basic science is required, and like interdisciplinary requirement is for pharmaceutical. Same is the case with the forensic. I think that's.
No, I think that's a great perspective. I mean, it is something which we as chemists normally don't get to listen from. I can tell you from my own experience that I have participated as a chemistry faculty member in some of these forensic investigation on my own. I can't talk in public, but it's a very interesting point where organic chemistry knowledge actually comes in handy. Let me now come to a somewhat different topic in the sense that we talked all about chemistry, skill sets, what are required for becoming employable, and so on and so forth. But chemists don't get employed only in the lab, not only in the medicinal chemistry domain or a process chemistry domain. Chemistry knowledge also is useful in marketing. Chemistry knowledge is also required in the field when you talk to non-chemists. So my next question essentially is to Dr. Das, that what are the three most important factors or skill that you look for in a candidate in the pharma sector, particularly for sales and marketing?
Thank you, and to respected moderator, all faculty members, all students, thanks and congratulations for this beautiful first international conference of SIDD. And the host is Inaugural University, which is Kerala's one of the academic project, and I represent Kerala. So I'm very thankful to our CMD sir, Dr. Modi, Rajiv Modi, Master Rajiv Modi. And the name of the university is dedicated to the founder chairman and his wife, Indrashil. All these students in front of me, sitting around 200 students, I feel that this is the only session where you speak and others listen. And rest of the sessions, we speak and you listen. So as a last responder from this panelist, I'll be telling that I'll be touching the pharmaceutical industry, career opportunities for the students, and what are the prerequisites or what are the three skills setup you were asking. In pharmaceutical industry, in fact, there are three major career opportunity for chemists. I'll be telling that actually there is a very thin lining from chemists, biologists, and pharmacists. So if you see, pharma industry entertains chemists, whether they are BSc chemists, BTech, MSc, researcher, PhD. Similarly, for biologics, there are BSc biology, MSc biology, PhDs like that. And similarly, pharmacist. So that is B.Pharm, M.Pharm, PhD. So these three specialty of students or who are having the background qualification are being entertained mainly 90% of the cases of pharmaceutical industry. And there is a very thin lining. So we do not differ all three. In fact, if you see, here in Inaugural University, we come very regularly for the collaboration of pharmacy students training. And our dean sir and all sirs are there. So there the same thing is there. So if this international conference could have been also attended by pharmacy students or the biology students, it would have been the same topic and same deliberation of me what I am telling you. So these three specialities or the academic backgrounds are being treated almost similarly in pharma industry as of today. Now there are three major career paths for you or the parallel two people. One is discussed from the morning, that is the research and development. So it's a research work, development work for the drug, for the discovery of the drug, for the process patented drug, new drug, NCE, all the things. So major pharmaceutical companies who are top 10, top 20 pharmaceutical companies, they are having a very good R&D sector in every pharma, whether it is a Sun Pharma senior is sitting here, whether it is a Cadila, whether it is a Jaidas, whether it is a Intas, Dr. Reddy, every. All good companies are having a good R&D center. So there your career path will be there. And everywhere the hierarchy is maintained. You cannot jump to a senior level. So your entry strategy will be as a junior chemist or junior researcher, then there is a hierarchy. You will be going up to director level. This is one part, which has been taken 80–90% of the time today. The second part is the production. In pharmaceutical industry, production is the backbone of all products. So there also, you will be finding the production chemist or production in charges. There, the chemists are taken, pharmacists are taken, also biologists are taken. So huge career path is there in production unit also. So it is almost equal. And the largest career opportunity, which has been ignored here because it is an academic session and all researchers and scientists are there, the largest career opportunity is there in the corporate office in sales and marketing. This is the largest, it is 80%. So if I see the job opportunities, career opportunities, 10% is there in R&D, 10% in production, but 80% in marketing, sales, and strategic marketing. And that is normally not known to the students. So when we were the student also, we are not aware of it. So the largest pie is sales and marketing, which is a sales, marketing, strategic sales, and the corporate office job, including the sales of the field. So there also, we take the chemistry background, whether it's a BSc, MSc, PhD, similarly biology background, similarly pharmacy background. And there also the hierarchies are there. So there is a sales hierarchy, which starts with field executive, ends with national sales head, GM sales, vice president, director. Similarly, a marketing background person will be a product manager, group product manager, marketing head, GM marketing, and then president marketing. And strategic marketing is also there. What Dr. Srivastav has told, there is vigilance and all these forensic is another career growth. So in this entire process, I'll be just summarizing that career opportunity in pharmaceutical industry, it is there with you. 10% will be R&D, 10% in production, 80% will be there in sales, marketing, and strategic marketing. And the prerequisites of all these three are different, actually. So if you ask me what are the major prerequisites, so all these three sectors, three career opportunity, you will be getting two types of students or two types of employees we take. One is fresher, who are from college. So all of you are students. So that will be the first step. So freshers will be always treated with his academic background, smartness, communication, that's all. A family background, no other thing. Fresher will be always treated as a fresher because he has no experience. So it will be his academic background, his family background, his communication, his overall get-up. That's all. But after fresher, whether we take in production or R&D or in sales and marketing, then real challenges starts with the experienced candidate screening. There we see the skill level, the achievement of the past, and where you are fit. In sales, we'll be seeing the enthusiasm of the person, self-conviction of the person, how his total get-up is good, how his communication is good, how his interpersonal relationship is good. In marketing, we'll see that how is his innovative, how is his skill set of the previous job he has done with the development of some brand, or what is the achievement he is carrying, or introduction of the brand, or innovative strategies. For strategic marketing, we see that how the person is strategic by nature, how his brain is working, how innovation is working. But when in production it comes, you will be finding that how he has done the skill set of production, his ability of doing it. In R&D also, what are the skill sets, what are his innovations, what is his past record we see. So two kind of vision we see, or two kind of candidates we see. One is fresher, where most of you are there. All these three places, there is nothing challenges. You be prepared with good interview skills and give the interview with conviction, with the confidence, you will get the job. There is empty jobs in the pharmaceutical industry, all these three sectors. But when you go up, we see one skill, which is the trustworthiness and your longevity of working in the previous organization or your previous career. So here we see challenges with the new generation, that they do not stick to the organization for 6 months, 1 year, or 2 years, or more than that. It becomes very, very, very negative factor for the candidate. So my suggestion to all of you is that wherever you are, whatever you join, be focused. I'll be joining R&D, I'll be joining production, I'll be joining sales and marketing or vigilance. But you please join there with focus, learn, contribute, stay for a good number of time, and then go for a career growth. That's all, sir.
I think that's a very elaborate point that you have made in explaining the nuances of various factors that one should consider while taking a job in the pharmaceutical industry on different domains. Now, the last question, actually two of those, one in the last question in the first round, and then there is going to be only one question open for everybody, but I will probably toss it depending on my choice, probably, right? So last question obviously is to DS. And this question, while it is important from a student perspective, whenever we talk about industry and academic participants together, the burning question or elephant in the room most often is industry academic collaboration. So I'm going to give you a blank check, and what are your thoughts on industry academia collaboration? How we can enhance it? What are the things that one can do on either side to take this issue further? Because it has implication eventually for the students as well.
This is one of the boring topic. So we have at least listened to this industry academia collaborations for many, many conferences, and we only talk and go. And most of the cases we talk and we forget about it. Again, we'll start fresh discussion in meetings, and I'm sure myself and we both are there in a few yeah. So I think now things are changing, of course. There is a hope, but I think, see, right now we are the way we are looking at now. Look at like we are talking about that Viksit Bharat, right? Viksit Bharat 2047, 100 years of our independence. What our government of India is doing. India is looking forward. I think we all are going to play a major role, particularly younger generation. You all going to play a major role in that. I think there's a lot of opportunities. I think you need to start thinking what you exactly want to do it. Again, is industry or academic job is again second thing, I mean, like which sector you will take. But again, collaboration is something personally, since I worked in industry, I know what are the like advantage, disadvantage working in industry and what industry looks from academic point of view. What and at the same time when I come to academia, I also look at what industry looks forward. I think both ways you need to analyze. I think one should get experience in both places and what. Dr. Yadav sir was also telling, get experience in industry. That actually I like that statement, but again, don't stick to industry alone. Again, come back to academia and learn both sides. And 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 again, you again both have advantage, disadvantage. I don't think we can keep on discussing about this one. I think look at the opportunities, and already 6:47 now. I think we'll move forward. But again, coming back to my simple suggestion is to the students when we started this discussion, I think the backside rows are empty. So now started feeling, I was wondering what is happening. But like, are they really interested in the discussion? I'm not sure that. I think they're looking for cultural program. I think there are. So no, I think no. Coming back on a serious note, I think opportunities for industry academia collaborations are huge, and things are changing. Particularly from Government of India, there are a lot of schemes. And look at our Professor Santosh Garpure. Like he is heading that SIAN in incubation center in IIT Mumbai, and like that we have NCL Venture Center in Pune, and I think there we are working very closely with the students and with the faculty. And if you have idea, we can approach them. And also, I'm sure in either these places also we have a lot of opportunities. We can move forward. I think there's a lot of opportunities in the future. But we're not moving forward.
Yes, you made very important point. That's the point I was trying to elaborate, right? If I were to paraphrase Harry Potter movie, Dumbledore's dialogue, right? Help is available to anybody who will ask for it. Today, that is the situation
Right.
that we have fortunately in our ecosystem. That anybody wants to take up entrepreneurship, there is help available. There are incubators like SIDD who are available in the country, and they are open not just it's not IIT Bombay exclusive club. It is open for anybody who comes with good idea, and then they will be able to get all the.
No, just 1 second. So I can tell you, like right now, Government of India running. I'm sure you must have heard about this 1 lakh crore, 1 lakh crore, right? Yeah, that's what. Yeah, there's a lot of opportunities, like in 20,000 crores also. That is another one project. And another one is in Department of Pharmaceutical, 5,000 crores. They want to spend on this kind of industry academia collaborations, and they want to give loans and like in-Mitli program. Similar model, they are going to take it forward. There's a lot of opportunities, I think, for startups or for young faculty. Why young faculty? Even like Dr. A.V. Ramarao, at the end of his, like after ending his career, he started his startup company. I don't think that startup is only limited to age. I think we can do many things. Yeah, go ahead.
Going up. So what numbers have gone up? Right? The numbers have gone up. I see more girls now there than they were earlier, right? And that is what my last question is about. And I'm going to first toss it to you and then maybe take it to processing, because you are the current director of an institute. I want to put you on the spot. If you look at gender diversity in science, particularly chemistry, numbers have started improving at the student level. When you and me, we were PhD students, largely male-dominated area, but today you see that almost 40–50% of the students are women. But we don't see that translating. I mean, just look at us, right? Not even a single women faculty member or from industry who is sitting here on the dais, right? That is not speaking very highly of our status today. So what we need to do as a policy, what we need to do as certain measures, where this translation of students coming in but not going on to the higher positions, higher career aspects from the women side as well. What steps can we as community take, and administrators as well?
So from administration side, I can talk about it. Of course, we are encouraging, we are trying to hire wherever possible more women faculty or women basically any other positions also, any administrative position also. We are trying to give whatever possible way. But coming back to, I think students, I think number of students increased definitely in, I can tell you in my own institutes, now about 40 plus student population, student research scholars are women, particularly there is a chemistry dominant institution. Earlier, mostly girls used to be there in biology, at least life sciences areas, but now we can see that's a good trend. I think I'm sure I'm optimistic about it. I think things are going to change in the future, and I'm pretty sure. I think if even like men at home or parents at home, if they can support more girl students to take up careers and moving forward, I think I'm sure more people will come into the science, and I'm sure they'll also come up to the leaders. I think things are changing. I can tell you my own CSR in students now about 7-8 directors are women directors. You don't tell me that they are not there, but we have, even including our director general is a woman director right now. So there are their positions. Don't look at only this and make judgment. Okay. Okay. Thank you.
Don't have eight women director, if I'm not mistaken. So, you want to take the last dig at that particular?
In fact, anyway, I'll let's speak in short. I was going to tell the same thing what you already noticed, that none of the ITs and ICERs, or I don't know about an IT, have any director. Anyway, that is not important because things are changing. But you have to go to Genesis. Large number of people want to be in comfort zone. My question is, why two people have to be together if the career is the priority? And people, quite often I have seen people coming for job, one person is giving application of the other person, spouse. If spouse needs a job, that person should apply and give a biodata. But it always happened the other way. I myself gave a large number of jobs to women, especially non-teaching. Faculty is very complicated hiring, so it's not that. But I refuse to give child care leave because leave is not anybody's right. You have a proper leave. I still believe that 6-month maternity leave is too much in India. Beyond that, become very difficult. So I think if you want to be in a comfort zone, then you cannot have a career. If you want to have a career, don't be in a comfort zone. People, you know, your own former director, Devang Khakkar, case, somebody lived in one country, the other person lived in another country. So it has, balance has to be worked out. But as Srinivas mentioned, things are changing at a faster rate.
Okay, so on that optimistic note, I think things are changing and changing at a faster note. Okay, so now since student session and student question. Only one question. Last. Yeah. Okay, so maybe the student who has asked the question can stand up and ask that question. Okay, I'll try to read it. It's a very pertinent question. So it is about salary package for encouraging people to look for industrial jobs. It's related to salary, but basically what is the question, if I understood it correctly, is IT pays big amount of money, pharma doesn't pay much money. So how do you remove that disparity so that if I were to paraphrase the question, let's make chemistry great again even on the pay packages. That's what it essentially means.
We are saying IT pays more.
Yes.
You mean pharma pays less?
Much lesser. That's what is the question essentially. I'm not very sure.
Sir, I'll give the quick response of this as I represent pharmaceutical industry. Students' response is partially correct. Pharmaceutical industry does not pay much more like the industries like IT. Pharmaceutical industry will give a stability, actually, the job. The stability of the career graph. It is an
Первое
evergreen
правило
industry, but
клуба —
it grows.
не упоминать об
But if you
антисоциальном
see the
клубе.
banking
Второе
industry,
правило
IT
клуба —
industries, and other
не
industries,
упоминать
sometimes they pick
нигде об
up, they come
бойцовском
down.
клубе.
Sometimes there is a
Третье
huge
правило клуба —
attrition, and
боец
sometimes there is a
крикнул
removal
«Стоп!»
of people.
Выдохся,
Pharma
отключился.
industry will not
Бой
give that
окончен.
shock, but there is a sustainable growth. And when I was talking and referring to you, yes, pharma industry, again, there are three types of salary. Production, less salary. R&D, more salary. Marketing and sales, best salary. So you choose your profession.
So at the end of the day, it is a profit from which you get your salaries. And pharma industry is tightly regulated. In this country, the margins are very low. So profits are low compared to turnover. So if you look at a turnover and profit margins, IT is a maximum in this country and pharma is a minimal. And that's where the salaries are coming from.
Если не стелить свои чувства, а разбитые сердечки.
And sometimes pharma job is more stable. I'm talking from job perspective. So you will end up having
You should give correct message actually. We should not look for salaries where you pay more, or where you have more satisfaction, where you can contribute more. That is, I think that is also you should take it in when you are deciding your career. Don't look at only salaries.
That's a perspective side, but that's a perspective which I was trying to represent from the student side. And I think that's a very, very valid perspective. When I talk to students around, I get distinct message every time that chemistry is not as attractive as career option. And that's what I tried to kind of put forth today in this last particular question, because that's a constant message we get it. Your point is well taken. That's probably my view as well. But you can't ignore the reality of the numbers that get reflected on a paycheck day, right? So that's something which is all of us need to kind of think about. So let me thank all the panelists for the passionate views that they have put forward. And thank you, audience, for the patient hearing. And thank you, organizers, for giving us all the opportunity to participate in this panel. Thank you. Thank you, everyone.
Thank you everyone for making this session very interactive one. Just a last bit of giveaways, I suppose.
Now I request Dr. Chandra Kumar Apaya to come on the stage and felicitate our session chair and moderator, Professor Gharpure and Dr. Sri Ram Gundi Madam. Dr. Apaya. We are thankful to all our dignitaries. Before the conference, we boosted a lot that we are going to bring so many big experts and we are going to have a really stimulating session. So we thank all of you to come over here and give us a very comprehensive guide and for a very stimulating student interactive session. Thank you so much. Now I request all of you to please remain seated because we are going to have one very nice cultural program students have made prepared for you. It's very compact, so kindly be seated. Thank you so much. Now I request my student Asima to come and take the stage. I request all the student participants to remain seated, please. We will have a quick cultural program. We request faculty delegates also if they can stay back. Thank you.
Good evening to all dignitaries, chief guests, and all my dear friends. A warm welcome to all of you to this wonderful evening filled with culture, music, and dance. Gujarat. It is a land of vibrant colors, rich heritage, and unmatched traditions. From the beats of Garba to the flavors of Dhokla, from the shores of Dwarka to the grandeur of the Run of Kutch, Gujarat is a true celebration of life. Gujarat is a state where history and modernity walk hand in hand. Tonight, let's take you on a journey through the heart of Gujarat, where every color, every dance, and every melody tells a story. So let's begin this mesmerizing evening. Let's begin it with a mesmerizing inaugural dance by Sushmita and Khushi. The energy will set the perfect tone for the evening. Give them a big round.